Morning, everyone. Morning. We're going to start shortly, so if you'd like to take your spaces. There's people joining us online, so we want to start punctually. How's everybody? Wednesday's hump day. You over the hump? Any runners here? I don't look like a runner, but I am a runner. You know, when you run a marathon, you get to about 25 Ks into the 42, and you just run out of energy. And then if you carry on, you get what they call the second wind. Huh? Suddenly things start coming back into place, but you've got to press through. They also talk about hitting the wall. Do you feel like you're hitting the wall this morning? Anyway. All right, so I'm Tony Hawkridge, as you heard yesterday. So I'm the chair for this morning, the first part of the morning. And we've got 3 talks lined up. But before I get to that, we've got 2 people who've joined us after the first day, and I just want to give them a chance to say hello and tell us who they are and where they're from. So the one is Mercy, is that right? Mercy, would you like to, is there a roving mic? Just to say who you are and where you work and...
All right, good morning, everyone. My name is Mercy Mwausega Yohani. I'm from Malawi. I'm working with the Ministry of Health. I'm a senior nurse midwife. My main focus is on maternal, neonatal, and adolescent vaccines. Thank you so much.
Lovely, and welcome. Welcome to Cape Town.
Thank you.
We organized this wonderful weather, especially for you. And the other one is David. Is David not with us yet? All right, so he's probably still having a bit of breakfast. Okay, so we've got 3 talks this morning. The one is on the mRNA hub, which I think came into its own with COVID, but I'm not going to steal any of Pierre's thunder. And then we've got a talk on meningococc, and we've got a talk on birth dose Hep B, and then it'll be tea time. So the first talk is by Pia Steigler, and she's from African Biologicals and Vaccines SA, Pia. And she's going to talk on advances in local and regional vaccine manufacturing capacity, experiences from the mRNA hub, Afrigen. So welcome.
Hello everyone. So yeah, I'm Pia Steigler. I'm the senior scientist in immunology at Afrigen Biologics, and I will give you an update about the recent advances in local and regional vaccine manufacturing based on the experience at Afrigen. The global vaccine manufacturing space is centralized in just a few regions. So at the moment, over 70% of global vaccine production by value is just from 4 companies that are located in high-income countries, particularly in the US and in Europe. In contrast to that, over 70% of global vaccine production by volume is produced in developing countries, particularly in India and China. This discrepancy in the vaccine manufacturing space was also reflected in an unequal access to COVID-19 vaccines in the recent pandemic. While many people in high-income countries, such as the US or in Europe, already received their second dose, many people, particularly on the African continent, had yet to receive their first vaccine. So there is a growing consensus that we urgently need to geodiversify the vaccine manufacturing space. So the vaccine manufacturing capacity of Africa is very limited. At the moment, we are actually only producing 1% of our current vaccine requirements. That means 99% of all the vaccines that we are having in Africa is imported. but we are consuming 25% of the global vaccines by volume. To change that, the African Union Partnership for African Vaccine Manufacturing was introduced in 2021 with the aim to produce 60% of local vaccine needs by 2040. In 2021, we just had 7 vaccine manufacturing sites on the whole African continent. And the majority of them are only able to fulfill certain parts of the manufacturing process. Many of them were specialized on downstream processes, such as fill, finish, packaging, which means that the products still need to be imported from other countries. The reason for that is that the vaccine manufacturing is a very high-risk undertaking, particularly the upstream processes where you have to produce active ingredients. Much less risky are downstream processes such as fill finish, packaging, lot release, and shipment and distribution. In general, making a vaccine is a very complex journey that is costly and time-intensive. So of 100 candidates that enter the early drug discovery phase, only 1% make it onto the market. And it usually takes 10 to 20 years for a vaccine to get onto the market. But the COVID pandemic showed that it can be different. The COVID or SARS-CoV-2 sequence was posted online onto the internet in January 2020. Within days, 350 laboratories committed to produce a COVID-19 vaccine. Within 1 week, the first laboratory had a nucleotide-based vaccine, which was based on mRNA or DNA. Within 3 weeks, the first laboratories had a viral vector vaccine. And within 2 months, we had the first protein-based vaccines. And just a few months later, the first vaccine candidates were making it into clinical trial. And by December 2020, we had our first COVID-19 vaccine on the market. That is just 10 months after discovery of the sequence. And the first vaccine that made it onto the market was an mRNA vaccine. So during the COVID pandemic, we found that mRNA vaccines have a huge advantage. They can be rapidly developed, which makes them ideal to respond to pandemics or disease outbreaks. They're easy scalable. You can quickly go up from a lab scale to hundreds of liters. And they work according to a plug and play principle. That means once you have set up the platform, you have a construct, you can quickly just exchange genetic information to fit it to a new disease target. And the production is relatively low in cost, so facilities don't have to be very huge. The vaccine itself has also many advantages. It's non-infectious, which means it's good to be used for immunocompromised individual, which is important for low and middle income country where there's a high rate of co-infection with HIV. During the COVID-19 pandemic, it was applied to millions of people, and it was shown that it has a high clinical safety profile. The side effects were low. mRNA vaccines also do induce a robust humoral as well as cellular immune response, which make them ideal to be used for versatile disease targets, including viral diseases, bacterial diseases, or potentially also parasitic diseases. So for that reason, the mRNA vaccine was selected to be suitable for a technology transfer. So the WHO and the Medicines Patent Pool founded the mRNA Technology Transfer Hub to improve the health security in low and middle income countries through sustainable regional production of mRNA vaccines. This technology transfer works according to a hub and partner model, which means that one facility is developing all the technologies, developing the platform, and acquiring all the knowledge required to produce an mRNA vaccine, is then transferring the knowledge and all the skills to the different manufacturers so that they don't have to start from scratch again, so that they save time and cost. So, and the hub was awarded to Afrigen Biologics. So Afrigen Biologics received a lot of investment from local and international funders, received a lot of guidance, expertise from different experts and researchers around the world to build up a platform to produce mRNA vaccines. And Effigen Biologics is transferring or teaching or training in total 15 different partners in different countries around the world, from low and middle income countries that are distributed around 4 continents. And all these countries combine a population of 3 billion people, which makes 40% of the global population that can be supplied with vaccines. So before I go a little bit further into the advances that we made at Effigen Biologics, I wanted to tell you a little bit more about Effigen Biologics and its history. So in 2021, Effigen was awarded the Technology Transfer Hub. And since then, Effigen is quite well known. But Effigen was already founded in 2014-15 with the aim to strengthen local vaccine capacities and capabilities. In 2017, Effigen was the first adjuvant and formulation center in Africa. And Effigen is still today producing adjuvants and formulation for wellness products, for healthcare products, for animal and human use. So for those who haven't seen Effigen Biologics yet, Effigen Biologics started in a warehouse in an industrial area. So at the very beginning, Effigen was just one single warehouse, and then we expanded rapidly. Now we have 5 warehouses, and again, we are running out of space. So in the future, we probably acquire a few more warehouses. Effigen Biologics has a unique position on the African continent because it will build end-to-end facilities, meaning that everything from start to finish to produce a vaccine can be done in one single facility at Effigen. So therefore, we had to set up a quality management system so we can validate equipment, we can quality control, the material handling. We have an own research and development unit where we can work on new vaccine constructs and also improve the current vaccine. We have a manufacturing site. Our GMP assessment will be at the beginning of next year. Then we have an in-house quality control where we can assure that the vaccine product that we are producing is of high quality before we then fill it, pack it, and ship it to its destination. Here you can see an overview of our GMP facilities. Our GMP facilities are over 2,000 square meters large, and we can do everything. So from the starting material, which is the pDNA, to producing the drug substance, the drug product, and we do the fill and finish in these areas. We also do the quality control in the GMP facility, and we also have a stability room. And this stability room, we store the vaccine that we have produced at different conditions, such as at room temperature, as -20, -80, for an extended amount of time to see how stable our product is, which also guarantees good quality for long term. Then we also have an R&D center where we work on new vaccine construct for new disease targets. And apart from working on new vaccines, we're also working on new formulations to make the vaccine that we currently have more thermostable. As you probably know, mRNA vaccines do require cold chain storage, so they need to be stored at really low temperatures of -20, -80 degrees, which unfortunately don't make them suitable for low and middle income countries. So to produce mRNA vaccines, we are building 2 processes. So one of the processes that you see here is the conventional process. So broadly, the manufacturing process can be divided in 3 different steps. So first of all, we have to produce our pDNA, so our plasmid DNA, which serves as a template for the mRNA. Then the plasmid DNA is transcribed in an in vitro transcription reaction to mRNA. This mRNA needs then to be purified, kept to prevent the degradation of the mRNA. Then it gets further purified in many, many steps to guarantee that it's clean, that we don't have any remaining enzymes or any remaining products. And then it gets formulated in a lipid nanoparticle. Then we talk about the drug product, which then again gets purified to make sure everything is clean, no remaining unwanted reagents are in the vaccine before it then gets added with sucrose, which guarantees the thermostability of the vaccine, and then it can be filled and finished. So with this conventional process, which takes about 2 to 3 weeks, we can produce 25,000 doses of vaccines. And this process is not very fast, so 2 to 3 weeks is still a relatively long time. But this conventional process allows us dual sourcing of raw material. That means we can get material from different vendors or distributors. And that is very important for low and middle income countries, because often, as you probably know, there are long lead times, there's always a backlog in orders, there's shortage of stock. And the majority of the material is still coming from overseas, like the US or Europe. So it's important to not delay vaccine manufacturing and be able to source material from different vendors. We also have an automated process. That automated process is faster, but it also comes with disadvantages. Again, this process is divided into 3 different steps. So first we have the plasmid production, then the production of the mRNA, and then the formulation. The difference of this process is that the mRNA is produced in this machine. And this machine is provided by a Quantum, which is in Belgium. And it can produce the mRNA within 1 single day. So we're overall saving about 4 to 5 days in the production process. And it produces the same amount that the conventional process produces. It has a small footprint. The machine you see here is about 2 meters big. And there are even smaller machines if there's less space available. But the disadvantage is that we need all the reagents from this one manufacturer. Meaning, if this one manufacturer has a shortage of supply, if there are issues with deliveries from Belgium to South Africa, our vaccine production would come to a halt. And for that reason, we have 2 different processes to guarantee, in case we have any issues with the automated process, we always have a backup option. So when I said we need about 2-3 weeks to produce a vaccine, that means actually that we have optimized the process already. So making vaccines is really costly. So before we go into the final batch size, which is 1 liter, an IVT, we start very small. We start with 100 microliter scale, test the quality to make sure it's of good quality before we then move on to the next scale, which is just a 200 microliter scale. Then again, we have to test whether the vaccine is of good quality before we move on to the bigger scale. And eventually we reach the larger scale, which is the 1 liter scale. And here you can see the list of all the analytic testing that we have to perform to make sure that the vaccine is of good quality. So on the left side, you can see all the tests that we do for the drug substance, so to ensure that the mRNA is of good quality. And here are the tests that you can see that we do for the drug product, so for the mRNA LNP. So I'm not going through all the assays, but I just want to highlight that these are a lot. It's a lot of work, and it takes like 3 weeks to just quality control your product. Apart from looking for physicochemical quality, we also have to ensure that our vaccine works in vivo as we want it to. So we have to confirm immunogenicity, that our vaccine is inducing the immune response that we want, such as antibodies that are specific against the disease target or T cells. And we have to make sure that our vaccine is actually safe before we go into human clinical trials. So at the moment, the majority of our animal models is still outsourced. So our hamster model, our non-human primate model is done in France at the University of Marseille. Our GLP toxicology model that was done in rats was done in India at Agile. But our aim is that we build up a network of preclinical studies here in South Africa to be completely independent. And we're working together with the University of Cape Town, who have already worked on our mouse models and is currently setting up our hamster models. And we also want to set up a GLP toxicology hub. And as you can see here, we have a lot of collaborators to do that, because GLP toxicology studies are very difficult. So you need a lot of testing, and they all need to be strictly regulated. So at the moment, we're building a manufacturing facility, but that is not enough. At the same time, we have to improve the formulation. So some of you maybe know that we base our platform on the Moderna COVID-19 vaccine. So the lipid nanoparticle we're using is actually still Moderna's, which means it's IP protected. So we at the same time work on a new formulation to try to have the freedom to operate, to produce our products without requiring a license from Moderna. Then we want to further improve our mRNA vaccine. We want to make it more potent, to improve protein expression, and also to minimize reactogenicity, to minimize side effects. And then we look for different disease targets that are relevant for the local context. And we're also working on alternative delivery methods. So there is unfortunately the fear of needles, and we are now working on a patch that would require no needles for immunization. And to do all that, we need a lot of help. So Afrigen has established a lot of collaborations to improve DNA and RNA development, look for additional disease targets, improve their manufacturing technology, look for alternative delivery methods, and also look for alternative LNP formulations. And all of these collaborators are sitting all over the world. We have basically collaborators on every continent, North America, South America, in Europe, in Africa, in Asia, and Australia. So currently we're working on several disease targets. So our pipeline includes vaccines for TB, RSV, gonorrhea, HIV, and Rift Valley fever. You might remember the slide I showed you. In 2021, we had just 7 manufacturing facilities. Just 3 years later, 2024, we are now having 25 manufacturing sites. So within such a short time, we made huge progress. These manufacturing sites are all at different stages still. Most of them are still to receive their technology transfer to be producing vaccines, but we are getting there. But building manufacturing sites is not enough. So that is not the only thing we need. We need to collaborate with each other. We need research institutions feeding us new antigens. This is a usually very long process that can take years. Then we need to further improve the vaccine design. We need to set up a preclinical network in South Africa. And we need to establish clinical testing sites that are able to do a lot of phase 1 testing. Then our regulatory needs to mature to be prepared for mRNA technology. And we also need to update our formulation, which is currently IP protected by Moderna. Then we further need to improve the manufacturing process. We need to optimize technologies to be more efficient, which means we can make the production more cost effective. We also need local, reliable GMP certified suppliers. So at the moment, we source our material from overseas, which of course comes with lead times and additional costs. Then we need local service support. Our equipment also mainly comes from Europe, from US, which means if anything happens, if something breaks down, if we have an issue, we often need to get someone down here, fly them in, which delays again the manufacturing process and also is an additional cost. Then once we have the vaccine, we have to produce them at a commercial scale to be able to supply African people. The vaccine needs to be cost-effective, suitable for low and middle-income countries. We need to have an infrastructure to distribute the vaccine. And of course, the vaccine needs to be administered to people that need them. So the work I showed you was based on experience on Afrigen, but all of this is only possible because we had a lot of support from local and international researchers, funders. And thank you.
Thank you so much. I think comprehensive but concise. Wonderful. A lot of information there to digest. I'm sure people will want to go back and have another look. We've got about 10 minutes for questions. So let me start off with one. Today's, I'll come to you now. Today's, as you all know, they're busy counting votes in the US. And I think it was in the last year of Donald Trump's previous presidency that they produced the the mRNA vaccine. He likes to take a lot of credit for it. But my question to you really is how important is political will and political backing to get this kind of thing going? But let's take a couple of questions and then maybe you can answer them. So go to the room first. I saw a hand there, and then I'll come to you. Okay.
Hi, thank you for the wonderful presentation. I think it's lovely to see that there's more production happening in Africa. So that's very encouraging. So my question for you is, we saw during the COVID-19 that they did not want to share the vaccine designs and formulations from the high income countries. So what measures have you and your collaborators and stakeholders put in place so that in case we do have a pandemic next tomorrow, you know, you have these production places and whatnot, how will you make sure that once there is a formulation from high income countries that we are able to get it and also use it to mass produce our own vaccines in Africa?
Yeah, I think you mentioned an important point. So we unfortunately did not get any technology transfer from any of the big companies. So whatever we have set up, we set up based on public available literature or based on knowledge from other experts that supported our technology transfer. So whatever we set up now is basically own generated. And this knowledge will prepare us for the next pandemic. So we have set up already the platform. It needs to still be GMP accredited, which hopefully happens next year, beginning of next year in March. And yeah, based off that, we can produce own vaccines, and we have the research and development center as well to work on new disease targets. So as I mentioned before, the mRNA technology allows that once you have your platform set up, you can quickly change the genetic sequence to a new disease targets. So you can really quickly adapt if there's a new outbreak and produce vaccines. Yeah.
Thank you so much for the opportunity and for a wonderful presentation. I would like to ask two questions. The first one is with regards to the patch vaccine. So I want to understand, because with different modes of administration, there's always have to an alteration to a dose or the strength of the particular active ingredient. So in this case, we are looking at the antigen. So did you alter as well? Let me say, for example, just giving an example, then with BCG, we've been doing the injection, and then now let's say we are moving to the patch. Did you also alter like the strength of the antigen? And then I also want to understand also the onset of action, how quick is it with compared to the IV as well? And then the second question that I want to ask, you also mentioned something about that, okay, we have so much sites that develop and manufacture vaccine in Africa. So I want to also understand that, do you think that we have enough capacitated like personals that are very much capable with, in as far as the production and the development and the manufacturing of vaccines are concerned? Thank you.
Okay, to your first question about the patch. So the patch is still in early development, but we will have to adjust the dose. So at the moment, the vaccine is given intramuscular, and the dose of the patch will not be the same, but we will have to define the dose. We will do animal models and test different doses to find out which dose is suitable. Then to your second questions, yes, we have more manufacturing facilities, which means that we need more skilled people. So at the moment, yeah, we need to train a lot of people. There's a lack of skills, which is natural because the manufacturing space hasn't existed here. People are not trained in these technologies, but we are constantly working on training more people. There are more programs, such from UE Bios, where people get trained in biotechnology and to be trained to work in the manufacturing space.
Thanks. We're going to take a couple of online questions now.
Yep. Pia, over here. So we also have our colleagues online who are actively following your talk, and they all say thank you very much for it. They really appreciated it. So I thought we could take two questions online and come back into the room. So the first one is from Muhammad Hamayun, and he's asking why mRNA vaccine platforms. And his sub-question is, why are there no such vaccines against HIV? But as you mentioned, you know, there is some work around that, and we also have a talk later after tea on HIV vaccines. But could you touch on why this focus on mRNA vaccine platforms?
So yeah, the mRNA vaccine platform has shown to be promising because it is very quick to produce a vaccine, and it is suitable for low and middle income countries because the facilities can also be relatively small. So as you saw the machine for the automated process, this machine is 2 meters big, which means your facility can be really, really small. There are even smaller machines available, which are just 1 meter. And the facility size is actually a huge cost contributor. So overall, it's the time, it's the cost saving, which makes mRNA suitable, and also the flexibility, so you can really quickly change from one disease target to another.
Thank you. And then the second one is from an anonymous attendee, and they're asking, what are the selection criteria to start producing vaccines for a particular disease, considering the unique African context?
Yeah, so we do have recommendations from the World Health Organization which diseases are relevant. And based on their recommendation, we do select vaccines for the African continent.
Thank you.
Hello. So I think this is something really good in terms of producing on the African continent. But I worry a bit where our brothers in Asia, like Serum Institute of India, they can produce this same vaccine and the cost comes down to say $1. We do it on the continent, we are just starting up, your cost ends up being $8. What is the cost effectiveness? Because why should we buy $8 per dose and SII in India can do it for $1? How are you trying to navigate those kind of difficulties and ensuring cost effectiveness? We also get the market from our own continent.
Yeah, costs are, of course, an important point for low and middle income countries. And at the moment, our vaccines are probably more costly than the ones produced at India. But we are working on improving the technology, and this is a step we have to do. We have to start somewhere. The vaccine costs will be probably higher. We want the South African government, for example, to support us by saying, okay, we buy your vaccine, even if it's slightly more expensive, so that we can be sustainable and at the same time work on reducing the cost by improving the technology.
Thank you for the presentation. I have about 3 questions actually. The first one is on the 25 manufacturing sites that you Afrigen have set up. Is it they were set up by Afrigen in those countries, or you gave them the rights to produce with your technology? That's the first question. The second one is that I was actually talking about this with my colleagues last night, that would it not be, because as it is now, it seems like almost everybody wants to produce, wants to manufacture. Would it not be a better strategy to select a few that can manufacture and then others following along the chain of vaccine production? Maybe some can distribute, some can do this, some can do that. Not everybody producing. That's the second one. And the third one is in the technology that you use to produce, before filling the vaccine into the vials, what is the anti-contaminant that is generally used for the mRNA vaccine? Thank you.
Okay. So your first question, can you just repeat it again? Remind me of your first question.
Okay, I was asking whether when setting up the 25 manufacturing sites, where you...
All right. So yeah, these 25 manufacturing sites are not set up by Afrigen. So Afrigen is the hub which develops the technology, and the technology is transferred to the other manufacturing sites, but that is facilitated by the WHO and the Medicines Patent Pool. So Afrigen is not establishing any of the other hubs or manufacturing sites. And they're also not all focusing on mRNA. So we also have different manufacturers that focus on different technologies. So not all of them do mRNA only, but many of them try to diversify. For example, they use protein-based vaccines plus mRNA. And there was a last question, right? Was it about contamination? So yeah, you saw the process of purifying the vaccine. So it's very thorough. We do purify a lot, and we do a lot of analytic testing to make sure that there are no contaminations.
Thank you very much for a very insightful talk. Towards the tail end, I think you really hit the crux of the matter, because everything cascading from upstream ends up downstream. The vaccines need to get to those who actually need them, right? Now, seeing the reluctance as shown during the COVID-19 pandemic of the populace to accept mRNA vaccines, and since this is the product you're pushing, what are you doing in the areas of community engagement, vaccine confidence increase, making this beautiful technology known? Because today, thank you so much. You've given me a better understanding. Even as a scientist, I had my own reservations about mRNA vaccines. So what are you doing to make this kind of a knowledge available to the layman on the street? Thank you.
Yeah, that is a good point. mRNA technology is new, so we don't have a lot of long-term experience. There's always the question, can it have any side effects in the long term? But so far, there are no evidence. And we do need to educate people. We need to have advocacy programs that educate and facilitate the knowledge that we have in science to make it understandable and explain what the risks are.
Okay, are we gonna?
Yeah, so Afrigen itself doesn't run advocacy programs, but there are a lot of stakeholders that do.
Let's take two more online, and then I've got one here, one there, and then I think we're going to have to call it a day. Yeah.
Yeah.
Nadine?
You're going to have to come back again, Pia, because so I think quick questions from online. Al-Haj Abdullah would like to know what African governments need to do to scale up productivity, seeing as we now have 25 manufacturing, units, but we're only still producing just under 1% of the vaccines we need. And then secondly, any points on AAFIs associated with mRNA vaccines that might be different from the standard vaccines that we already have in place?
So yeah, government definitely support is really important. So producing a vaccine is only one part of the equation. We need regulatories that approve the vaccine. And for that, we also need regulators that understand mRNA vaccine technology and everything around it. So we need to mature regulators, specifically on the African continent. They still haven't reached level 4 maturity yet. So we have to work towards that.
Okay, but one here. One.
Thank you for your wonderful presentation. So I have two questions. So the first one, do you have any WHO GMP? If no, did you plan to get it? The second one, did you provide any manufacturer for antigens? So if yes, how does it been managed? And did you ensure any tech transfer for vaccines in somewhere in the world? Thank you.
Okay, so if I understood it correctly, you asked about GMP readiness. Was that right? Did you ask about the GMP readiness? Yeah. So yeah, we're preparing for being GMP audited. So our audit is at the beginning of next year in March. And then we hopefully are GMP ready and able to produce.
All right, thank you. I might have missed this, but you mentioned something about thermostability. What are the storage conditions of the vaccine, seeing that we have a serious cold chain problem in Africa? And then the second question is on the production. Said about 3 weeks to start and finish production, and then 3 weeks for QA. Are those 3 weeks the same 3 weeks, or they're different timelines?
Yeah, so the cold storage is, of course, a big issue still for mRNA vaccines. At the moment, we need to store them at -20, -80 degrees, which is very costly if you want to transport mRNA, and a lot of low middle income countries are resource limited and don't have the facilities to store the vaccine. So we are working on improving the thermostability. So we are working on lyophilization technology that we are able to store the vaccine at room temperature, and that makes it suitable for low and middle income countries. And then to the analytics, yeah, the analytics comes on top after production. So after we've produced the vaccine, which takes 2 to 3 weeks, we then do the analytic testing, which can take another 3 weeks.
Thanks. Okay, last question.
Thank you so much. I think this is the part of my question. So yesterday we heard that acquiring a vaccine is cheapest than anything else, or producing the vaccine, because there is a lot going downward. My other sister also said about that. So when you guys are trying to have many countries to produce vaccine for Africa, is there any plan that really would ensure that the vaccine reach the destination and those vaccines are safe? Because if the conservation of the vaccine is not appropriate, the quality is not going to happen. For example, regardless of natural disaster whatsoever, if you take a country where it's like hot and there is less people, we have less people that can deliver that vaccine. Is there anything that you guys are putting back associated with production of vaccine that, okay, we'll have this amount, but this is what we need to do in order to increase the human resource? That is first. My last question is that the production that you are saying is true, that is 25 countries, and you said that the regulation is not South Africa that give, but how are you guys trying to control, or is there any regulation that is going to make sure that not each country decide to manufacture their vaccine, or is there any way without the transfer of regulation? How about just having someone that in each country can have the knowledge about conservation, following up to assure the security of vaccine? Thank you.
Yeah, so to your first question. So yes, we test the stability of our vaccine product and we make sure how long it is stable under certain conditions. So for example, if you leave the vaccine at room temperature, it doesn't mean it automatically degrades. So you still have a certain amount of time. And based on our research, we know the amount of time it can be at room temperature or at different temperatures without having any negative effects, without degradation. At the same time, when you ship a vaccine, you have a temperature monitor. So you always know that the vaccine was at the right temperature to make sure, okay, while it was on the way for a few days, the temperature remains stable, so the vaccine is of good quality. Then to your second question, so there is, so I'm not in the public health field, but there is, you might have heard about EMA. So EMA is set up to coordinate the regulators in Africa to make sure that they all work according to the same principles.
Wonderful. Thank you, Pierre, for a wonderful lecture and also for taking lots and lots of very difficult questions. It must have felt like you were defending your PhD before all these professors. So let's give her a big hand. Our next speaker is Lauren, and she's going to talk about meningococcal vaccines, progress, challenges, and opportunities in Africa. I thought because she comes from a French company, you know, you must pronounce her surname with a soft G. So I was going to Gerety, but she says, no, it's Gerety, Lauren Gerety. So welcome, Lauren. Over to you.
Everyone. Well, I'm a bit tall for this microphone, so I'll speak loudly. So morning, everyone who's present, everyone who's online, and then to the faculty of VACFA, thank you for the opportunity to present. So as mentioned, my name is Lauren, and it's okay. Thank you. It's the joys of being tall. And I'm presenting on meningococcal vaccines today, progress, challenges, and opportunities in Africa. Oh. Oh, thank you. Okay, so I'm going to start off with a quiz because I've noticed from this week you guys are very interactive. Okay, so it's going to be I'm going to read you what the question is and then the potential answers. Don't raise your hand until we've gone through all of them, and then I'll go A, B, C, and then you raise your hand. All right, so the first question has to do with epidemiology. So the question is, which environmental factor significantly contributes to the seasonality of IMD, or invasive meningococcal disease, in the African meningitis belt? Is it A, rainy seasons that increase water sources? B, low humidity and airborne dust during the dry season? C, humidity levels in urban areas? Or D, proximity to coastal regions? Okay, hands for A. Okay, hands for B. Okay, hands for C. Salim, was that a hand? Oh, was it a this? Okay, and then D, proximity to coastal regions. Okay, well done, everyone. So you are correct. The answer is B. All right, so we're going to come back to that a little bit later because it plays a very important role in having very dry, dusty conditions is strongly associated with the epidemiology of meningococcal disease. All right, question 2, vaccine types and mechanisms. Okay, this question is a little bit challenging. Given that this week we've spoken quite a lot about conjugated vaccines. So my question here is, why are protein-based vaccines used for meningococcal serogroup B instead of polysaccharide conjugated vaccines? Okay, so the answer A is group B, polysaccharide, capsular polysaccharide, my apologies, is highly immunogenic. B, that group B, capsular polysaccharide is poorly immunogenic and structurally similar to human neural cell adhesion molecules. C, that group B, capsular polysaccharide cannot elicit a sufficient immune response when conjugated to a protein carrier. Or D, protein-based vaccines are cheaper to produce. Okay, so A, B, oh, a couple of B's. Okay, C, okay, and then D. Okay, so the correct answer, it is a bit of a tricky one, and my wording is also quite tricky, is B. So we're going to come back to that a bit later when we talk about vaccines, looking at why the serogroup B is a bit more complex when it comes to developing the vaccines compared to some of the other serogroups. Okay, and then the last question has to do with public health and vaccine strategy. What is a primary reason for targeting adolescents? Okay, because this week we've spoken a lot about infants or other at-risk populations, but here we're talking about adolescents in meningococcal vaccine campaigns. Is it A, adolescents are a key reservoir for asymptomatic Neisseria meningitidis carriage, facilitating transmission to the community? B, vaccinating adolescents helps to reduce bacterial carriage, lowering the risk of spread and supporting herd immunity? C, globally, adolescents have an increased prevalence of Neisseria meningitidis carriage, making them an essential group to target for transmission control? Or D, all of the above? All right, so hands for A, hands for B, hands for C, and hands for D, all of the above. Ah, you guys are so smart. It's all of the above. So given the great answers, I think my presentation is done. No, I'm just kidding. All right, so I don't have a lot of time. So there are 7 aspects to my slides, or 7 different topic areas. So we're going to look at invasive meningococcal disease. We're going to look at what the current vaccines are globally on the markets, IMD in Africa, progress, challenges, and future opportunities. Because I don't have a lot of time, my slides will be available afterwards. So I'm going to jump around a little bit, but feel free after the time to go through it. And if you have any questions, you can contact me. All right, so starting off with IMD, or invasive meningococcal disease. For those of you who are not aware, but you guys seem pretty knowledgeable, meningococcal infections are caused by the bacterium Neisseria meningitidis. It is an aerobic, my apologies, gram-negative, oxidase-positive Diplococcus bacterium. And I think the picture is quite cute because although this is a little crude, but it looks like a little bum. So it's a nice way to remember it. It's a commensal organism in the nasopharynx, and you can get either capsulated forms or unencapsulated forms. Now, historically, we spoke about 13 different serogroups, although what they found over time is that actually there's 12. One of the unencapsulated groups was classified as D, but actually it turns out to be an unencapsulated form of serogroup C, right? And then there's 12 capsulated serogroups. And then what I want you to remember, because everyone's been saying this week, if you only remember one thing, but if you only remember one thing from my talk, is I want you to remember the serogroups. And they're really important from an epidemiological perspective, but they're also really important from a vaccine perspective, okay? So it's pretty easy. It's your ABCs, okay? So your 6 most common serogroups are A, B, C, and then because we're scientists, we make it difficult. We jump to the other end of the alphabet, X, W, and Y. Okay, so you're going to easily remember it, A, B, C, X, W, and Y. Okay, very, very important, because when we talk about the different vaccines, we're either going to use one of them, a monovalent vaccine, we're going to use 4 of them, a quadrivalent vaccine, or we're going to see the vaccines that are coming in the pipeline, which are fantastic, which are 5, so pentavalent vaccines. And the one thing that we're going to see that we're going to swap out with or play with a little bit is with the pentavalent vaccines, we're either going to see a B in there or we're going to see an X in there. So currently, for those who are not aware, our quadrivalent vaccines have A, C, W, and Y, but the pentavalent is either going to add a B or it's going to add an X. Okay, so we're going to see that when we get to vaccines or potential vaccine candidates a bit later. And then my last bullet point, I actually realized this morning I should have had a third factor, but we know that invasive meningococcal disease, the virulence or the likelihood that you'll get the disease depends on the virulence of the strain. It depends on your host characteristics. And then what I left out is that it also depends on environmental factors. All right, so just at a high level, IMD is a global problem and it can vary, right? So it varies in some areas to being just sporadic. Other areas we know it's more endemic. And then in the African meningitis belt, which we've discussed this week, it can be hyper-endemic, right? By hyper-endemic meaning that we have these really bad outbreaks in a cyclical manner approximately every 5 to 12 years. All right, then if we take a look at the general population, and by general population, I mean everybody. So from infants, children, adolescents, adults, the elderly, the young, the immunosuppressed, those with comorbidities, all of those factors, general population. And then in different areas, you can have between 4.5% up to 25% of the population at any point in time having asymptomatic carriage of Neisseria meningitidis in the nasopharynx. All right, it's going to depend on where you're located and a whole bunch of factors, but you've got this range of asymptomatic carriers. So for example, in this group here, the amount of carriage would probably be very low. You guys are not quite the ideal ages. I don't see any adolescents or infants, and I definitely don't see any elderly, so you guys are all good. But if you take that population and you take that percentage, of that percentage, you get another sub percentage, which can then develop invasive meningococcal disease. And that's usually between less than 1% up to 5%. So yes, it's a very low incidence, but it is a very severe disease. And we're going to take a look at that now. Transmission, how you get it, essentially it needs to come from human to human, and respiratory secretions, usually through some form of droplets, are the way that you acquire it. Okay. I just wanted to make a very important note here that carriage is really important, and I'm going to come back to that in the next slide as well. But there's two things that we want to keep in mind with meningococcal vaccines. Number one, we want to prevent invasive meningococcal disease. And number two, we want to reduce carriage. And you need to keep that in mind the whole time that we're talking about these vaccines. We want to achieve those two goals. Okay, so carriage is critical to preventing transmission from one individual to another. All right, so looking at a study by Christensen et al., they did a meta-analysis of 89 studies across 26 countries. So again, quite a big cohort of countries, quite a lot of studies looking more at a global level. And what they found in terms of carriage is that the carriage rate follows this distribution graph. It's not a, I don't know, when you do, at university, all the different types, I never saw one that's got a squiggly line and then increases and goes down. But so I don't know what you define this as, but the graph you can see has got definitely a lower number of carriage in infants, okay, so approximately 5%. It then peaks in your adolescence at approximately 19 years of age, and then it declines thereafter with age. So what's really important from the study and that you need to keep in mind again when we look at vaccinations and vaccination campaigns for meningococcal meningitis is that targeting your adolescents, particularly around this age of 19 to 24, is critical when we do vaccination campaigns in order to control invasive meningococcal outbreaks. Right, I just wanted to make one note on this graph though that is worth considering, is that if we look at information from the Africa meningitis belt, we actually see this graph move earlier. So we see a higher incidence in your infants and your children, and we actually see a peak in carriage at the age of 10. So given that this is an African conference, that is a very important thing to keep in mind. Okay, so this one that you're looking at is global, but if you consider meningitis belt, you need to shift that graph a little bit earlier. Okay, so this is a very, very busy slide, but essentially what I want to show you over here is an illustration of a cross-section through Neisseria meningitidis. And what you're looking at over here are really important when we look at vaccine targets, okay? So on the outside, we have the lipo oligosaccharide or the polysaccharide or carbohydrates or sugar coats. And underneath that, you'll see there's two membranes. You have your outer membrane and you have the inner cytoplasmic membrane. Now, throughout this week, we've been talking about these polysaccharide capsules, and it takes that, being sugarcoating something to a different level. These bacterium are so smart. And what they do is they've got this outer sugar coat that protects the bacterium from a host's immune system. So as you know, any outer membrane proteins are very good antigens or sites for either a vaccine or for the host to attack. So what this outer layer does is it actually helps to protect the bacterium and from the body's own immune system. And yesterday we learned about innate immunity and adaptive immunity, but particularly from an innate immune system. This is really a fantastic mechanism that this bacterium has to avoid your host-mediated complement-dependent bacteriolysis and phagocytosis. So super smart. And then we're going to talk a little bit more about the different proteins on the inside over here of the membranes a little bit later. But I just wanted to mention their names. So we have porins, pore A and pore B. And we're going to see that when we look at the vaccines, because those are some targets. And we're going to see opacity-associated proteins. And then, of course, what's really important are these little pili, and we're going to come back to them as well because they play a very important role not only in attachment, but they actually form part of the endotoxins, which leads to the severity of the disease in a patient. All right, so I've touched on the capsular polysaccharide. It's very important because we're going to come back to that being a target for our vaccines. But something else that I haven't mentioned yet is that this bacterium, super sneaky, also has an IgA1 protease. So it also, again, overcomes the host immune system by actually cleaving one of your key antibodies that sits on the surface of your respiratory cells. So yeah, so the first thing the bacterium has to do is it has to overcome the host immune system, right? So mostly you're innate. And then after that, it actually has to attach. And it does this through various mechanisms. So it uses those proteins that we find on the outer membrane, and it then attaches together with the pili onto the host cells. And then I was actually reading up on it. It's got this incredible mechanism where they start to multiply and they actually mesh together. So these guys are super, super smart. In addition to that, something that's also important to think about in the future when it comes to vaccines is that Neisseria meningitidis also does what's called a horizontal transfer of DNA. So it's almost like they communicate with one another and they can actually switch different parts of their DNA, which makes it quite challenging, right? Because they can change serogroups, they can change proteins, they can change in virulence. So these guys are very, very clever. So you can't see this picture well, but as I mentioned, you can get the slides after the time. But what I wanted to show you over here is that once these bacterium are able to get through your host defense mechanisms and into the body, they then circulate. And when they do so, they release a bunch of endotoxins that also help them to move through the blood-brain barrier. And that's really where this concept of meningococcal meningitis comes from, because you get such a high prevalence of these central nervous system symptoms because of the bacterium being able to cross that blood-brain barrier. Okay. So just a really high level picture of some of the clinical manifestations of IMD. You can either end up with just one or two symptoms, or you can end up with a host of symptoms, but it is quite challenging to diagnose initially. So the most prevalent symptom is meningitis. I think most of you have heard about meningococcal meningitis. So more than 50% of cases develop that. Pneumonia is pretty common. And a nice way to remember which serogroup is most commonly associated with pneumonia is actually serogroup Y. And it's an easy one to remember because remember, like I said, you would have your different serogroups. If you remember Y as an upside down, like a respiratory tract, most commonly Y. And for gastrointestinal symptoms, you know, like the squiggles of your gut, W. So just some key things to help you to remember. All right, one of the main challenges we have, of course, is the bacteremia, so it's circulating around the body and releasing endotoxins, so septicemia and meningococcemia. All right, so I'm going to skip over some of the other clinical symptoms as I do have some time constraints. And I'm just going to quickly go through just really high level, just mention it, is that you have a very high case fatality rate with invasive meningococcal disease. Yes, it has come down over the years with the advent of antibiotics. However, even with that, even with symptomatic treatment with fluids and antibiotics, we still have a very high case fatality rate. So it's rapidly progressive. If the patients do survive, there's also a lot of sequelae after the time. Often you'll find patients with amputations or long-term central nervous system side effects. So hearing loss, cognitive decline, and other related symptoms. Also, I don't see Jennifer, but from Jennifer's side, a lot of behavioral and psychological consequences. So just super quick, with regards to diagnosis, the gold standard has been to culture it. In order to do that, you need to make sure you've got a sterile site. So cerebrospinal fluid works really well. You can also use blood, and I've also seen that you can get samples from skin lesions, although the science is not as good. Just a word of precaution, if you have put this patient onto an antibiotic, you should just be careful around the culture. In that case, you'd rather want to use PCR. And then with regards to how do you manage these patients, symptomatic treatment is very important. These patients, especially if they go into shock, you'd have to monitor your circulatory levels. And then what's really well known is that you should also be using antibiotics from the start, and your third generation cephalosporins is what's recommended, including in South Africa. So I don't have time today to go through it, but work by Susan Mehring, as well as a lot of people from the NICD, have found that we're having increasing levels of resistance to penicillin, especially in Neisseria meningitidis. So just be aware of that. Rather start with the cephalosporins. Okay, so just really quickly, I don't have a lot of time, but just quickly to touch on, I'm going to skip through these on the vaccines that are currently available on the markets. We have discussed this week plain polysaccharide vaccines. So essentially what that was they took the sugar coat and they used that pretty much in its form. So quite a cute little picture here. They injected that to be the antigen. And so the B cells could then basically mount an immune response to that and develop antibodies. There were a number of problems with using a plain polysaccharide vaccine, particularly in infants. And we also know that you struggle to get a booster effect with these vaccines. So really not ideal. They're hardly used anymore in the world. Sometimes they are used in severe outbreaks, but it's really with declining frequency. And the most common ones that we use nowadays are conjugated vaccines. And again, we've touched on this during the week. You take the polysaccharide layer and you join it together with protein carriers. And those protein carriers can differ, but the most prevalent ones that we see in this space tends to come from derivatives of your diphtheria toxoid or your tetanus toxoid. And what's really great, as we also mentioned yesterday in the immunology session, is that it helps to stimulate both B cells for antibody production and T cells. Because of that protein carrier, it can present that to the T cells. So you're getting a much stronger and lasting immune response. All right, I know this is very difficult for you to see, but I just wanted to show that there are a number of conjugated vaccines currently on the market. The top one I haven't included because that's no longer available, but there are 3 monovalent type C, which is a more prevalent serotype in Europe and the UK and a few other places in the world. And then we've got our monovalent type A, which has been used extensively in the Africa meningitis belt. And then there are 4 quadrivalent ACWY conjugated vaccines. Just from a time perspective, I'm just quickly going to mention type B. They've been very difficult in terms of vaccine development because of the fact that the polysaccharide outer layer is not very immunogenic for the serogroup. So they've actually had to make use of those proteins that sit underneath that surface. And so for that reason, we find that there are 2 on the market, and they use combinations of these different proteins that are on the outside membrane. So there's more details in there. Right, in South Africa, just quickly touching on that, meningococcal vaccines are not included in our EPI, but they are recommended in the private sector under the pediatric management group. And then we do have some of the authors from the guidelines present. I don't see Dr. Parker, but I did see him on the program. So there are recommendations, particularly in high-risk groups, and we would obviously really recommend this, particularly at the moment for university students, given the unfortunate case, that fatality that was recently in Potchefstroom, which was actually serogroup Y, by the way. All right, since I only have 5 minutes, I'm going to go super fast and just quickly summarize the Africa story. So if you take a look at this graph of global epidemiology, what you see over here is various different serogroups. So don't worry about all the text. What I want you to really take a look at is what stands out. So what's really standing out in terms of epidemiological data is over here in the Africa meningitis belt, we see a very high prevalence of serogroup C. Now, the reason for this is historically we had a very high prevalence, 90% of the cases with serogroup type A. They implemented a mass vaccination program together with a number of stakeholders who were involved in that. And subsequently, we've seen a massive decline of serogroup A. Serogroup C is becoming a problem as well as serogroup X. And then in South Africa, which I'm not going to touch on now, but we're seeing a massive increase in serogroup B, up to about 44% of cases, even more of that. And particularly in the Western Cape has a higher prevalence of serogroup B as well. So what I want you to take from this over here is that there is a need for vaccines that are multivalent. Okay, so you need to make sure that you're not just targeting one serogroup, but multiple different serogroups. And again, to prevent disease as well as to prevent carriage. And then I'm going to jump ahead because I've mentioned the Africa meningitis belt before to a fantastic story, success story of progress in Africa. And that has been the vaccination campaign that took place this year in Nigeria. So Nigeria had massive outbreaks of MEN-C, which is one of the more common serogroups. And they were facing huge problems. So actually, between the period of at the beginning of October last year and March of this year, they had 1,742 suspected meningitis cases with 153 deaths. And so really through a global collaboration of a number of stakeholders, including PATH, Gavi, the UK government, the Serum Institutes of India, they were able to develop a pentavalent vaccine that had A, C, W, Y, and X. And it was really just through an incredible stakeholder network and a commitment by so many people, from the government to the CDC to all the healthcare workers, that they were able to roll out a vaccination campaign over 3 days, targeting individuals between the ages of 1 and 29. So 90% of your most at-risk individuals for vaccination. And they vaccinated over 1 million people in 3 days. And we haven't seen yet the full results from that campaign, but it just goes to show one of the success stories of how Africa is really coming to the forefront and really showing development and, the commitment to public health in this space. All right, and I'm out of time, but I just wanted to mention the thing which I kind of touched on, is that there are some fantastic pentavalent vaccines in the pipeline. There's 2 pharmaceutical companies, Pfizer and GSK, who are currently working on combinations that are slightly different. So their combinations are A, C, W, Y, and B. So we're really hoping that we'll see that in the future to help with this global problem with B as well. And that is all from my side. Thank you.
Thank you very much, Lauren, for very positive. Yeah, your enthusiasm is infectious. That's wonderful. Let's go to online first this time. Have you got some online?
No questions yet. I think there's
No
still
questions yet. All
thinking
right.
up
So
smart
I
questions.
see Prof. Rabi at the back there. I can run across.
The online team is here.
Thanks for the great talk. I have 2 questions. The one is about access to Men B for South Africa. I recently inquired, it's licensed, but it's still on Section 21, 3,800 Rand for a dose. Even the quadrivalent vaccine, 1,000 something for a dose. So, that makes it very unaffordable if you're thinking how many cases we are notifying. So that's just my one question. And just a side question. What is your view on the meningitis vaccine's effect on gonorrhea?
Okay, so thank you, Dr. Rabi, for your questions. I can't unfortunately comment on the monovalent type B. It's out of my scope in terms of where I work. And unfortunately, the price, I understand it is a challenge. But I do know that there are a lot of initiatives working in collaboration with the public sector and some universities where there are lower costs. But yeah, it is always a challenge. And I'm going to have to ask the audience to help with the gonorrhea question.
Gonorrhea, can anyone speak to the gonorrhea, Greg? No? All right, so that's homework. We all have to go and look in PubMed. All right. Other questions from the floor? Anybody? No? No? Here we go. Lucy from Malawi.
Thanks for the nice presentation. I have 2 questions. The first one is, how many doses is a person supposed to receive? And then the second one is it safe to use in pregnancy? Regarding that, we have a lot of adolescents who get pregnant in the adolescence period. Is it safe to use in the pregnancy? Because you talked of also its high prevalence in adolescents. Thank you.
Thank you for your question. So I'm just going to speak on behalf of the vaccines that we have at Sanofi, which is a quadrivalent ACY-WY vaccine. So with regards to the dosing, we have a big campaign of awareness that we try to do of 2 under 2. So from 9 months of age up to 24 months of age, you can give 2 doses, 3 months apart. And then thereafter, above 24 months of age, you can give 1. This is assuming a normal, healthy individual with no other contraindications. And then currently, no, I cannot advise its use in pregnancy. We have insufficient data to support that. Thank you.
There's a question from Tambe Elvis. He's saying, can you say something about the Men-Afri-Vac used in most of the outbreak in Africa? And what are the criteria for declaring an outbreak of meningitis?
Very good question. So my knowledge of the Men AfriVac was that it was a very successful campaign. It started in December of 2010, and it was done across all of those meningitis, well, 26 of the 28 countries that's found in that meningitis belt. They vaccinated over 350 million individuals, and the results have been phenomenal. By 2021, there were almost none outbreaks. And we also believe that there's been a significant reduction in the carriage rate. So very successful from that perspective. As to the World Health Organization, I think what declares an outbreak, I would have to ask the audience if they maybe know what the criteria is for that. I'm not too sure offhand.
I think it's contextual. So in the Western Cape, we have sporadic cases. Prof. Robbie, yeah? When we get a case, we still get, it makes the news kind of thing, It's notifiable and what have you. Whereas in the meningitis belt, it's a very different thing. So your criteria for declaring an outbreak would be different. Prof?
Yeah, we do see sporadic cases often in winter and groups of cases, mostly in our setting. And I think it would be the same as it's very usually quite young children that present with sepsis or meningitis. And I think Susan Meyring presented last year or the year before that there were only 50 reported cases in South Africa in 1 year. So it is, I think, when it happens, it's devastating, especially when you have this kind of setting where there's lots of students or young adolescents, young adults in a cluster. But it's not the most common disease that we see. But we're all very aware, and I think the notification process is very important. And then I think even in the CHAMP study, when they looked at causes of death in children, I mean, Klebsiella pneumonia is the main bacteria I think that we need to deal with.
Thank you very much, Prof. Any last question there at the far side there?
Thank you. Out of interest, are there any antibodies that are being developed for meningitis, seeing that it is a high fertility rate?
Antibodies, as in monoclonal antibodies,
monoclonal
like
antibodies.
we were hearing about yesterday for RSV.
Yes.
Anybody know? Lauren? No? Anybody? No. Prof. Hassie says no. But maybe that's a fruitful area for future research. Okay, I'm not seeing any more hands. Yeah.
2 more questions online.
Sorry, 2 online. Let's go online first.
Okay. Gabriel says, thank you very much for an insightful presentation. His question is, are there emerging strains or serogroups that present in which vaccines do not effectively cover? I'm paraphrasing, I think it was a bit unclear. So are there serotypes in which there's, I guess, no vaccine?
I would have to get back to you. I'm sure outside of because there are 6 most prevalent, maybe Prof. Hassie might have a better understanding, but there's definitely 6 most prevalent. So I'm sure that there are outside of those which we currently have vaccines for, because there are 12 serogroups. It's potentially possible.
And the second question from Bridget is, why is the meningitis vaccine not routinely administered to adolescents, given the high prevalence in this age group?
Yeah, so maybe I can ask anyone who sits on those advisory boards for their opinions.
I'm saying it's a cost. That's it. Okay, thanks to you.
Yeah, thanks so much for a very insightful presentation. Extremely comprehensive. No, so I just wanted to add, indeed, the outbreak response in Nigeria that took place a few months ago with the MEN5 was a great success and a good example for the countries in the region. But also we're expecting a number of countries in the meningitis belt to transition to the MEN5 in the next years. We have Niger that will be switching in 2025. Even though the price of the vaccine, it's much more expensive, but countries are opting for that, as you said, because of the prevalence of MEN-A decreasing. And indeed, in the Gavi supported countries, at least it is included in the routine. So it's giving at either 9 months or 15 months, depending on the country's schedule. And when the country introduces, they also do a large-scale catch-up campaign. For the MENA, I believe it's from 1 to 29 years, as you said, through the MENA Free Vac large campaign. For the MEN 5, it will be up to 19 years as a catch-up campaign. Yeah, thanks.
Thanks very much. Greg, did you want to say something?
No. I think the issue of cost has been covered by the previous speaker.
Okay. Thanks. Bang on time. Big hand for Lauren. And our next speaker is Michael. Is Michael with us? There's Michael. Okay. Okay, Michael Besong is from Gavi, the Vaccine Alliance in Switzerland. And he's going to talk on a very important topic. And I think it came up in Greg's talk yesterday, was hepatitis B birth dose vaccination in Africa, scaling up for impact. Over to you, sir.
Okay. I want to. Okay. Good morning, everyone. So I'm Michael Bisong. I work as a program manager in the vaccines programs at Gavi, and my portfolio, or the portfolio under which I work, is specifically on the hepatitis B birth dose as well as rabies vaccines. So these are like two new vaccines which were added in the Gavi's portfolio during the last vaccine investment strategy. And the program has been launched, and we are currently in the phase of supporting countries to like apply and to introduce these vaccines in their program. So for this talk, I'll be talking about hepatitis B birth dose vaccination in Africa, scaling up for impact. So I'm going to follow this outline. My slides don't want to come up. Happens, you know. Okay. Okay, sorry about that. So I'm going to follow this outline. I'll speak briefly about the link between hepatitis B and liver cancer. And then I'm going to provide an overview of hepatitis B infection, the epidemiology of hepatitis B infection in Africa, the recent WHO recommendation regarding hepatitis B bird dose vaccination for infants, as well as key considerations for implementation of the hepatitis B bird dose vaccination. And I'm going to end about talking about the role of Gavi in scaling up vaccination in Africa. So this is a study that I found, a global burden of disease study, which looks at the worldwide burden of liver cancer across childhood and adolescence. And we see that in children aged 5 to 19 years, apart from, besides like primary liver cancer, hepatitis B related cancer comes second in terms of incidence of liver cancer in this age. And the other chart shows us like percentages of cases as well as mortality. And we can see from both charts that there has not been a significant change in either incidence or in mortality related to hepatitis B related cancers between 2000 and 2001 being compared in both charts, which shows that there's been kind of like a significant but constant incidence in hepatitis B related cancers as well as deaths in this population. And knowing that an effective and cheap vaccine has existed for hepatitis B since maybe the 1980s, maybe the older folks could correct me if I'm wrong. It's really like a cause for alarm, knowing that all of these deaths could be prevented. So this brings us to hepatitis B infection. So the hepatitis B virus is a hepatotropic DNA virus, meaning that it replicates by infecting the liver, replicating in the liver, inducing inflammation in the liver, which could subsequently lead to liver disease. Unlike any other blood-borne pathogen, it could be transmitted perinatally or during early infancy from mother to child, as well as through sexual and parenteral routes. And one of the key features of the hepatitis B virus is that it can cause lifelong infection in individuals who are initially infected, so after an acute infection. And it's also important to note that the risk of chronic infection is inversely related to the age at infection, meaning that the younger children get infected. So if children get infected very young, maybe in the first year of life, they have a higher risk of having chronic hepatitis B infection, which could subsequently progress to liver fibrosis, cirrhosis, or hepatocellular carcinoma. So that's why it's even more important to prevent young children from having these infections with the vaccine. And in terms of prevention, as I said, a safe, cheap, and effective vaccine has always existed for hepatitis B. So looking at the global burden of the HPV infection, in 2024, the WHO released the Global Hepatitis Report, which highlighted key features. And one of the findings was that viral hepatitis, including hepatitis B, has been on the rise. So be it cases, be it deaths due to hepatitis B has been on the rise worldwide, and with about 1.2 million new cases, and about 63% of all new cases are in the WHO African region. So we can say that the epidemic is quite serious in Africa, which poses a huge responsibility for the different health systems and the different health programs, given the alarming burden of disease in the African region. And secondly, global coverage of viral hepatitis prevention, diagnosis, and treatment has remained very low. So in terms of like testing, it's still very low. Treatment of those who have been tested still remains very low, with only about 13% of people with chronic HBV who were diagnosed by the end of 2022, and only about 3% who received treatment. So it's very little has been done in the space of HBV testing as well as treatment. Maybe all the attention has been given too long to the big threes like HIV, malaria. Maybe we should come back to thinking also about hepatitis, given the severity of the burden of hepatitis on the continent. And from this chart, we can also see that overall, the global coverage has been very low, but it is even lower in the African region. And globally, it is estimated that about 45% of infants received the birth dose within 24 hours in 2022, but only 18% of newborns in the WHO African region received the hepatitis B birth dose vaccine. So you see, like, African region bearing 63% of the burden, but only 18% of the children received birth dose in 2022 within the first 24 hours after birth. And just to emphasize, another study looking at the HBS antigen prevalence in children under 5 in sub-Saharan Africa found about a prevalence of about 4 million, with about 2/3 of all children with a positive hepatitis test being from the African region. And it's sad to know that up to 25% of these children who have chronic HBV infection will definitely progress to liver cirrhosis and to liver cancer. And in most parts of Africa, like a cancer is synonymous to a death sentence. But given that there is an effective intervention which can prevent cancer, I guess it's a call for action for, those working in immunization to see how this vaccine can be scaled up to reach those who really need it. Now, in July 2017, WHO released their position paper recommending that all infants should receive their first dose of hepatitis B vaccine as soon as possible after birth, preferably within 24 hours. And this is quite challenging because unlike other vaccines where there's kind of like flexibility with respect to timing of the vaccination, this one, for the hepatitis B birth dose vaccine, the efficacy depends greatly on the timing with which the newborn received the vaccine. So it cannot be like delayed, like maybe BCG, which can be given maybe 3 days after or 1 week after. It has to be given for it to be most effective, it has to be given within the 24 hours, which leads to diverse operational challenges in practically implementing this program in the real life setting. And so after the birth dose, it's also recommended that children should continue receiving either 2 or 3 doses of hepatitis B birth dose vaccine. And I believe that most immunization programs already have the hepatitis B birth dose vaccine in the pentavalent given at 6, 10, and 14 weeks. So a birth dose and 2 or 3 additional doses has been shown to be 75 to 95% effective in preventing mother-to-child transmission. So this chart shows us hepatitis B birth dose vaccination in immunization programs as of April 2021. And we can see that most of the countries, especially in sub-Saharan Africa, are still to introduce, though they have the hepatitis B birth dose vaccination as part of the immunization schedule given with the pentavalent, most are still to introduce the birth dose vaccination. And we can also see that there are some countries where some countries have not yet introduced. Some provide the vaccine selectively to those who are born from mothers who are tested positive for hepatitis, as well as in other settings, it's like a universal birth dose for everyone. So it's like different schools of thought. Some people feel rather strengthen testing and selectively give the vaccine, and others think, well, I mean, since it's a cheap vaccine and it's proven to be effective, let's roll it out to everyone. And I'll be happy to hear your thoughts on that, based on your country, different country settings, which model might be most appropriate for you. So I just want to hear from you before I go to the second part of my presentation. So given that the hepatitis B birth dose vaccine poses this condition that it needs to be given within the first 24 hours of birth. So timeliness, it's really an important factor, right? What operational challenges do you think in your various settings, in your various countries, what operational challenges could you think about as which could stand as a potential barrier to effectively delivering this vaccine, and what can you propose as a solution to that operational barrier? So maybe in a few minutes, I'd just like to hear from some people.
Thank you. So, given that in our African settings, we still have a sizable number of births occurring outside the health facility. So I believe we need to still work with, in some communities, traditional birth attendants are still existing. So I think we need to work with, for example, in Uganda, the village health teams to be able to locate these birth attendants to say, yes, you've delivered this mother within, outside the facility, but please, within the next 24 hours, make sure this mother accesses the nearest health facility.
Okay, thank you. I think I'll leverage on what she said. Community linkage is important because you know that in Africa, quite a number of people still deliver at home in the absence of skilled birth attendants. So if they're able to leverage on perhaps linking up a community birth attendants with a healthcare worker, or rather a healthcare facility, and then they're able to send the signal that yes, we've had a birth, and we can get it within 24 hours. Of course, you know that there are other challenges such as bad roads, cold chain storage to ensure that those vaccines get to them. It's not just about getting the vaccines, getting it at optimal condition that would be able to benefit children. So I think we need to look inwards to country context to see how these things play a role. Thank you.
Okay, so during my PhD, this was something I studied quite extensively in Gambia. So the Gambia has a coverage of hepatitis B birth dose 94%, but timely coverage is barely less than 10%. So you have 90%, which looks good on paper, but you have just 10%, 1 in every 10 kids having it within the first 24 hours of life. So what we did was, or what I did was try to now spatially map which locations in the Gambia. So we specifically know which areas are where those delays are. But one key challenge is the fact that the way services are delivered, even if you deliver in the health facility, most health facilities are not immunization sites. So a woman delivers by, let me say, 2 p.m. at night. Where they immunize is another place. So the government had to ensure that vaccines are in some cold storage within those facilities. You deliver, the midwife has to deliver that vaccine before the child is handed over to the woman. So that's a key thing. Of course, we also just published a paper that then looked at how far people are from their homes to an immunization clinic. We found that if you stay more than 30 minutes to an immunization site, model travel time, people had a higher likelihood of having delayed hepatitis B. So there are other factors.
Thanks. Are there any inputs from online? No? Yeah, there we go. JP. Sorry. And then I think we'll call it quits. Yeah, all right.
So we have a comment from Animut and Leah, and they're basically saying the same thing, the importance of working closely with community and community gatekeepers. And there's also a comment from also Leah talking about issues of myths and misconceptions regarding vaccination, happy vaccination, and that sensitization of the community is very important.
Thanks, JP. Back to you, Michael.
One last.
Actually, also, I think that the complicated delivery and postpartum issues related to the delivery on those low income countries and poor community may divert the healthcare workers from providing the dose for the newborn child. The other complicated issues regarding the delivery, the postpartum issues like the postpartum hemorrhage and so on, may divert the the intention of the healthcare provider to provide the dose for the newborn time.
Sonia's got a burning comment.
In South Africa, we've got a new policy with targeted vaccination. So the idea is to test the pregnant mothers for HPV. Hepatitis B, sorry, to test them and then to start treatment and vaccination. Unfortunately, there's a challenge with the test and the vaccination. Our testing is extremely low, and the idea is then to vaccinate the babies born to mothers that tested positive. But there's a lot of challenges in the program. And I believe the reason why we're doing the targeted testing in South Africa is because of cost. Thank you.
Okay.
Thank you.
All right. Thank you very much for all your perspectives. And yeah, because regarding her comments, I think Gavi is working with the modeling team to trying to look at the cost effectiveness of the two models, looking at universal testing and then no universal vaccination, birth dose vaccination, and then targeted testing, targeted vaccination after testing, seeing which one is more cost effective. Maybe universal testing might be still cheaper than targeted testing. Okay, so most of my points have been mentioned. So in introducing this vaccine and ensuring that it is timely delivered to newborns, there are several operational considerations which needs to be taken into account. And for those who are born in health facility or out of health facilities, there are different considerations that we can look at. And for those children born in health facilities, we will need to like increase access to skilled care at the time of birth, because if this is given as a delivery package, like after birth, in addition to placenta care, to the vitamin K which is given, usually if it is given as a package of care which has to be given immediately after birth, it becomes more effective. So and after it is delivered by a skilled birth attendant. And another consideration is integration. Like he said that in certain health facilities, you have like the delivery room, which doesn't talk with the vaccination room, or the vaccination center is different from the delivery center, and it becomes very challenging in administering the vaccine within 24 hours if there is no coordination and integration between these services. And it's really quite a challenge. For the first applications that we received from two countries that applied to Gavi, Ethiopia and Sudan, to introduce the birth dose, it was really difficult because though one of the main considerations is they have to demonstrate how they are going to integrate that, but it at times is different if systems have, it's difficult if systems have not been put in place to ensure that there is this kind of coordination, or there has not been kind of like alignment in country to know how this intervention is going to be delivered. It can become challenging. So integration, which could be at the level of local health facility policies specifying the hepatitis B birth dose, standing orders for hepatitis B birth dose vaccination, reliable vaccines availability in the delivery room, because if the vaccines are not present in the delivery room, if they are not available 24 hours on 24, there are many children who will be missed, because maybe the immunization nurse who gives the vaccine is not there at the moment, doesn't work maybe at night, or the delivery occurred during the weekend. Those are some of the factors which could lead to either delayed or missed birth dose vaccination. And also there needs to be coordinated planning by immunization and maternal health staff in health facilities or in districts for the delivery of the vaccine, as well as availability of free vaccination in the private sector. So in different geographies where you have maybe a huge number of like private health facilities, this vaccine should also be placed there. Maybe they could be given free to these health facilities in exchange for them reporting about vaccination and the timeliness of such vaccination. Now, the bone of contention is with out of facility births. It becomes very challenging vaccinating these children. But as we heard about equity yesterday, the EPI staff or the EPI program has to make every effort to reach children in remote areas or to reach children in different areas or children that are hard to reach. And that is why when countries are thinking about introducing the vaccine, they should also think about strategies to put in place to reach children born out of health facilities. Some of it you have already mentioned could be establishing home visits, to provide timely vaccination. I don't know if there are certain countries that allow traditional birth attendants or community health workers to deliver the vaccines. Are there any? Okay, so it's kind of like mixed. Some countries allow, some countries don't allow. So in certain okay, it's not allowed. It's like a policy.
So community health extension workers in Nigeria are trained, and so they also work in the facility because they are ideally expected to spend 60 to 80% of their time outside of the facility, providing home services and outreaches, and then they spend the other 20% inside the facility. So they are trained to give they administer vaccines. So they are able to do that.
Yes, so that's a platform which can really be leveraged in such settings, in settings that they allow community health extension workers to do. I think even Ethiopia has something similar, and it could really be effective. But in areas where it is strictly trained health professionals who have to administer the vaccine, it can become really, really challenging or a daunting task. So another consideration could be like having the vaccine storage outside the standard cold chain. So because if you have to take the vaccine to the community, it might be difficult to maintain it in the standard cold chain. And it was also part of the learning agenda that Gavi was trying to do, looking at to generate evidence about being able to use the hepatitis B bird dose vaccine in a controlled temperature chain. And another consideration will be establishing careful pregnancy tracking and notification, like someone mentioned, to timely deliver hepatitis B bird dose vaccination, as well as using single dose vials, knowing that there are multi-dose vials, like 10-dose vials, which if you have like very few pregnancies in the communities, there'll be a very high wastage rate if you have multi-dose vials. So amongst others, these are some of the considerations that different programs should think about and try to adapt it to their setting with the aim of reaching these children. But it's true that it still remains a major challenge reaching children born out of health facilities. Now, briefly, I'll speak about the role of Gavi in scaling up vaccination. So the Gavi hepatitis B bird dose program was launched sometime this year in June, and the target is to support all the 35 Gavi eligible countries to introduce the vaccine by 2030. So as we saw on the map that I showed, most of the countries in sub-Saharan Africa have not yet introduced the vaccine, and many of those countries are Gavi eligible countries, and they now have the opportunity to apply to Gavi for support to introduce the hepatitis B bird dose vaccine. And like for any other program, Gavi provides support to procure the vaccines, with the country co-financing a percentage of the doses of the vaccines. But given that the monovalent vaccine is really cheap, the co-financing cost to the country is also very affordable. And in addition to that, Gavi provides a cash grant to help with the different introduction activities, which could be training, updating data collection tools, and other activities which the country needs to do to be able to introduce this vaccine, or to implement activities which are specific to this vaccine, and as well as technical assistance for the development of the application, as well as for implementation. In addition to that, we are currently in the process of rolling out a learning agenda to test different strategies that will be most effective in reaching children born out of health facility. Because, I mean, there are different ideas, there are different propose strategies, but it will be important to have like evidence-based strategies and cost-effective strategies of what has been tested to work in the different settings. And hopefully maybe by the end of 2025, we might have results from that. And with Gavi support, there's a potential that about up to 1.2 million perinatal related infections, as well as 1.2 to 1.5 million cases might be averted between 2021 and 2025. So the rolling out of this program really has a high potential for impact given the effectiveness of this vaccine. But however, all the different operational challenges could one of the risks which could reduce the effectiveness or the impact or the potential impact that this program could have. So just to conclude, as we saw, the WHO African region accounts for 63% of new infections, with only 18% of newborns in the region receiving the birth dose vaccination within 24 hours. Several operational challenges need to be considered for timely hepatitis B birth dose vaccination, and the support that Gavi provides has an opportunity to introduce and to scale up the vaccines in Africa, thereby averting a significant amount of deaths. So thank you for listening.
Thank you very much, Michael. We do have a little bit of time for questions. I've got a hand here, but some online as well. A couple of hands there. Okay, let's start here.
Thank you very much for your presentation. Mine is not a question, but to answer to your previous question. Actually, the delay, because in my setting, like the Nigerian guy, the vaccine program is separate from the maternity or delivery center. So if the program could combine like having a vaccination team working together with the midwife or the maternity or at the health center, it would be great. Second approach is to have people among the community to track down to see who is pregnant, who is not being seen, but maybe is being followed up by the traditional midwife so that they can report back to the healthcare system so that they can prepare and get in touch with that lady in order to have a rough idea when she's going to deliver. If they cannot convince her to come to the healthcare facility to deliver her baby, then someone would be able, because in the community, I had a experience that they are ready to assist, so someone can deliver that vaccine, which will limit the time frame for the 24 hours. Thank you.
I think that was more of a suggestion and a comment. Did somebody have a mic there?
Yeah. Thank you very much for that presentation, Michael. So I also feel that it's absolutely unacceptable that we have a vaccine that is cheap, effective, safe, and yet the African region that bears the brunt of the disease burden is not using it. So I think two quick things for me, questions. The first being that I think a lot of our African countries get the sense that because they already have the infant vaccination program in place and a perceived low risk of mother-to-child transmission, that they may not need the hepatitis B birth dose. So if you could comment on that. And then the second thing, from the Gavi point of view, the countries that are being supported to introduce the hepatitis B birth dose, are these countries all going for the universal approach, which in my opinion is the way that we should go? Or are there some countries that are going towards the targeted approach? I would assume that Gavi would also be in support of a universal approach.
Can you bring me your first question?
It's with regards to most of our African countries having this perceived low risk of mother-to-child transmission, and the fact that maybe if we have the infant program, then we don't need the birth dose.
Oh, okay. So regarding that, I mean, from my perception or from my interaction with the countries, I guess the different NGOs, civil society, or people with lived experience living in these communities have done a great job in advocating for the vaccine. So I don't think there is kind of any hesitancy in wanting to introduce the vaccine. But I guess the main issue now with most of these countries is around prioritization, given that there are other vaccines which they intend to roll out, like HPV, malaria. I mean, the big ones with, you know, political implications, and it's a matter of like when they'll want to introduce the vaccine and not if they want to introduce the vaccine. So most of these countries have already told us that, okay, they plan to come forth with an application, but they are trying to like situate the hepatitis B bird dose vaccination in the midst of the different priorities they have, like HPV, malaria, and so on and so forth. And regarding the question about oh, so, yeah, all the countries are going for universal, given that testing is really very suboptimal in many settings, and the vaccine is cheap. Gavi is providing it. I don't see why they should go for targeting. So all the countries are going for universal vaccination. Oh, that's what is recommended.
Thanks, Michael. So we got one in the middle here, then we're going to go online, and then I've got two here, and that'll probably take us to Tita. But carry on, ma'am.
Yeah, thank you, Michael, for your presentation. It was very insightful. This is my question. Why Gavi is more focused on hepatitis B more than others? Because we know we can have, I think we have 4 or 5 types of hepatitis B. Why we only focus on B rather than A, B, C, D, and E? That's my question. Thank you.
Well, I guess the main reason is that I don't think there's a vaccine for the other for hepatitis C, right? Is there a vaccine? I don't think. There's just treatment. So the vaccine which is available is for hepatitis B. Notwithstanding, in the current vaccine investment strategy, hepatitis A, I think either hepatitis A or E was shortlisted as one of the vaccines which was to be included. So it's a matter of like prioritization. There are many vaccines which Gavi can offer. Resources are limited, so it goes through kind of like an evidence-based process, the vaccines with the potential for the highest impact, value for money, and so on and so forth, which is included in the portfolio.
Thanks. Okay, let's go online.
We have a question from Kalisu Yaya, who wants to know if the 24-hour deadline cannot be met for valid reasons, and they are asking whether the newborn can still be vaccinated after 24 hours.
Yes, so ideally within 24 hours, but if not, as soon as possible after, as soon as the newborn can take the vaccine. Given that we say that a late vaccination is better than no vaccination. However, beyond 2 weeks, the vaccine has been found to be not to be as effective as if it was taken in the first 24 hours. So that's why it's recommended that all efforts should be made to ensure that the newborn receives the vaccine within 24 hours. However, the newborn can receive the vaccine during the first contact with the health system up to before the next timing of the vaccination, that will be at 6 weeks.
Okay, and then we have a comment from Daniel. He's saying we used to have a surveillance program at the Navrongo Health Research Center, and then he's saying that outbreaks used to occur every 10 years, and in 9 to 10 years there were always signs of an outbreak and would be likely more asymptomatic. And he is just saying that he thinks they could time vaccinations to the 8 to 9 years as to break the cycle of outbreaks in endemic areas. So what's your comment on that?
I don't get the comment.
I think he's just mentioning that in where he works, they have, I guess, Hep B outbreaks every 8 to 10 years. So his comment is that he thinks you should time the vaccination campaigns or vaccination drives, for the 9 to 10 years. So is that correct? What's your comment on that?
Well, the birth dose is a specific intervention for the period after birth. It's true that there is also adult vaccination, which should be given when people are tested and are negative, they can also receive the vaccine. But the birth dose vaccine is a specific time point to be given as soon as possible after birth.
Needs to do a study and write it down. So one question there, one there, Anita.
Tony, just hang on one minute. You want to respond to that? No, I have a question.
No, thanks so much, Michael, for the very insightful, comprehensive conversation. My question is more to the folks in the room. In the countries where BCG is also delivered at birth, are there synergies that are being explored for kind of using that same window of administrating BCG, which is, I believe, is more established and longer been like in the immunization schedule? And have there been studies about like, I don't know, some kind of interactions, or should there be like a delay of the administration of both vaccines? Thanks.
Can I just comment? So Daniel did clarify that that comment was for meningitis, not hepatitis B. So, yeah.
Thanks, Amina. So we actually conducted a systematic review just looking at, the studies that have been done on co-delivery of birth dose vaccines within the African region. And we actually found that healthcare workers sometimes tend to do this thing where they want to batch all the birth dose vaccines and deliver them at the same time. And when that happens, although you will get good coverage for BCG, OPV, you significantly delay the hepatitis B birth dose, which is actually quite concerning because for the hepatitis B birth dose, it needs to be timely. The exposure has already happened compared to waiting until the baby leaves. So by batching them, that created issues. So from the systematic review, we actually saw that while you have good coverage, 70 to 80 for BCG and OPV, you have very low coverage for Hep B birth dose, or the timeliness is just really, really poor. There were some countries that were delivering the hepatitis B birth dose at 6 months, which, as you can imagine, is not a birth dose anymore. So those are the issues, and I think we need to give clear guidance to countries to make sure that they do not delay the birth dose or try to batch them with other birth dose vaccines.
I have a question. So my question is, there is some hope of vaccinating adolescents or other age groups, especially women, in order to reduce the risk of transmission. That's my question. Or the issue of testing can be a problem.
Well, yeah, I mean, because it's also recommended that the vaccine should be rolled out in other high-risk populations, be it adolescents or in adults. But given that it's also an issue of cost and an issue you need to test before you administer the vaccine, those are all the challenges with rolling out adult vaccination. Another question is who pays for the vaccine, given that the Gavi funding goes specifically for children, like the standard API vaccines, as well as the new time points in the second year of life, which has been introduced. But for adult vaccination, maybe the country would have to think of how they can provide that, or the people who need the vaccine will have to pay for it.
Okay, so my question has already been answered. So it's just a suggestion strategy. Yeah, you've talked of like the Hep B dose is effective at birth. So my suggestion is like, isn't it possible to manufacture another vaccine that can target at least up to 5 years to target the out-of-bed children, so that when these women, they come at the facilities, let's say to get the family planning services, to get the postpartum services. So when at any time, at least up to 5 years, the babies, the children, they can still get the vaccine, because we are having issues with the out-of-bed babies to get the vaccine. Isn't it possible for our friends, maybe in research, or for those going to conduct the clinical trials, to come up with another vaccine targeting at least 5 years? Thank you.
Well, the problem is not the vaccine, but it's just the timing of the vaccine. Because even with the new vaccine, the issue with the birth dose is that it has to be given at birth. So it's the timing which is an issue. The vaccine is effective, but it's just the timing. Yeah.
But I think the point you're raising is, what about the other children who may have missed the birth dose? And hepatitis B is part of the EPI program, so they should have been caught up in the normal EPI. And if they weren't caught up in the normal EPI, then they should have been caught up with the JAK EPI.
Okay, this seems to have stimulated a lot of interest and discussion, not only this one, but all three topics. But you are 5 minutes late for your tea. I think if you could just join me in giving Michael a big hand. And then to all our presenters, I'm sure that you can continue the discussions over tea. So thank you. Enjoy your tea. Half past 10, eh? 10:30 we start again. Do you want them here 5 minutes early? Okay.
Is there anyone? There is a question online, which Chepiso is going to read to you.
Okay. We have a question from Tambe. He is asking what happens to a mom who skips doses, and he's giving an example of tetanus vaccination. If the childhood vaccination status for that pregnant woman is unknown, and she receives 2 doses during the first pregnancy and does not complete all the 5 doses, and then she gets pregnant 4 years later, what's the way forward? Do you start all over, or you start from dose 3 to 5?
Yeah, so essentially you give 2, or if you can fit in 3 doses of tetanus in the first pregnancy. And then for every subsequent pregnancy, give another tetanus toxoid containing dose.
You give all the 3, or you just give one?
Then you just give 1 in subsequent pregnancies.
1 in subsequent,
Yeah.
okay, thanks.
Thank you. Is there another question online? No.
Just to follow up on that. So if the pregnant mother doesn't receive all 3 doses in the first pregnancy, is it still effective?
Yes,
If
it
maybe
is.
they receive 1 or 2.
Yeah, it is. It is effective. So in the countries, fortunately, with Gavi support and so on, even our low-income countries have got quite extensive rollout of multiple tetanus toxoid containing vaccines in childhood. The pertussis tetanus containing vaccine, which has now been rolled out in South Africa, only 1 dose is being given. South Africa has got a very high rate of childhood vaccine coverage as well. So yes, 1 dose, just get in at least 1 dose in every pregnancy. If you can get 2 doses in the first pregnancy, that's great. We've done some work which is going to be published shortly looking at how many doses do South African women receive? This is going back through 2021. And very few women, only like 8 or 9%, receive 3 doses of tetanus toxoid containing vaccines in South Africa. Most will receive either 1 or 2 doses.
Thank you, Claire. There's a question on this side.
Thank you so much for the presentation. Actually, just regarding the maternal vaccinations, the most important thing is the safety and timing. So regarding the timing, is it affect the amount or the maximum antibodies that could be transferred to the child if they missed or miss the right time to get the vaccine?
So the timing of the vaccine in relation to gestational age. So normally vaccines are recommended to be given in the 2nd and or 3rd trimester. The later you give the vaccine in pregnancy, the higher the mom's antibodies are. So you need to give it at least 2 weeks before she's due to deliver. It's a bit difficult because you don't know if she's going to have a preterm. So you want to sort of give it in the 2nd trimester, then the mom's antibodies rise. That's the point that with the higher antibodies, those are the ones that are then crossing the placenta to the fetus. Over time, the antibodies levels does decrease. So if you give it early in the 2nd trimester, by the time the baby's born at the end of the 3rd trimester, the antibodies are lower, but it's much better to give it at the first time you see the mom, just in case she delivers preterm. If you can give boosters, for example, tetanus toxoid, then you can give it later in the 2nd trimester, early 3rd trimester.
Thank you, Claire. There's an online question, which we are going to.
Tambe again, he's asking, can you please shed some light on pneumococcal conjugate vaccination given to children? He's asking how different it is from the GBS vaccine for pregnant women, and how is the use of pneumococcal conjugate vaccines in the EPI? How is it in the EPI programs in Africa?
Sure. Okay, it's a bit off topic for maternal, but so pneumococcal conjugate vaccines are multiple, depending on which one, 10 valent, 13 valent. the way that the vaccine is made is similar. It's conjugated to a protein molecule. Pneumococcal conjugate vaccines are not recommended and given in pregnant women. And in vaccinations for pneumococcal in childhood, there's multiple schedules that the vaccines are given at either 3+0 doses, 2+1 doses, 1+1 dose, or just a single dose. So it depends on where the vaccines are given. So yeah, there's a very long answer to
Thank you, Claire. There's a question on this side.
Thank you, Dr. Claire, for your presentation. I guess I'm just wondering if there is any difference in antibody levels based on the mother's age. I don't know if younger mothers or older mothers, if there's any difference there.
That's a good question. There is, if you look at antibodies and age, immunosenescence is normally the elderly, but an elderly woman is not going to be pregnant. So in the reproductive age, I stand to be corrected, but I don't think it would be significantly different. What you do have is that an older woman might be exposed to things more often, but antibodies do decay. So then as she's boosted with things, then she'll make a good response. So it depends on how old. She can't be too old to be pregnant.
Thank you. There's another question on this side.
Should I take this one? She's got the mic.
Oh, sorry. You go ahead.
Yes, sorry, I just want to ask, in some extreme cases, we have women that do not follow that, the schedule of the tetanus toxoid that you showed up. So my question is, if you have a woman that just shows up whenever she's pregnant, and that's when she gets to take tetanus toxoid. So imagine if she just has one this year, and then she comes another 2 years, and like that. With that time duration, compared to what's on the schedule, does she get compared with the same immunity? Or are there?
Yeah. So if you think about tetanus toxoid vaccination generally, it's recommended that adults and so on have a booster every 10 years. And that's why if you end up in a GP practice or a casualty needing stitches, you will get a tetanus shot. Because most people don't go and get their tetanus boosters. I mean, who's had a tetanus booster in the last 10 years? I don't want a show of hands. So yeah, if a woman is pregnant every year, every second year, third year, give her a tetanus shot in each pregnancy. They do last. Yes, yeah.
Yeah, thank you so much for your presentation. Just a little concern. What's the ideal number or the total number of vaccine that should be given during pregnancy?
That's a good question. There's a lot of work that's happening on co-administration of vaccines and what happens if vaccines are given at the same time. We've got two arms, hey? So I'll say two. No, but if you think about it, Tdap, so tetanus, diphtheria, acellular can be given during pregnancy. That's already two pathogens that you're giving. Okay, so then you can give flu at the same time. So you're giving your tetanus, you're giving your pertussis. Can you give RSV vaccine as well at the same time? There is a lot of work that's happening. And there's, from a safety perspective, it's immunogenicity perspective, you can give them, yes, at the same time.
Thank you. We've got time for some more questions. Is there anyone who would like to pose another question? Otherwise, we will just give Claire some hands. And thank you very much. Okay, our next speaker is Dr. Erica Anderson-Nissen, who is a laboratory director at Cape Town HVTN Immunology Laboratory, shares insights on HIV vaccines, covering recent advancements, challenges, and the path forward for Africa.
Hello, everyone, and thank you so much for the opportunity to speak to you. Again, this year, I love this course, and it's just a pleasure to be back again this year. So I'm going to sort of talk to you a little bit about, to start off, just the burden of HIV and the potential impact of a vaccine on the global epidemic. And I apologize, I didn't change this slide, but they're actually results from four of the most recent HIV vaccine efficacy trials that have been conducted recently, and then looking ahead, what is the picture? So we start with global HIV numbers. When I looked them up again this year, they're actually slightly better than they were last year, but they're still just staggeringly enormous. And I love the image of the airplane, and I think I'm going to adopt that one for some of these statistics. But of the 1.3 million new cases that happened in 2023, almost 500,000 of those, or a third, were in East and Southern Africa. So clearly, the epidemic is still going strong, and we must really work towards an HIV vaccine. So this is a somewhat dated paper, not taking into account some of the newer modeling aspects that have been done, but this is just to give you a sense of what the impact of an efficacious HIV could be on the epidemic. So this is a graph showing the number of new infections with HIV over time, and it's modeled using basically sort of current trends, a 50% scale up in the UN/AIDS investment framework enhanced, or a full scale up in this IFE. And what you can see is that if you assume that a vaccine were developed that had 70% efficacy, that was given over 3 doses and needed a boost after 5 years, you could really see the different impacts on the number of new infections. Again, of course, if you do a full scale scale up, that you could really reduce them quite significantly and avoid millions of new infections over time. So I am part of the HIV Vaccine Trials Network. Our mission is to fully characterize the safety, immunogenicity, and efficacy of HIV vaccine candidates with the goal of developing a safe, effective vaccine as rapidly as possible for the prevention of HIV infections globally. We're a large clinical trials network. I work on the laboratory side of things, but our funding comes from the U.S. National Institutes of Health and the Division of AIDS. And so we have clinical trial sites that change over time depending on the different trials coming through, many of which are concentrated in Southern Africa. And we really work extremely hard to conduct our vaccine trials with communities as partners in the research process. So if anyone is looking for contacts within a vaccine clinical trials network, I think our community staff might have some really helpful insights to offer you. And they work extremely hard to make sure that the communities in which these trials are conducted are really supported, informed, and have a say in the decisions that are made that come from having a clinical trial. We have very engaged and respected community advisory boards that advise our network on how to conduct the trials. So when do we educate and involve the community? Well, we do it before the study. We inform the community and provide feedback to the researchers. During the study, we obviously need to really follow study progress. And then after the study, what can often get forgotten is to really share the research findings with the community so that people understand how they have contributed to the trial ongoing. We know that HIV infects at mucosal surfaces, and if you want to make an HIV vaccine, you probably need to involve multiple branches of the immune system. So HIV vaccines over the years have changed sort of what their target is depending on the research that's going on. There's a movement, which I'll talk at the end of the talk, to really swing us back towards development of neutralizing antibodies. But we know that binding antibodies are probably important as well. And we know that T cells also could play a very important role in vaccination. And HIV prevention immunology is these approaches also include passive neutralization, passive administration of broadly neutralizing antibodies. And I'll talk a little bit about that at the end. But you can see for a pathogen that infects at a mucosal surface, it's of course a very complicated process. You've got a layer of mucus, you've got a mucosal epithelial cells, you've got underlying immune cells, and there are many ways that you can actually target the virus. Why is HIV such a difficult vaccine target? We hear of successes in many different pathogens, but not really in HIV. And the story I'm going to tell you today actually has a lot of failures along the way. So we still don't really know what immune responses actually protect from HIV infection. Infected individuals don't clear infection, and infected individuals can be reinfected. A superinfection can occur. There's incredible viral diversity and immune evasion. As in many other pathogens, animal models don't actually always predict human infection. You can get a great result if you work in a non-human primate, such as a rhesus macaque, and then you put it in a human and it doesn't work at all. And since there's no cure, you can't do sort of whole inactivated strategies. So this is a dendrogram just to show you, a phylogenetic tree just to show you the diversity of HIV. And I really like this, sort of visual depiction of how variable HIV is, just showing you how the scale of the problem that we're dealing with is really quite large compared to other pathogens. So this is showing the HIV diversity in the Democratic Republic of the Congo in the whole country in 1996. And this is showing the diversity of global flu in that same year. On the right-hand side, we have HIV diversity in Brazil in the year 2010. And this tiny, tiny dot that you can't even see, my pointer is too big, is actually the SARS-CoV-2 diversity in the first year of the pandemic. So the challenge is great. And the issue is that HIV has envelope protein on the surface of the virus. It's highly variable. It has the ability to mutate and introduce many mutations over the course of even an infection in one person. And it also does things like put sugars on the sides of this envelope that make it really difficult for antibodies to bind to it. So there's a lot of barriers to actually getting a good HIV vaccine. In an ideal world, we want to develop one that prevents acquisition. So here's a graph showing HIV RNA levels, exposures, and if you were exposed to HIV, you would not get infected at all. But obviously, there are alternatives to this. You could have a vaccine that would actually prevent prolonged systemic infection. So you might get acquisition of HIV, but it would really become quite low and reduce the possibility to transmit to others and also maybe eventually be controlled by the immune system. And you can also imagine a scenario where someone could acquire HIV, but actually the vaccine would prevent disease. So there would be no mortality associated with it. So there are many strategies. The immune system is extremely complicated. And I think you'll see over my talk that we've really learned a lot of immunology and vaccinology from all of the funding and attention that's gone into HIV. And this is not in vain, despite the fact that we don't have an HIV vaccine yet. So this slide, I'll just go over very quickly. It summarizes four key previous HIV vaccine efficacy trials. And the big message is basically none of these large efficacy trials actually showed any protection. And in fact, this Merck Ad5, this was a sort of a pair of studies, one called the STEP study in the Americas and one in South Africa called the Pambili study, actually had a terrible result. So this was an adenovirus 5 vectored HIV vaccine. And it actually looked like, especially in uncircumcised men who had previous immunity to Ad5, that it actually slightly increased the risk of HIV infection. So as soon as that was discovered, the trial was halted, and Ad5 has not been used again, despite the fact that it's incredibly immunogenic and had lots of promise going into this trial. So we've got huge trials that were conducted, and they've all failed. But there are some new trials that were conducted in Africa that I think have really taught us a lot about HIV vaccines. And so I wanted to talk about those with you. So the first is the HVTN 702 or Uhambo trial. And this was following up on a successful trial in Thailand. And the second was using a mosaic vaccine approach. I talked about the diversity of HIV. Some very smart people have looked at the sequences and made a mosaic vaccine that takes pieces of sort of the most common sequences across the globe. You know, if you focus on just one clade of HIV, you might be developing a vaccine that only works in certain countries. And what we want, of course, is a universal one. So people have looked at the mosaic and diversity and put them all together in one vaccine to try to see if that would work. And then most recently is the PrEP-Vac trial, and this is using a DNA prime with a protein boost or a different vector, a modified vaccinia, as a different strategy. And I always like to show this slide. I don't think there's an updated one at the moment, but if you look back into 2018, I think it's really incredible that the vast majority of the HIV prevention trial participants were from Africa. So this is just showing the number of trial participants in thousands in different parts of the world. And there's just incredible will on the continent to get an HIV vaccine. And I think we need to keep that going and apply it to other vaccines. And we've got to have these vaccines, you know, tested and hopefully found efficacious in the populations where the epidemics lie, so that we can make rapid progression to licensure. So I mentioned this RV144 Thai trial, and this was really the only trial that was actually shown to have any effect on prevention against HIV. It was conducted in Thailand in 16,000 volunteers ages 18 to 30. And it used a 4-dose regimen that used the ALVAC vector, which is a canary pox vector, for the 2 primes. And then with the boost, it included a protein boost. There was evidence for partial efficacy, and this really rocked the whole HIV vaccine world, where it was modest, it was 31%, but it really was the first hint that you could make a vaccine to HIV. Until then, people had said, it's too diverse, the virus is too tricky, we're just not going to be able to get there. Everyone is extremely excited. Now, this is all the way back in 2009. And so one of the questions, of course, that came up is, great, we have this vaccine regimen, it shows some efficacy, especially if it shows partial efficacy. You'll have some people in the trial who must have had a good immune response that would have protected people, and maybe other people didn't make such a good immune response. So this wonderful consortium of investigators came together to really choose which immune response measurements would be the most important to do on this trial and which labs actually could do them the best. And so this was a great collaboration, and a case control study was designed. There were 41 samples from 41 individuals who had acquired HIV and 205 who had not acquired HIV, all of whom were vaccinated. And there were 6 sort of immune response variables that were selected as endpoints for the primary analysis. And what were the important findings? We looked at T-cell responses, we looked at antibody titers, we looked at antibody functions, we did all this work. And what did we find? Well, in fact, IgG binding to a certain variable region on that envelope protein on the surface of HIV was actually associated with a decreased risk of infection. So that's good. I told you that there was no real knowledge about what immune response was important for protecting against HIV. So, okay, if we know what we're going for, then we can test vaccines and we can see which ones induce that response and go for that. Now, interestingly, IgA binding to envelope was actually associated with an increased risk of infection. So if you had IgA at the mucosal surface, more of that, you actually were more likely to get infected than if you had IgG in the serum. Well, how do we have these 2 different antibody responses made to the vaccine, but they're exerting it looks like the opposite effects? And so what's thought, in fact, this is an example of the HIV envelope trimer, just one of those on the surface of the virus. And what it's thought is that if IgG binds, IgG is not just something that will neutralize a virus coming in, it has this end of the antibody called the FC portion. And then you get other cells that come, NK cells, macrophages, and those can help clear the virus via this FC portion. And so it looked like the IgG was really recruiting other cell types and having a good effect and clearing HIV. But if you had IgA, the IgA just bound to the same places that the IgG was, and not as much IgG could bind. And so you wouldn't have that beneficial effect. So you can see when we're really picking apart the immune responses to these vaccines, it gets really complicated. You can even have two immune responses, but one's good and one's bad, and they might actually either cancel each other out or one could prevent the other. So in the aftermath of this RV144 Thai trial, there was a huge set of trials that were designed, and the South African Medical Research Council was incredibly supportive and advocated to get these trials here in South Africa and some of them conducted in Southern Africa, because the hope was that if this vaccine actually worked, we could license it in South Africa first. Well, there was a slew of trials. So first we had to test the same regimen that was tested in Thailand and test that in South African volunteers. And that was when my laboratory here in Cape Town had just started, and we were able to look at the T-cell responses. And we found that actually South Africans even had better T-cell responses than what had been seen in the Thai volunteers. So we were really excited that this might actually be a good thing and that the regimen might even work better here than what was seen in Thailand. But of course, the issue is you're going to have this HIV diversity. The strains of HIV circulating here are different than those in Thailand. So the vaccine needed to be adapted. And in that process, so clade C sequences were introduced into the vaccine, so with the hope that those would match the circulating strains of HIV in South Africa better. And in addition, the adjuvant was changed, and we'll talk about that a little bit more in subsequent slides. But basically, the decision was to use an MF59 adjuvant, whereas alum had been used in the past. And this was because MF59 had been shown in several cases to induce much higher titer antibodies. And so it was thought we changed the sequence to match the clade C strains. We give a better adjuvant, we're going to get even higher levels of antibody responses. Maybe we'll actually get efficacy with this. And so you have to obviously progress slowly from early phase to larger phase studies. But the large HVTN 702 Uhambo was kicked off then. And this was a phase 2b/3 efficacy trial. Same initial regimen as the RV144, of course, with the South African adapted vaccine. And then some additional boosts were given because we know that antibodies wane over time. Maybe we get an even better response if we gave 2 additional boosters later on in the series. And again, the adjuvant was changed. Unfortunately, it didn't work. So in January 2020, the pre-specified criteria for non-efficacy were met at an interim analysis, and there was no efficacy seen with this regimen. So there was, of course, then a huge effort to try to figure out why, what had gone wrong. Were the immune responses different somehow? Had something changed when the vaccine was altered to try to work better here? And we've really spent a lot of time on this. And one of the key findings was that, in fact, so this is the, on the right is the Thai trial, the RV144. And this is just showing IgG binding antibodies, again, to that variable region of HIV. And at the bottom, you just see the net responses is just basically like the titer of antibody that was present in the serum. And then this is the response rate, the proportion of responders who respond. So you can see 100% response rate in Thailand and, pretty high titers. Well, then you look at HVTN 100, which was the lead up to the 702 efficacy study. And all of a sudden, we realized that actually the antibody responses were not as good. And so the hypothesis is that in sort of trying to make this vaccine regimen better, it somehow turned out that it wasn't as good. Was it the adjuvant? Was it something to do with the sequences? Because you have to look at the matches between the vaccine and circulating strains. All of those things probably played a role, as well as the fact that the epidemic in South Africa is different. There's larger infection rates, and maybe some of the behavioral and exposure factors could also have been different. So this is a huge disappointment after really being so excited to see to see the potential of this regimen. And I won't go into detail, but it definitely seemed to be that if you picked this apart, you could find subgroups of people who are in the trial who seem to have better responses and those who didn't. And if you work with statisticians who deal with immune correlates, they can do some incredible work to look at subsets of people in the trial and to understand that if you had a CD4 response, a T cell response, and an antibody response, that those people actually seem to be more protected. Whereas if you didn't have your T cells working well with your antibodies, they seem to be less protected. So even within the framework of a trial that didn't work, there were definitely some people who seemed to be more protected than others. So I mentioned at the start a second approach. So I've been talking about matching the vaccine to the strains of circulating HIV. What about this mosaic, this global vaccine approach? So this was an approach that's been developed over many years, been tested in non-human primate models. And again, it's how can you make a vaccine using sequences from HIV from different parts of the world that would hopefully be a global vaccine. Well, you can see again, over the last more than 10 years, the trials started to go and then progressed so that each blob is basically a trial on this timeline here. You can see that there were trials that were conducted in the US as well as in Africa and in Thailand. And those sort of progressed into additional, basically trying to look at a couple of different regimens and try to figure out which one was likely to be the best one to go into the larger studies. So HVTN 705, or the Imbokoro study, just completed a couple of years ago. And this was, again, using the mosaic. It's actually the Janssen Ad26 viral vaccine vector, which was the same vector that was used in their COVID vaccine and worked really nicely in their COVID vaccine. And then the first trial was Imbokoro, and this was conducted in women in Southern Africa. And then there was the companion study, a Mosaico study that was conducted in men who have sex with men to look at alternative transmission methods in the US and the Americas. And so then again, this was again sort of a 4-dose regimen. You have these slides so you can go back and look at the exact timing of the doses, et cetera. And it was the Imbokoro study was conducted in 2,600 African women. Well, when I gave this talk a couple of years ago, we were waiting, so with bated breath on the results from this trial. And then sadly, the results came out again. The vaccine was safe, which was wonderful, but the vaccine efficacy was 14%, and the 95% confidence interval crossed 0. So no efficacy yet again, with a different approach. Again, immune correlate studies were applied to try to understand, even though the vaccine didn't work, how did it look? How did it compare to the non-human primate data? Where did something go wrong in this whole thing? And in the previous study, I showed you that there were some immune signals that seemed to be associated with protection. In this trial, we couldn't figure anything out. We looked at correlations of all these immune response readouts, and nothing seemed to correlate with protection in this trial. And then you look at the measures that were in the preclinical studies in the non-human primates, they didn't even correlate, like not even a hint of it. And then we were waiting for the second Mosaico trial that had started just a little bit later, and then also came out with no efficacy. So huge disappointments in the field, huge amount of efforts, many, many trials leading up to large efficacy trials, and then getting nothing positive. There was also the PrEP-VAC trial, and this was a phase 2b African-led, European-supported HIV prevention study that was in East and Southern Africa from 2018 until this year. It enrolled 1,500 adults from Uganda, Tanzania, and South Africa, and it did this combined evaluation of 2 vaccine regimens. One was a DNA prime with the the same protein that was used in the RV144 ties study, and then another one where there's a DNA prime and a vaccinia-based vector with a different protein. So there's been a lot of products, luckily, in the pipeline for HIV, so we haven't had to scrounge for potential candidate vaccines. But then this trial was stopped, vaccinations, in December 2023 due to futility. And the results were just announced in July 2024 at the International AIDS Conference. So where are we? I've given you all these terrible results. It's completely depressing. It's more depressing than the US election results, which are killing me at the moment. But an efficacious HIV vaccine really still eludes us. I've shown you all these trials. We haven't found anything that's really the goal that we're going for here. But as I said, the pipeline is rich. So we're not out of luck. We're not out of opportunity to try things. And so I want to talk briefly just about two other approaches. In fact, the HVTN conference is actually going on this week at the Westin at the waterfront. And there was a sort of session today on these neutralizing antibody approaches. So I've focused on sort of binding antibodies, and I've mentioned T cell responses. Now the field has really swung back to let's try to get really much better at neutralizing antibodies. So there are non-human primate studies that show if you put enough neutralizing antibody in a macaque and you infect them in a laboratory situation, you actually can prevent HIV infection. So let's just take a step back and go back to that fundamental. And then two trials were conducted called the antibody-mediated prevention, or AMP trials. So they took a serum antibody that we knew was really good at neutralizing. One of them was isolated in the US at the Vaccine Research Center. There are others at Caprisa and other places in Africa that have been isolated since then, and they're also going into trials. Tried to ask, okay, if we can't figure out how to get a vaccine that's going to give really good serum neutralizing antibodies, at least in the meantime, we can take antibodies that we know work. We're not trying to induce them new from people. We'll take one that we know that works. We'll infuse them into people and try to figure out how much antibody do you actually need in the serum, which is, of course, is going to be different than at the mucosal surface where you're getting infected. How much do you need in the serum? How much do you need at a mucosal surface? So at least we know what we're aiming for when we develop new vaccines to induce neutralizing antibodies. And so there are two trials that were conducted. One is HVTN-703 or HPTN-081, and that was conducted in women in sub-Saharan Africa. And then 704, which was conducted in men who have sex with men and transgendered people in North America, South America, and Switzerland, again, trying to cover different modes of HIV transmission. And what was found was, in fact, that volunteers came in, they were infused with this antibody IV every 2 months. I mean, this trial, when you heard the design of it, you'd think, this is insane. How would you do this with thousands of people? But it was done. And even though what happened is that they didn't give enough antibody to actually prevent acquisition, they were still able to figure out how much antibody was needed. Because you infuse people, obviously our bodies are all different. We have different rates of turnover. Some people will have that antibody stay for longer, whereas for other people that serum half-life just drops off. And so we know now sort of what concentration we're looking to achieve in trials going forward. And this, in combination with some really amazing protein engineering strategies that have happened over the last 20 years. People have taken the structure of envelope, they've looked at it in depth and tried to understand what structures do you need to trigger B cells? Because what happens is when you immunize people, you give them a protein or a protein produced by a viral vector or however, and their germline B cells are recognizing it. So B cells that haven't seen this antigen before, they're new B cells, they haven't really matured their receptors. You have to find a protein that this B cell can target. And then what you do is you give it a boost that's slightly different to really get that B cell receptor, because B cells are rearranging their receptors. What they're trying to do is get antibodies that bind really well to the antigen. And so there's this incredible process that's been pioneered in mice, moved to non-human primates, and now is actually being tested in humans, where you can prime these precursor B cells. You boost them with a slightly different protein to expand the ones that you want. You boost them more, and then you eventually get to B cells that actually can produce really good neutralizing antibodies. And so this is in early stages in humans still. But it's really testament to, I think, a lot of the scientific progress that's happened in terms of protein engineering and also sequencing. We can now take B cells out, look at their sequence, and understand which ones are in families and are the ones we actually want to get. So there have been huge advances. And this is just one summary slide of some of the trials going on with some references on there in case people want to go and look up more information. But there's some exciting early phase trials that are being conducted both by our network as well as the International AIDS Vaccine Initiative. And what they've done is made this protein nanoparticle in a nanoparticle form called the EOD-GT8. And it's really engineered so that it has the right sugars on the surface, the right sequences of envelope to really try to trigger a really good neutralizing antibody response. And I just heard yesterday about a trial that's happened now, which is the new using an mRNA technology to actually make this protein and try to get volunteers in Kenya and South Africa to produce neutralizing antibodies due to this new technology. And again, using mRNA, which of course exploded after the COVID vaccines were developed. And our network has more than 20 what we're calling discovery medicine trials, so very small trials where we can really do in-depth immunology and understand whether these new vaccine strategies are actually working. And I just want to point out also that all of this work on HIV vaccines, even though we don't have an HIV vaccine yet, all of that work set a huge foundation for the really quick development of COVID vaccines. So thanks to many funders putting money into HIV vaccines, it hasn't just all gone to waste in efficacy trials that haven't worked. It's also really helped with many of the strategies to develop a COVID vaccine. And I also just want, I'm sure this will come up in the discussion, I also just wanted to mention on the non-vaccine side, there's also been, enormous advances in PrEP or prophylactic exposure prevention. And the good news in 2021 was this Purpose 1 trial, which was spearheaded by Linda Gale Becker and published just recently in the New England Journal of Medicine, which evaluated basically subcutaneous antiretroviral lenacapavir versus daily oral treatments. And they enrolled girls and women in South Africa and Uganda. And it was really the most amazing result that if you gave this lenacapavir subcutaneously twice a year, there was 0 infections in that lenacapavir group. So I think, many people can look at this and say, oh, well, why do you even need an HIV vaccine? We're getting so good on the prevention strategy. But, the argument is always that we sort of need this toolbox of multiple modalities. HIV continues to evolve incredibly well. Some people, lenacapavir might be a great way to prevent HIV, but it's not gonna be the answer to everyone. And always a vaccine is what what we're aiming for. So exciting results and lots of new science happening in the HIV vaccine field. I'll just thank the members of my laboratory here in Cape Town, and I'll be happy to take any questions.
Thank you very much, Erica, for this excellent, sobering, yet optimistic presentation. Thank you for that. Are there any questions?
There are a lot of questions online.
Okay, then let's start online.
So the first question is from Mohammed. He's asking how are these vaccines coping with the mutation strategy of HIV?
Yeah, it's a very difficult problem, and I tried to sort of illustrate some of the approaches that have been taken. There's also there's a lot of work being done. I sort of mentioned this, but to really look at how HIV diversity is even increasing now over time. So if you study HIV sequences across the world now versus what they were 15 years ago, there's certain evolution of HIV. So we've really got to think extremely carefully about how to make a vaccine that's going to work for most or if not all of these strains. This mosaic approach was tried, but it clearly didn't cover the diversity well enough. We know that T cells recognize HIV as well. And so one strategy that's being employed is to look at the particular sequences in HIV that are really important for its structure and need to stay conserved. So if you change those sequences, then the structure falls apart and the virus can't replicate or infect and do what it needs to. So trying to target conserved regions of HIV is an approach that's going forward now as well.
Thank you. The second question is from Premise, and I think it builds on the answer that you just gave. They're asking, how have HIV vaccine trials contributed to understanding of the virus and the genetic diversity of HIV strains in the African continent?
Yeah, I mean, definitely a lot. So one of the things, especially in the AMP trials that's being done, is to try to understand in individuals who acquired HIV, can we look at the sequences of the HIV that they acquired and see whether there was what we call pressure from the antibody. So if you had that antibody in your blood when you were infected with HIV, would you have strains of HIV that were less likely to be recognized by the antibody? So the thought is the antibody was there, it was doing something, it was preventing infection by certain strains of HIV, but not by others. So there's a lot that can be done looking at this evolution of the sequence diversity.
Thank you. There's a question on the side. Yes.
Thank you for that wonderful presentation. I was just wondering, with some of the low efficacy trials where there's been failures of HIV acquisition, how are you navigating the issues of the ethical issues around those type of HIV infections, and what's the impact that it's had on the people that have acquired the HIV infections during some of these trials, and would that affect future trials that may be planned?
Yeah, I mean, it's a huge issue, and we actually have a session on Friday with some of the people who work, the community group people, to get more feedback on this. So my knowledge is a little bit dated, but yes, I mean, you know, it's so difficult to have to go back to a community and say, actually, the vaccine didn't work. All the work that you did, you know, making your bodies the test case for this vaccine. You know, it didn't work, and we did everything we can. You know, there's always at least the people in the trials, they're getting sort of supportive healthcare, they're getting, you know, access to PrEP. There are benefits to sort of being in the trial, and I think that can buoy people going forward. But it's a very difficult challenge to then have to come back and say, oh, now we've got another one we really want to try, and you lose people's motivation, definitely.
Okay, thank you very much. Maybe,
yes.
Yeah, example maybe for COVID-19.
I think it was okay.
Maybe let's go maybe the last pandemic of COVID-19. Vaccine was produced maybe within 10 months, and maybe after 15 months, it was globally everywhere. Compared to HIV/AIDS, which is also was another pandemic globally, for almost 40 years, there's no still effective vaccine available in the world. So what could be yes, I know maybe the differences of the magnitude and seriousness of COVID, where thousands of people were dying daily. A lot of efforts were made because of that maybe. But what are the main limitations and challenges maybe to get effective vaccine for HIV up to now? Thank you.
Yeah, I mean, it's just, I think the issue, and it's very difficult if you're not sort of in the virology field to understand the basics of these different viruses, but they're just really different. So SARS-CoV-2 is a virus, it puts tons of this spike up on its surface. The spike isn't that different. You know, we do have there's Omicron and there's different strains that go around, but it's not that different. Whereas in HIV, it's like, it's an incredible virus that I mean, first of all, it takes out cells of the immune system. it's super variable. So you make a vaccine and then all of a sudden you've got a virus and the vaccine doesn't match the virus anymore, so it doesn't work. And it's just been incredibly difficult to understand how to get the immune system to to recognize this virus over time and across the world. And I think your point is well taken that, something that's also happened and probably still needs to make sure it's maintained despite the failures of these efficacy trials is a plan. So when there's an HIV vaccine, how are we going to get it out and across the world? And I mentioned that the South African MRC was very interested in trying to get trials within South Africa to try to speed some licensure process if that happens. But you've got to work on that sort of what if, when we get a good vaccine, how is it going to be rolled out is incredibly important to do at the same time.
I think very nice talk. Probably my question is a little bit philosophical. I went to medical school in probably the early 2000s, and I remember entering the internal medicine world and how the HIV patients looked back in Nigeria. And years down the lane, we now have PMTCT, we now have there's use of protective condoms and all that. So as a doctor or as a patient, somebody like, why do we still need HIV vaccines when we have strategies that really do work, the things I have mentioned, compared to 2001 when you go into the wards and you see everybody looking cachectic and all the other HIV things that follow? Why do you still think we need a new vaccine for HIV and not prioritize other things that plague our part of the continent?
Yeah, no, I mean, I think it's a really good question. I think there's the cost issue. So this one, I didn't go into that, but that apparently was a huge topic of discussion at the International AIDS Vaccine Conference. So lenacapavir, it was developed by Gilead. I mean, the cost is just unreasonable. No one can afford the cost of this preventive treatment. So there's cost issues. I think there's also the issue of we need to look at sort of across the age spectrum. So you have babies that are born to mothers who have HIV. How do you obviously there are drugs that can treat that, but imagine if we had a vaccine, you could just give it once. We wouldn't have to be doing this over and over, all the long course of drugs. There's also the the evolution of the virus and how we need to sort of take that into account. But I think it's an excellent question, and I think in an ideal world we would have all these modalities kind of working together. You would be able to work with the person individually and say, well, here are your 3 options. From a public health standpoint, hopefully we can have a bigger impact if we've got this whole toolkit working together. But it's a very valid question.
Thank you very much for the talk. Sorry. So my question was, you mentioned that in terms of the ethics, the participants also had access to other preventive measures. So I was just wondering, to what extent did this influence the results? Were they also on PrEP medications during these trials? And so what was the comparative effect? Was that also evaluated during these trials? Thank you.
Yeah, it's a very good question. And, years ago, those trials were designed assuming no PrEP because it wasn't available. Now you ethically can't do a trial without PrEP. So that's definitely increased the number of individuals that need to be in the trial because, you have a trial, you've got to have enough HIV acquisition events in order to tell if the vaccine works or not. So it's basically, everyone's counseled, everyone's given access to PrEP, although surprisingly, a lot of people actually haven't taken it, even though offered it. But you've got to make the numbers just big enough so that by chance you're going to get enough acquisitions of HIV to be able to tell the difference between the two. But it's double, it's increased the cost, so it makes it more difficult to do trials too.
Thank you for that. We do have time for another online question.
And there's one.
We have a question from Festus. He's asking whether, can this mode of neutralizing antibodies be used in the prevention of other diseases? And he also wants to know whether it's been used with positive results before in other studies.
No, that's a great question. So we're still in the early stages in terms of understanding the evolution of that B cell response to the different antigens. But yes, it could totally be applied in other strategies. And we aren't at the point where we know, we're sort of at the point where we know we can just barely get some of those neutralizing antibodies in humans, but it's not going to be good enough to prevent HIV yet. So we're early days, but yes, definitely applicable to other pathogens as well.
Thank you so much, Erica. That was very inspiring and excellent talk. Let's put our hands together. And let's invite our next speaker, Dr. Albie de Frey, an expert in travel health with 30 years of experience, highlights the often underestimated role of vaccination in protecting travelers, stressing its importance in comprehensive health risk management. Albie, thank you.
Thank you very much. Can you guys hear me? Yes. Right, okay. First of all, I've got a confession to make, and that's that I'm a rabid vaxxer. Okay, so you've heard lots of science now. Now I'm going to hear a little bit of the art of vaccines and of immunology. So let me just see how this works. Thank you very much for the invitation to Prof. Kagina, Prof. Ridzani, everybody else. It's always a pleasure to come. I feel like I come home when I get here. So let's see how we can go on with this. Okay, so I'm supposed to talk about targeted vaccination, in other words, vaccinating travelers. But I'm going to take you on a little bit of a journey about that. So just watch this space. Okay, so that's a quick overview of what we're going to do. My last confession is I've got to catch a plane. So I'm not going to talk too much as I normally do. But just a quick introduction to travel medicine. Who of you have got an idea of what travel medicine is all about? Just quick hands up. Yes, tell me. Say a lot. Yes, and which type of travelers? Yes, and
example, if you are going to yellow fever endemic area, then you get the shot.
why do you get yellow fever vaccine?
Because it's not endemic in this area.
Okay, so that's for a public health reason.
Did you say where or why? Albie.
No, you're quite right. So it's why did you get it? Because of public health. I don't want you. You come from Nigeria?
Yeah.
Yes, I don't want you to bring your yellow fever here because we've got the mosquitoes, okay, but we haven't got the virus. So that's public health. So are you at risk when you come here of getting yellow fever?
Sorry?
Are you at risk when you come here of getting yellow fever? No. Okay, because we don't have it. Am I at risk if I go to Lagos? Yes. Okay. So that's to protect me. Okay. So sometimes it's protecting the public and sometimes it's about protecting me. But what I wanted you to say is, oh, travel medicine is for rich people with 4x4s that go on safari to Kenya. Okay. And that's not like that. Okay. So fantastic. You get full points on that. So this is a little definition of travel medicine. It says it's a multidisciplinary part of medicine of which the primary goal is to teach people how to protect themselves, to prevent illness in travelers. Okay. The secondary goal is if you are traveling and you get diarrhea or you get a fever, is to teach you what to do. You've got diarrhea, you've got to rehydrate, maybe you've got to take antibiotics, antimotility drugs, and if you've got a fever, you've got to think you've got malaria. Okay. Right. And then the thing that sort of was added on a little bit later is that we also have a concern about protecting the people in the host country. So if you come from Nigeria, you come here, we're the host country. If I'm going to Kenya, Kenya is the host country. Okay, right. Okay, so there's three simple things you need to know about travel medicine. Okay, what you bite into, what bites into you, and what hits you. Then you know travel medicine. So what you bite into, food and waterborne disease, okay? If you're not careful, if you don't cook it, boil it, peel it, or leave it, then you're going to get traveler's diarrhea, and also you're at risk of hepatitis A, cholera, typhoid, etc., etc. God's gift to travelers is bananas, because you can peel them without touching the inside. You don't need a vaccine. If you've got a banana to eat, you can survive on bananas and bottled water or beer. Okay, right. Watch out what bites into you, and watch out what hits you. So there's no vaccine against that. It's taxis and AK-47s, okay? And that's just to show I'm not a chauvinist, so there's something for the girls as well. And that's just because we're realistic about what's going on in the world. So a little bit more formal is you bite into it. Traveler's diarrhea, hepatitis A, typhoid, Lassa fever, cholera, it bites into you. So not just the hippopotamus that goes for the veterinarian, okay, but also the most important one for us in Africa at least is malaria. Yellow fever we've spoken about, rabies, ladies of the night, we just talked about HIV, sleeping sickness, and gentlemen of the night, sleeping sickness, dengue, river blindness. And then it hits you, we spoke about that, so that is injuries on duty. I work with a lot of corporate travelers, road traffic accidents, and interpersonal violence. So where did this idea of travel vaccines start? It started in the 1980s, roughly, in Switzerland, squeaky clean Switzerland. Everybody vaccinated against every possible childhood disease, just like little Swiss chocolates packed into neat little boxes, no infectious diseases, okay? And they had lots of money, so they liked to leave their fancy castle in the Alps and go traveling to developing countries. And in those developing countries, they were exposed to infectious diseases that were no longer at home, okay? And that's where the idea came from. You've got your childhood vaccines, but if you're going traveling, I'm going to vaccinate you against diseases that we do not have back in Switzerland. So just park that idea a little bit. So travel vaccines, all Swiss or European children, and therefore adults, are fully vaccinated. I went to a travel clinic once and saw a 35-year-old guy in Zurich. He came in, the doctor says to him, right, good morning, welcome, where are you going to? Why are you here? I've come for my travel vaccines. I'm going to London for the weekend. I kid you not. Swissy going to London wants travel vaccines because London's not so clean, you see. And he could take out his childhood vaccination certificate. Who of you have had a tetanus jab in the last 10 years? Hands up. You're all going to die, okay? And who of you can show me your childhood vaccination certificate? Are you Swiss? Vrachtig. We're proud of you, okay? All right. I certainly can't show mine anymore. Okay, so that's where the whole thing came from. So let's just think a little bit about it. Now, sometimes people tell me travelers are rich people with 4x4s going traveling, and it's got nothing to do with public health. But in fact, as a travel health consultant, if I don't know what's going on in public health, I can't help my travelers because we're really all on the same planet. Okay, so travel health practitioners are dependent on public health practitioners for epidemiological data regarding disease risks and vaccine coverage to make good evidence-based recommendations. That always comes up in medicine to travelers. So I need to know what's going on in that little rural clinic somewhere deep in Liberia, somewhere in the northern part of Liberia. And then when I see return travelers with a fever or an ache or a pain or a diarrhea or something, they act as sentinels. They give us an idea of what's going on out there. Okay, right. So I go and look at public health pages. I see up there the WHO is saying there's a big measles outbreak in the Congo. This is from a few years ago. So I think if I've got travelers going to the Congo, I need to make sure that the measles vaccines are up to date or that they've had measles and therefore they've got lifelong protection. But it's not only in the Congo that we're seeing measles, okay? There, 2022, we see in 2020, 3 laboratory confirmed cases of measles. And then we go to last year, we've got 960 cases of measles in South Africa, okay? And there's the statistics for the last year, a huge spike in measles cases up there. So should I be vaccinating my travelers going to the Congo? Probably yes. And what about people coming here? Okay, the Europeans, the Americans coming here, presuming that they've been vaccinated, should they, or not been vaccinated, should they be vaccinated if they come into South Africa? Not all the diseases you can contract is vaccine preventable. Travelers as sentinels. A couple of years ago, we had East African trypanosomiasis from Uganda, from Malawi, and Zambia. And we know how does it present? It presents with a fever and a headache and body aches. And a lot of these guys get diagnosed as malaria, okay, because they missed the diagnosis. And then that sort of went away. And then last month, we had 2 cases from Zambia. A father and son that went traveling. They were in Zimbabwe and in Zambia, in fact. But we think they actually contracted it in Zimbabwe. So trypanosomiasis has not gone away. So we need to stay up to date with what's happening. And in Cape Town here a couple of years ago, 2023, they diagnosed a case of West African trypanosomiasis in a lady from Kinshasa, but she presented here. Unfortunately, too late, because the diagnosis was missed in Kinshasa, where it is endemic. Okay, she'd been ill for 2 years. So in travel medicine, we're always sort of balancing travelers' health versus public health. Sometimes we concentrate on the traveler, and other times we're concerned about the public health, just as we demonstrated with yellow fever right there in the beginning. So a percentage of food and waterborne diseases and a percentage of vector-borne diseases we can prevent with vaccination. Okay, but the rest is all about education. I've got to teach people what to do not to get sick, and I've got to teach them what to do once they are sick. It does not cover malaria. From a travel point of view, it still doesn't cover malaria. We know that there's a very promising vaccine against malaria, but that is intended for babies that grow up in endemic areas. It's not for travelers right now. We get lots of inquiries about that. And I had to say a few years ago, it also doesn't cover COVID. And now, fortunately, it did cover COVID and it went away. And then what happened? Who of you know that you can go back home now and get a COVID vaccine, an up-to-date COVID vaccine back home? Come, hands up. Who's from Tanzania? Somebody's from Tanzania. Have you got COVID vaccine available in Tanzania? Sure. Where do you work? Where do you work in Tanzania? In Dar es Salaam. But you're in the public sector. Okay, who else has got access to COVID vaccine here? We heard lots about vaccine apartheid and no access, and we were at the back end when the world got COVID vaccines. So eventually we all got access to COVID vaccines. Who's still got access? Where's the South Africans? Do we hear about people getting sick and ending up in ICU with COVID? Yes. Do we see people dying from COVID? Yes. So where is our COVID vaccines? The one that we shouted so much about. And I went to Tanzania, I went to 2 mines there, and they opened the fridge and they had in-date J&J COVID vaccine available. And I was like, wow, I'd have to stop talking about Tanzania as a third world country because you're better off than we are, okay? Because where's our COVID vaccine? I want to protect individuals against COVID. People who are vulnerable should still be able to get access to COVID vaccine. So you're lucky in Tanzania, but we're not so lucky because we don't have, in the rest of Africa, for all the fighting we did, we don't have vaccine anymore. Okay, so if I'm going to decide on vaccinating my travelers against, to protect them, I always start off with childhood vaccination. So none of you should be here that have come from foreign countries because you do not have an up-to-date tetanus, pertussis, diphtheria vaccine, polio vaccine. Did anybody ask you? Okay, let's stop picking on Nigeria. You can take it. Okay. Were you asked to provide your international certificate of vaccination and prophylaxis against polio when you arrived at the airport?
Solo fellow came in on the 3rd.
No.
No, at first. Where did you get the 3rd?
Oh, okay. All right. I want to know about a recent traveler.
For module 6. Could have only been module 6.
I'm coming from Sudan.
Yes.
At interpoint to Egypt, they ask for the certification of polio.
For polio?
Yes.
In Egypt?
Yes.
And when you arrived here?
No.
No.
Oh,
okay. Is polio a fake?
Okay.
Public health emergency of international concern.
Yes.
It still is.
Yes.
It has been since 2014. If you go and
So
read
she
in the fine
was-
print, the
they
WHO
were
says if you're coming from a
highest
country that's
of
endemic for
Sonia.
polio
But-
or even
but
circulating
correct.
vaccine-derived polio, you should be asked and you should
Yes.
show vaccination against polio because travelers carry
Philemon.
infectious diseases.
Because
Okay.
Sudan is currently under the
Right. Okay.
outbreak of certification.
So we
of
start with the
polio.
childhood
Really,
vaccines. I'm not going to go through all of
but
them. And then we've
Mercy
got a
was
set of
on
so-called
the
compulsory
on the
vaccines,
10th of
yellow
October.
fever
So those are
and
the 3
meningococcal
names.
disease when you're going where? All the good Muslims?
Shall I
to the
tell
Hajj.
this
Okay. Right.
to
Why?
one
Because we're
person
crowding
that
2 million
you were
people over a
second
6-week period
highest,
in a
but you
tented
only
camp.
completed
And
it
we've
on
seen
the
that we
3rd.
get huge outbreaks of meningococcal disease. Unfortunately, I missed my good colleague,
They got
the
50, right?
veterinarian's talk this morning was I had to go out for something. But anyway, so meningococcal disease, it's not part of the international health regulations per se, but the WHO supports that because of big outbreaks we saw. And the Saudi Arabians actually want to see your flu vaccine and a few other things as well, which makes good sense. And then we get to the so-called travel vaccines. Hepatitis A, typhoid, rabies, Japanese encephalitis, European tick-borne encephalitis. Right. So who in this room who is immune against hepatitis A? Hands up.
And I mean, we did tell them that the cutoff is
Most
the
of you. Who's
31st,
saying
and
so?
Several.
Yes. Where are you from, ma'am? From? Chad. Chad. Oh, okay.
Yeah, we said we're not going to
Okay.
close because we're
So,
going to give
yes.
everyone the
So
opportunity.
90%
Yeah,
of Africans,
they
we
still
grew up we got infected in
Lag
infancy, asymptomatic, we're immune for life. Okay. So hepatitis A, we don't need the vaccine. Okay. All right. But if we haven't had it, who are the South Africans that have not had hepatitis A vaccine? South Africans? No hepatitis A vaccine, and you've not had hepatitis A either. Do you need to go traveling to be protected against hepatitis A? No. We're 60 million people in the country. 90% of us have had hepatitis A. We're immune. 6 million people are not immune. And you don't have to travel to be at risk of getting hepatitis A. Okay, so I'm just scrambling a little bit. So there's the EPI, sort of the small one for Africa. Then there's the South African one. Then there's the American one. They don't only have elections, they have a huge vaccine regime as well. You know, rich people, lots of vaccines. Okay, but what it tells us is that not all EPIs are the same because the EPIs are adopted to the different countries. There's a WHO recommendation, but then there's what actually happens. And in some countries, yellow fever is part of the EPI. It's not a travel vaccine. Who's from Angola? Nobody from Angola. Who comes from a country where yellow fever is part of the EPI? The children get yellow fever vaccine. Sir, where are you from? You stay in Zambia. In Nigeria. Yeah, but not in Zambia, eh? Because Zambia is a bit dodgy with yellow fever. Do you have or don't you have? Oh, Gambia. Yeah, okay, Gambia, of course. All right. Yes, ma'am. Yes. So yellow fever is not a travel vaccine, okay, because some people it's EPI. And if you're Japanese, then you get Japanese encephalitis vaccine as a kid. Who's ever heard of anybody going to Japan getting Japanese encephalitis? Are you at risk? Yes, because the mosquitoes may still be carrying it, okay? But the people are protected. That's why I don't. And then the same thing with tick-borne encephalitis. So that's just the same. Botswana regime, Australian, and South African. It's all small print. But we've got to be careful when we say to somebody, are you up to date with your EPI vaccines? Because if you're South African and Tswana, then you've had BCG at birth. If you come from Australia, you've not had BCG at birth, okay? But we don't give BCG to adults because it's not what it's for. And then hepatitis B is part of EPI in some countries and not in other countries. Okay, so let's talk a little bit about yellow fever. We've really summarized it already because it's two stories. It's an arboviral disease. It normally occurs in non-human primates up in the forest canopies. It's transmitted by mosquitoes between the monkeys. Sometimes the mosquito will come down and bite a human. Then we've got rural outbreaks of yellow fever. If it goes into the cities, we get big outbreaks like we had recently in Africa, a couple of years ago. We had the big urban outbreak. Luanda. Okay, so the Angolans aren't here to protect themselves. So we've got a huge, big outbreak there. It spread into the Congo. So it's a public health thing. What is one of the big stories in the world at the moment? Everybody's talking about global warming. Which mosquitoes are going with it? Aedes, and they spread dengue. And we saw the huge outbreaks in South America with dengue last summer, okay? And we're holding what's going to happen to us in Africa because we've got Aedes all over the place, because we've got plastic bags lying all over the place. And in lucky places like Cape Town, it's full of water. In Johannesburg, all the plastic bags are empty at the moment. They're dry. So we're not going to have an outbreak of dengue. But certainly, so Aedes is the really bad mosquito because it's got dengue, it's got yellow fever, it's got chikungunya, it's got Zika. So we really need to keep an eye on that. So it's about protecting the public. It's about protecting the individual traveler. That's just getting your ICVP. And that's not a yellow card. It's not just for yellow fever. It's for polio. It's for anything else that needs international certification. Those are the areas in which yellow fever occur. I don't have to remind most of you about it. It's 200,000 cases a year, ballpark. A lot of it goes undiagnosed. If you're in a rural area somewhere in Africa, or for that matter in the Amazon, people are going to treat you for malaria, and you either get better or you die. And nobody's going to know you actually had yellow fever. But we know that there's approximately 30,000 deaths a year around there. And that's just a story that you can't read, but that's from cases last year, documented cases in Africa, because I've even had academics say to me, but what's this yellow fever thing everybody's talking about? Never heard of a death. That was a lecturer at one of our medical universities. Okay, right. So we're done with yellow fever. Very effective vaccine, live vaccine, certain contraindications, so you can't just give it to everybody. And then we move on to meningitis, or meningococcal disease. We talk about the meningitis belt. Some of you come from there. Stretches from Senegal all the way through to the Horn of Africa. And then, of course, the big one from a public health point of view is people going on the Hajj or even on Umrah. This is typically where we used to have big outbreaks. I'm sure I missed the talk, unfortunately, but we've made huge differences in terms of what happens there. But if you're staying in circumstances like this is luckily a couple of years ago in Liberia. It's not like that anymore. But for that matter, if you're going to work in a mine in somewhere in Africa and you're going to stay in a mine camp, and you've got like the mine I've just been to where there's 6,000 people working on the mine. So you've got crowded conditions. Mines, police barracks, army headquarters, universities, those are all risks for meningococcal disease. Okay, this is the so-called stephanogram. This is, I think, the original one. This was Professor Stefan that developed the idea of travel medicine in Switzerland. And ballpark, it was very easy 30 years ago when I started travel medicine. 25% of people going to developing countries will get traveler's diarrhea, no vaccine, okay, but amongst there is hepatitis A and typhoid. 2.5% of people going to West Africa in particular will get malaria, and 0.25% of people will get hepatitis A. So those are just the big ones to remember. And then the stephanogram changed a little bit, but traveler's diarrhea is still at the top. There's 0.2% malaria in West Africa, so things have improved from a malaria point of view. Influenza has suddenly climbed the ladder. It's now sitting at 1.5%. So it surpassed hepatitis A as the most common vaccine-preventable travel-related disease. Because we realized with the big outbreak, I think in 2009 already, influenza is always around, okay? And so certainly travelers also spread it, and you go traveling, you go from the northern hemisphere to the southern hemisphere winter, then you're at risk of getting influenza A. And hepatitis A, this is in Swiss travelers, actually went down to 0.03%. And so it carries on. But now we see dengue already cropping up. I think this was in 2018, if I remember correctly. There's 2018. Traveler's diarrhea still at the top. Malaria is still way up there. Influenza. And then we've got rabies, a bite with a risk of rabies right at the top there. But that's not actual rabies. And then we can see hepatitis A there. And now suddenly, we've got those childhood diseases, measles and pertussis, are appearing on the travel health-related risks. Okay, why is that? It's because the world has become too clever, and a lot of people have become spoiled. People in Europe and North America and Canada don't see children dying from measles anymore, like I did in my house job 30 years ago. And so now we're seeing huge outbreaks. This was all pre-COVID. So we all know that COVID even hammered our childhood vaccination campaigns more. And why is measles the one at the front? I think there's one more. This is just, I just put this in for my travel medicine students to say, measles is also not just a childhood disease. This is an aerostess from Israel that died from measles, okay? That she contracted on a flight between Israel and New York, okay? Not third world, first world, okay? And of course, measles is the big one because we all know that the reproductive number is huge. And of course, we've got dear old COVID in there just to get an idea of the reproductive number ballpark of COVID. I know you can argue about it for a day. There's HIV and there's measles. So we know if we see big measles outbreaks, what's going to follow? All the other childhood diseases, because people don't get vaccinated against measles, they don't get vaccinated against the other stuff. So we need to remain up to date in travel medicine of what's going on in the world to be able to advise people on what they should do. What's wrong with these slides? All these things I've just shown. Why is it not really applicable to us in this room? Because it's European statistics on European travelers. We don't have African statistics. We don't know what happens to African travelers, okay? Because we don't have good epidemiology. So this is a nice challenge for all of us, all of you out there, all of you youngsters out there, to go and build good statistical databases so that we can really advise African travelers better than we're doing at the moment. But that's what we've got. So we've got a guideline of what we should be doing. So now if we're thinking about all these things, you can remember this mnemonic. How are we going to decide who gets what? So we look at the destination. Where are you going to? Is there a risk? So if my great aunt is going to Vienna in Austria, she must be up to date with her childhood vaccine. She doesn't need a yellow fever vaccine. She doesn't need meningococcal meningitis. But what do they have in Austria that we don't have? That's a threat. That's vaccine preventable. We mentioned it just now. You go into the woods, into the forests. European tick-borne encephalitis. Okay, and that's also making a big thing, even in squeaky clean Switzerland. People have got a concern now about TBE. You walk through the streets in Geneva and you can walk past the pharmacy and they'll invite you in to come and get a TBE vaccine. Okay, so, but if great aunt is going to watch the opera in Vienna for 2 nights, then she doesn't need a TBE vaccine. But if it's somebody who can't find a job in Africa and is going to chop wood in the forest in Austria or in Switzerland, then maybe you should get a TBE vaccine. So the disease risks. So if you go out and you get this disease, is it just going to spoil your holiday, or is it going to make you sick and mess up your holiday completely, or is it going to kill you? And who is the traveler? So is it an Indian businessman who's going to India on business? He's going to buy a new motor car franchise and he's going to stay in the 7-star Taj Mahal hotel in Mumbai. Well, he should be up to date with his childhood vaccines and we should check on his hepatitis A situation. But that's about it. But if his 17-year-old daughter comes to my clinic and she says she's going backpacking in India to find her inner self, she's not going to the same India, okay? She's going to stay in an ashram and share lots of things with lots of people. And so her risk is slightly different, quite a bit different. Okay. And then we've got to look at the cost. So what is the cost of the vaccine? What is the cost of getting sick? Can you afford to get sick with X, Y, and Z? And then is there a vaccine available now? Effective is it? So where are you going to do what? We've got to be up to date with the geographical spread of disease, what occurs where. It's nice to go to these places and you walk into a little clinic. I think this was in Sierra Leone. And when in the rural clinic, there's a poster up to warn people about Lassa fever, then you say, hey, I can't vaccinate you against Lassa fever, but I better educate my travelers about Lassa fever, okay? And we've got dear old Ebola was up there as well. We look at the traveler. So we want to know about their pre-existing illnesses. What is unacceptable to go traveling? I see people, I've seen a guy who comes in and when he takes his shirt off to get his vaccines, he's going to work on a mine in Mali. He's got this fresh laparotomy scar and you say, hey, what's this all about? Oh no, he had a colectomy for colon carcinoma 3 weeks ago and he's just had his first round of chemotherapy and his white cell count is 1,000. He's not going to Mali, period. Okay, but people are desperate for jobs, so they will try to go. You've got to look at medical, legal, and financial risks, travel health education, then you decide about the vaccines. And this is 2 of my fellow travelers a few years ago, the one from London, the other one from here, a geologist and a security risk manager, and they're having coffee on the veranda of our executive suites in Konokoido in Sierra Leone. And if you look just past Don's elbow, then they're busy digging up the street in front of us for diamonds. Okay. Now, if you're going to a place like that, you probably want every vaccine in the book. Typhoid vaccine is only about 75% effective. It's actually lousy. But after we got 1 bucket of water for all of us to shower and drink and do everything with, I decided I'll have my typhoid vaccine. Thank you very much. Better than nothing. Okay, so we've looked at the CV, the cost, and then vaccine availability. So a couple of years ago, there's nobody here that I can give a bad eye for, but a few years ago in this country, there was suddenly no adult hepatitis A vaccine because the vaccine companies decided there's no market. Now, I just told you, 60 million people, 6 million people are not immune. Let's say half of them are children, so they're still okay. But we've got 3 million people in the country at any time that is exposed potentially to hepatitis A, and the older you are when you get it, the sicker you become. I won't ask all the gentlemen in the room that's as old as me whether they've had the hepatitis A vaccine. There's a couple of them around. Okay, right. So so things are changing. So the whole idea that everything was all nice and squeaky clean is fine. And so we've got hepatitis A, measles. We're not winning the battle against polio. Diphtheria, pertussis, we had a good talk on that. So if we put all of this in the mix, you don't have to remember this, but that's just how do we decide? This is the art of medicine. Who's going to get which vaccine? And then we decide. And then all the non-vaccine preventable diseases, you're pretty much aware of that. Everybody who's coming from outside, who's got travel insurance cover? Hands up, hands up, hands up. And the rest of you? If the car runs you over when you cross the street going to Cavendish Square this evening, what's going to happen to you? You can't go without travel insurance cover. I can't give you a vaccine for that, but you need it. Okay, so we parked this idea a little bit. I'm just looking at time. Things have changed, okay. The castle in the Alps has been overrun by migration. Apart from climate change and global warming, migration is a huge thing in the world at the moment, okay. And so the idea that we're sitting in squeaky clean Switzerland and we go out and we go and travel and we need vaccines for that has gone by the wayside. There's no more castles. We all live in the same global village. One virus affects all of us. Nothing demonstrated that better than COVID, in fact. And one vaccine protects all of us. Okay, we must just make sure once we get it that we hang on to it. It's not really true that migrants bring infectious diseases. A lot of migrants actually pick up infectious diseases in the asylum-seeking centers where they end up in many countries all around the world. So that's the end of my sermon. It's really vaccines for all. Okay, there's no such thing as a childhood vaccine and a must-have vaccine, and it's not just in the public sector. So if we vaccinate the children, then we protect the older people. We heard that this morning with our maternity lecture. Where's Claire? Okay, and if we vaccinate the grandmas, we protect the babies. You can't go and greet the new baby in Australia if you don't show your proof of pertussis or diphtheria vaccination, no? Because they won't allow you in to the maternity home. And then if you vaccinate the travelers, you protect the host country. If you vaccinate the travelers, you protect the home country. They won't bring diseases back. And if we vaccinate the animals, we don't have to worry about rabies. Okay? Because then humans are not going to get rabies. And I think one of the biggest failures we see in this country now is that we always had terrestrial rabies, and now we've got rabies in the seals. And squeaky, clean, boring Cape Town has now become too dangerous to go onto the beach because the seal is going to attack you. And it may have rabies. And it's much more, it's a disaster. The seal colony, I learned 2 weeks ago from the local vet, the seal colony that's affected stretches from Luanda all the way around to Gqeberha, to Port Elizabeth. And so anywhere there, you're walking on the beach or you go swimming like one of my crazy young doctors did last week. She swam 12 kilometers to Robben Island. And she says to me, but the seals always come up to you and they just nip on you. They're playing. Now, last week in Mossel Bay, a seal nearly drowned a guy and then let go of him, and he's got to be treated for rabies. But what if the seal just drowned him? Okay. So we didn't control rabies in the animals. The doctor vet, you're shaking your head. Cease. Okay. But that's it. So these are just from advertisements my clinic set out a few years ago. And it basically says, this holiday, will you go to Romania or are you going to Romania? For those of you that don't know, that's the real South Africa. That's the Johannesburg skyline. Cape Town's not real South Africa, okay? And if you could read the fine print there, you would see we say if you're going to Romania, you need to be up to date with all those vaccines, childhood, etc. And if you're going to stay in Johannesburg for the holiday, you need the same vaccines. That's the message. You don't have to go somewhere exotic to be up to date with your vaccines. And the same there, the British Isles or the Blikkies Isles, you need to be up to date. In this country, that was one of our problems with COVID. Suddenly we're telling adults, you must get a vaccine. It's like you're crazy. We only give vaccines to children. When you're 12 years old, you don't need vaccines anymore. Okay, so we tried to convince the public then that it's different. So here we're trying to tell the young adults that it's not just for them. There are some secrets they should know. That vaccines can protect you against cardiovascular disease, that vaccines can avoid cancer. So you don't just come to the travel clinic when you're going to travel somewhere and the travel agent says you must have a yellow fever vaccine. Okay. So we're all talking about Mpox, and you had a talk which I unfortunately missed because I would have loved to know what's going on there. So is Mpox a travel vaccine? Who says yes? Why? Okay, can we get that on the microphone, Skis?
I think yes, because now monkeypox is classified as a public health emergency of international concern. So I think that the vaccination for travelers should be.
Okay, so how many cases have there been in travelers?
No, I'm not. I'm not
Okay, and how many cases?
I base my answer based on that it's now under public health emergency of international concern.
Okay, and how many cases have there been in the DRC, in the Congo? Who's from the DRC? Is somebody from the DRC? Où êtes-vous? I thought there was somebody from the Democratic Republic of Congo. Okay, doesn't matter. Okay, but there are thousands of cases in the Congo, and they are affecting 50% of the people affected are children under 15 years. Okay, and what is happening in the world? What are we saying? Who should be vaccinated? Men who have sex with men. We've made it a gay disease. It's not a gay disease. It is a public health emergency in the Congo where they don't have vaccines at all. So am I going to give my Lani travelers an Mpox vaccine, but the people there don't have it? Okay, so it's a tricky one. So 3 weeks ago, maybe a month ago now, I still haven't written the report now, I've just handed in the report. I went to a mine in the DRC. It's way up there in the north where the Lord's Resistance Army runs around. And I walked through the whole clinic and I went to the laboratory. We went to the laboratory to have a look. And the doctor said to me, oh, these guys there, they're from the government. They're not part of our staff. I said, oh, what are they doing there? No, they're testing for Mpox. Okay. Oh, okay, they're testing for Mpox, but there's no Mpox in that region. Never been identified. The problem is the government facilities do not have reliable electricity. So they asked whether they can come and do the PCR testing for Mpox in the mines laboratory. That isn't that sophisticated, but it's got a reliable supply of electricity. So these guys were testing 21 specimens from Isiru, which is 200 kilometers away, the capital of the area. They were testing them for Mpox. So, of course, I was very curious because I've been telling Nagy here that we need to vaccinate our travelers going to the Congo. I need to vaccinate mine workers that are going to work on this mine. Because can you imagine the chaos when it hits the newspapers that Oom Giep came back and he had monkeypox? And not because Oom Giep went to a nightclub, because Oom Giep works in a dark tunnel underground and he's rubbing against the guy next to him who comes from the village, whose kids have got monkeypox. And not everybody's got horrible blisters all over the place. It can be very subtle. It can look like chickenpox or like herpes zoster or like anything else. Okay. So I wanted to know, so did they have any positive specimens? Now, I did have the circle around you. You see there? There were 3 positive specimens in that group of 21 specimens they got. So there isn't monkeypox in that area, excuse Mpox in that area. But what have we now got? So the next question is, you guys, have you been vaccinated against Mpox? You're working with the specimens, and I can tell you that the hand wash facilities in that clinic is not great, okay? And the answer is no, because Mpox vaccine only arrived in the DRC the week before. It's available in Kinshasa, but it's not available where this mine is. Okay, so it's just, and there is, in the same week, October the 18th, the CDC is saying people traveling to the DRC and to Central African Republic and to Rwanda should be vaccinated against Mpox or should be offered the vaccine. So in America, you can now walk into any travel clinic and buy a Jynneos vaccine. But we haven't got it available in the places we need it most in Africa, and I still can't give it to my travelers and to my mine workers. So some of our problems is this. It's just a little bit, who was the philosopher at the back here? Okay, private-public disconnect. We don't talk to one another. We don't share information. There's the academics, and there's the public sector, and then there's the private sector, and nobody talks to one another. But we're all on one planet, one health, same ark. So it's no, if there's no animal vaccines, then there's no food, there's rabid dogs and dead people. Okay, so, and these are all the problems that we face in deciding what to do. So a visit to a travel clinic anywhere in the world is actually a public health opportunity. It's an opportunity to add a little bit, a very small bit, one drop. Because South Africans, as a rule, don't go to their GP and say, I want to update my travel vaccines. They can't show, with the exception of one lady in the audience, can't show their childhood vaccination certificates. So when people come to us and say, the travel agent said, you must go and have a yellow fever vaccine, we say, what about your other vaccine? And the one disease that's got nothing to do with you, because we're talking vaccines, but the one disease I still cannot vaccinate you against is malaria. And the biggest killer of expatriates and frequent travelers in Africa is malaria, malaria, malaria. Okay, it's still malaria. And what's that? That is, don't get bitten, seek early treatment, take the pill, and yeah, easy as that. Okay, but I can't vaccinate you against that. All right. If you're interested in travel medicine, if you want to know a little bit more, there's a couple of books published by SASTM, the South African Society of Travel Medicine. And then there's the CDC Yellow Book, published by the Americans. And it's available online. So if you want to check something quickly, you can go and search that. You can get that on the CDC. You just put in CDC Yellow Book. And then, of course, there's a publication by the WHO. And that's the difference between the two. I've been neglecting you guys a little bit. So that's the WHO, and that is the CDC one. The WHO one is very much public health orientated. What is polio? Here's the fact sheet. What are we doing from a public health point of view? The CDC in America has got a nice public health viewpoint, but it also looks at individual travelers. And even without Trump being in, there's a lawyer looking over your shoulder, okay? So it's sometimes a little bit more aggressive in their preventive measures. So it's a nice reference to just look at the two of them together. If you want to know a little bit more about travel medicine here, SASTM, the South African Society of Travel Medicine, and once a year we run a travel medicine course. The next one will be in May next year. That gives you a certificate of competence in travel medicine, and that's backed up by the University of the Witwatersrand. And I think this is a record because I'm done. Thank you very much.
Thank you, thank you, thank you very much for a very lively and interactive presentation. I guess you don't have time for a question.
I can do 5 minutes of questions.
5 minutes of questions. Is there anything online?
There is one, I think, comment question. Adaura is talking about dengue fever, and he's wondering whether there are vaccines available in Africa for dengue.
With the exception of one clinic that I won't mention in Dar es Salaam, who admitted to smuggling in dengue vaccine. The vaccine is not available in Africa as far as I know, unless anybody can put a hand up and say it is. There is a vaccine available, Qdenga, in Europe and in North America, and again, it is freely available and people can buy it, but we don't have it. And I was told very clearly by the manufacturers that our market is not big enough, and that they can't keep up with making vaccine. So that remains a challenge. So who was talking this morning about making our own vaccines? We really need to get there, but I think we're still a little bit off. Yeah.
Thank you so much for the glad presentation. It's very nice presentation. Just I'm glad that you mentioned the issue of availability of the vaccine, and there is more need to be done in Africa, really, because it's under the IHR still, but want to come to decide that like in my case, as I mentioned, that now polio vaccine is recommended to be taken before from the host country like Sudan, and want to come to Egypt. I feel that it still needs to be more issue to be done regarding the burden of the disease, the availability of the vaccine, because requesting IBV vaccine, which is not that much available now. So as you mentioned, more to be done in this area, really. Thank you.
Good. Thank you very much.
Thank you. Thanks very much. Is there another?
There's a question on that side.
I just
Okay.
wanted to thank you again for this. And then obviously I wanted to ask about how do we advocate for migrants? Because for instance, in South Africa, there are a lot of people who are adults who have come into the country. They don't go to travel clinic, but they travel back home quite a lot. And they often forget that they're at risk for malaria. And they struggle to get prophylaxis. They don't consider vaccination because for them, they're not traveling, they're going home.
That's a huge challenge. So in travel medicine, we talk about VFRs, visiting friends and relatives. And that specifically refers to this type of traveler that have settled here, either formally and legally and officially. So that's been one of my big fights for the last 2 years, is that on our national guidelines for malaria, it's mefloquine for pregnant women, and then there's atovaquone proguanil for children of a certain age group. But we've not had mefloquine or pediatric atovaquone proguanil available in the country for the last 3 years, because the companies that either bought the rights or imported them just ran out of tablets and felt that it wasn't financially viable and just disappeared off the market. So we are now now the problem is, if an American wants to go on safari to Kenya and she's pregnant, you can tell her she shouldn't go because she can postpone her holiday. But when I've got a woman from Gabon who's been living in Johannesburg for the last 10 years, she's got 2 little snot noses, and her father died in a village in Gabon, she will go back for the funeral. And she's not going to stay in a 5-star air-conditioned hotel. So I really want to be able to protect her with mefloquine, which is the only thing that is safe in pregnancy, provided you don't have other contraindications to using it, like psychiatric, etc., etc. And now we have to bring it in under Section 21, and where the tablet used to cost 35 Rand, it now costs 125 Rand for the same tablet. But I've got a choice. Either she pays that, or she doesn't go, or she goes without protection. And so we've got a huge problem with things like that. So the same thing goes for vaccines under certain circumstances that is availability. And in most European countries, for sure, and in large parts of the Americas, travel medicine is based in academic hospitals. It's infectious disease based. And so when people get sick and they come back, they end up in an infectious disease hospital, which is why they've got excellent statistics on what happens to travelers. And they can show that there's a problem. Here, people are too scared to go to hospitals because they don't have official travel documents. And they don't have the money to go to the private travel clinics. And even if they do go to private facilities, and we've got a reporting system, I'm not sure that our reporting is always as good as it should be. So we don't really know what's happening. So again, we're back to malaria, but we're recording falciparum malaria, which is 95% of malaria in Africa, and it's the biggest killer, etc. But the other 5%, we're seeing snippets of there's a Vivax and here's Nuvale. But the Department of Health don't record non-falciparum malaria. So if you ask statistics about, so how many cases are we actually seeing? Is it increasing? We can't tell them. And similarly with diseases like meningococcal disease, we don't always know who goes where and comes back with something like that. Salim Parker does a great job here with UCT. With the Hajj travelers, we've got a very good idea of what's happening there, but that's a very defined group. With migrants, we don't know.
Thank you.
Thank you very much.
One more round of applause for Albie. Thank you very much. And it's lunchtime now. You are free to have a good meal. Not quite yet. Okay. Oh. It's quiz time. Sorry about that. It's fun time.
Okay, it's not getting your prizes now, but it's time to earn prizes.
Salim, while you set up,
Yeah.
just one more of our colleagues that didn't get a chance to introduce himself. He joined us yesterday. Can we have that? Is that okay?
Yeah.
Okay.
Yeah. So I am David Shakusise. I'm from Sierra Leone. I work with Clinton Health Access Initiative as senior associate, and I lead new vaccine introduction, malaria vaccine, hepatitis B bad dose. Thank you.
Can we all come in? Sit down. All right. Okay. So good afternoon, ladies and gentlemen. Welcome to the fun session for the day. Sorry you missed me on Monday. I also missed you. Alana said I've been upgraded, but actually I was just running an errand. Okay, so yeah, I was talking about Monday. So we are going to have the Energizer, but not quite yet. My colleague Edina says we must test the brains first before we exercise the body. How many of us agree to that? I think we should exercise first. My thoughts exactly. How many people support that we exercise the body first? All right. Excellent, excellent. But then, I'm a not so new addition to VACFA. I don't want to get fired, so I'll just do as the boss says. So, Edina, are we ready with the quiz? Salim, over to you.
You want the mic with you? Or I'm going to use this one. It's fine. Welcome back from lunch, everyone. I hope the food was good. It was? Good. Good. Okay, we just like Elizabeth said, we're going to have a quick quiz to warm the brain up. Make sure you're listening to the next two talks. Are we all connected? Anyone? Okay. No, we have time. We can take 2 minutes and wait for everyone. We don't have time, but we're gonna. On this. In your classes. Okay, we good to go? Let's go. First question. Okay, we're not good to go yet. You good to go? Good to go, everyone? Remember, I've been repeating it, fast and correct. True or false? The COVID-19 vaccine changes your DNA to RNA. There's some chuckles from the participants for this question. Are we going to be surprised by the answer? False. Are we surprised that 2 people chose true? Guys? Okay, it was a mistake. It was a mistake. You're forgiven. You're forgiven. Okay. Next question. The following vaccines are recommended for pregnant women, except which one is not? Five seconds left. Okay, let's find out the correct answer. Does anyone know? MMR. Correct. 58% of us was correct, and I'm adding myself to that.
The online people said 2. The online
2.
said
Influenza? No. No, no, no.
Sally,
Yes?
can I interrupt you? For those of us who said MMR, why is MMR not recommended? All right, because it was a chorus answer, you won't get a gift. Next time in my class, if you know it, you put up your hands. We do it the old school way.
Okay, next answer, next question. Which of the following vaccines are recommended at birth? You have 5 seconds left if you haven't chosen. Okay. Correct answer. All of the
All of
above. Correct. Well done.
online people, yeah?
Okay. Let's go to the next question. What is the most successful and widely used travel vaccine? Were people listening to Albie's talk? Did he say? Okay. He did. He did. Okay, some people weren't listening. Five seconds left. The correct answer? Okay, people are listening. Nice. Good. Well done. Next question. True or false? You only need to get the HPV vaccine if you're sexually active. Okay. How many questions are there? 10. This is 5. False. You don't need to be sexually active and get it. Cool. Okay. Next question is, which of the following confers passive immunity? I see people are thinking about this one. Okay. Times. Yeah. Time's up.
No, I was just saying the online people are saying 2 and 3.
2 and 3.
Good responses for both, yeah.
But the correct answer is
3.
3. Only 3. I was going to try and explain why only 3. But does anyone want to? Okay. I'm not going to try if it's... Okay, let's go to the next question. Development of a safe and effective HIV vaccine has been challenging because you have 10 seconds left. Okay, time's up. And the correct answer? 4. That is very close to 100%. That one person, we're going to find you. That's a mistake, sorry. That's a mistake. Yeah. Okay, question 8. True or false? The HPV vaccine only protects females. Five seconds. 3. And is it true or false? False. Good. Now we need to find 2 people. That was okay. 2 more questions left. Why do we need boosters for some vaccines? Ten seconds. Okay. And the reason we need boosters is to maintain immunity over time. So people who chose all of the above, right? To protect against different serotypes, to maintain immunity over time, and to sustainably weaken a virus over time. Why is it not all of the above? Che, tell us, why is it not all of the above? There we go. It's because the quiz says 2 is correct. They
No.
all... Okay, 2
Objection,
correct.
my lord. That answer is not correct. It isn't, because you cannot sustainably weaken the virus using a vaccine over time that has been administered. So that answer is very wrong.
Is it wrong?
Booster for which vaccine?
Okay, the brains are going on. The brains are going on. This is a debate we're going to have for another time. Feel free, tea times, have this debate. We have one more question. So let's just focus on the last question.
Yeah.
Get it right, correct, and fast. Are we ready for the next question? Let's go. True or false? Vaccines cause autism.
Now we know that who that one
We
person
found the
is.
one. We found the one. 5 seconds left. Okay. And true or false? False. Yes. Oh. Oh. Are those 5 people in the room with us right now? It's online. Okay, it's online. Okay. 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 So let's find out who was the fastest and most.
It's not online. The online
Not
are
online.
saying false. Yeah. Yeah.
Who's our winner for this quiz? Two more. Well done. Okay, cool. There we go, over here on the side.
All right. Salim had stolen the show, stolen the time as well. So now we don't have time for a energizer. But you know, I do have a gift, some gift. It's not as wonderful as the ones Salim and Edina had promised, but they are gifts all the same. And I would like to say I got that from my father, who gives and gives and gives. The Bible says, for God so loved the world, he gave. So, yeah. So now, for you to get my gift, I want the honest person who honestly got one of those 10 questions wrong and is willing to say, I got it wrong. Okay, let me rephrase the question. Excluding Greg. Thank you. Please step up and get your gift. Shall we jam our hands together for her? No, 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 no. That doesn't count. Change of mind. Change of mind, change of plans. I want someone who actually didn't make a mistake, who honestly thought the wrong answer was the right answer. Okay. Now there are so many hands and fewer gifts, so now I have to filter. Okay, so who will be honest enough to tell us the question they got wrong? All right, let me give her another. Sorry? Microphone, sorry. Seppi has one, I have one. Yeah.
So I was saying maybe I mixed up my immunology course yesterday. So when the question is out, despite me not logging in, I said it's all about, because I'm like, okay, the first one is against other serotype, and then is to maintain immunity continuous. That is the reason why I choose that it's all about.
All right, please bring the mic with you and let's jam our hands together as she steps up to collect her. Voila. So Crime doesn't pay, honesty does, hey? Okay, beautiful. Now I can't find my boss, Rudzani, because he put me on the spot by asking me, which day are you doing your jokes? So now I feel obliged to do jokes. All right. So in Wariri, where are you? All right, in his country, and that's a joke, because it happens to be mine also. In his country, a teacher walks into class and asks the students, guys, would you know what we are going to learn today? And the whole class said, no. So the teacher snugly smiles, walks out of the class, and on his way says, there is no point in teaching you what you wouldn't know. The next day he came back. The class was wiser. Would you know what we are going to? They all said, yes. He walks out snugly again, telling them what, Wariri? There is no point in teaching you what you already know. And then the third day, they were ready for him. So, smart guys, you know we are smart in that neck of the wood. Pupils, would you know what we... Half the class said yes, and half the class said no. Now, for a gift, who knows what the teacher said and did? Excellent. Step up, Amina. Now we know those people who are from that country. My sister. Nepotism, nepotism, nepotism. Step up, step up. Okay, just to satisfy the gentleman, I'm giving her a small one. All right. So now, oh, sorry, the last part. Okay, our answer was that the smart teacher said, let those who know teach those who don't, and walks out of the class. So it's my pleasure to introduce to you the next speaker, who will teach both those who know and those who don't. You know, one last joke about this speaker. I asked him, can you please provide me with your bio, or do I have the liberty of making up stories by myself? The rate at which he generated the bio was astounding. So he's currently an independent consultant and research associate with various universities. Left WHO last month, where he had worked for 17 years as the regional advisor responsible for coordinating surveillance for vaccine preventable diseases, focusing on disease targeted by new vaccines, the rotavirus, the PCVs, and of course, COVID-19. He supported the introduction of rotavirus vaccine in 39 out of the 47 countries in the WHO Afro region, and in 41 out of 47 countries. During his tenure at the WHO, he also established robust disease surveillance network for rotavirus, diarrhea surveillance, bacterial meningitis, pneumonia, COVID-19, typhoid, and so on. These networks have been instrumental in guiding informed decision-making on new vaccine introduction, monitoring the impact and effectiveness, as well as the safety and monitoring of many intussusception. He is, I don't want to mention his name yet, he is passionate about documenting the public health impact of vaccination, as well as capacity development for vaccinology in Africa. And today he will be giving us the talk on the new vaccine introduction principles and operational consideration. Okay, before I ask you to join your hands together and welcome him to the podium. I'd like to add that he's also a new addition, or it's a prospective new addition to the VACFA team. The husband of one wife, I believe. Let's jam our hands together for Prof. Jason. Wanda.
Thank you very much, Elizabeth, for that kind introduction. Just one disclaimer, one wife. And please, I hope you're not, you did not record what she said, because I might not be welcome home. So thank you very much for this kind of introduction. So today, or this afternoon, we'll look at the new vaccines introduction and focus on the principles and operational considerations. I actually thought that Elizabeth will give the presentation on my behalf, so I can walk out of the room. But anyway, thank you very much. And thanks a lot to the conveners for inviting me to give this presentation. Ben, Samuel, Ruzani, Endina, Professor Greg, and all the colleagues at the VACFA. And congratulations for the good work that you're doing for this continent. We are really very proud of the work that VACFA and NICH are doing, and UCT, to build the capacity for vaccinologists in Africa. So this afternoon now, I will talk about the new vaccines introduction. But first of all, how many in the audience have ever been involved in introducing a new vaccine or vaccines? Wow, there are not many. 1, 2, 3, 4, just 5. Wow. Any new vaccine, any new vaccine. We are not many. I thought we were actually more than that. But the good news is that we have a few. So what we'll do is I'll make the presentation on your behalf, and you can answer all the questions at the end of the presentation. So you've just been promoted. So first of all, look at the vaccine preventable diseases that are targeted by specifically the new vaccines. And I think the last 3 days we gone through some of these vaccines. So I think most of these will be familiar to you. But I think also try to look at, in terms of the introduction of the new vaccine, this is an area that it is quite critical, because they're not enough to do the clinical trials, have these very efficacious and safe vaccines, and we don't get these vaccines into the program or into the EPI. So it's really important. And I think over the years, as you saw in terms of the landscape of the vaccines, there have been a lot of vaccines that have been introduced into the EPI, which are having a major public health impact. So it's good that we look at the experiences of how we've done in the past and how we can improve in terms of deploying these vaccines into the national immunization programs. We'll have some of the examples, and I'm sure many of you can give experiences that you have from your countries, and then the take-home messages. This is a recap. I think Professor Gray and others went through this, so we'll not spend so much time in terms of the clinical trials. But I just wanted to say that what we are talking about in this presentation are the new vaccines, not the traditional vaccines, and how to deploy those vaccines after the clinical trials that actually establish the vaccines are safe, they're efficacious, and they can be deployed because they potentially have a public health impact. In terms of the policy and the guidance, Professor Gray and others mentioned that WHO has a group of experts which is called SAGE. How many of you know what SAGE is? SAGE. Yes, please. SAGE? Mike? Strategic and Advisory Group of Experts.
For our online audience, could you kindly repeat that
hear.
using the mic, please?
strategic advisory group of experts.
Correct. And these are drawn at the global level. They're drawn from different countries. These are experts who help WHO in terms of reviewing the data that is being generated either by pharmaceutical companies or other groups that are involved in clinical trials. They look at that data, and it's a very vigorous process just to make sure that the vaccine is doing what it's supposed to do. In other words, it's efficacious and it's also safe. And then look at what could be the potential schedule for that vaccine in terms of using that vaccine in the National Immunization Program. And based on the SAGE recommendation, then WHO pre-qualify that vaccine. But also there are vaccines that are not necessarily pre-qualified, but granted what is called emergency use listing, mainly for vaccines that are used for responding to emergencies or pandemic like we had the COVID. So the COVID-19 was not pre-qualified, but it was given or granted AUL in order to be used, meaning that the SAGE actually looked at the data that was coming through from the clinical trials and were convinced that these vaccines will have a public health impact. So these are some of the special considerations in terms of policy and guidance, just to say that it's not just any vaccine. And in the event that the dossier does not meet the criteria, then that dossier will be sent to whoever pharmaceutical company was developing that vaccine, that the SAGE and WHO feels that this vaccine does not meet the criteria to be deployed into the National Immunization Program. And then subsequent to that, WHO will issue what is called WHO position paper. And I think also Gray also showed some of the links. And for each vaccine in EPI, if you go to the WHO website, you'll have a position paper that describes how that vaccine, in terms of, to describe the disease burden, to describe how that vaccine is supposed to be used in the EPI. So these are very important resources. And this is just to recap as we go into the deployment of the vaccine. And in terms of the newer vaccines that have recently been introduced, I think you all know the rotavirus vaccine, PCV, malaria vaccine, which is a new kid on the block, HPV, and others. And in terms of the introduction, you can introduce through pilot, like the malaria vaccine, for example. You can do a phased introduction. In other words, you start with either few districts or provinces, and then you scale it up as you go forward. And the reason for that is that sometimes you feel that you don't have the necessary infrastructure to be able to deploy the vaccine because of the size of the country. So countries like Nigeria, for example, you look at how they introduced PCV, for example, it was not countrywide, but it was phased introduction starting with some of the states and then scaling up. So it could be either through pilot, phased, or nationwide introduction simultaneously. For smaller countries like Rwanda, normally they do that. You can also do mass vaccination campaigns, and also more recently, as it was highlighted in the previous presentations, there are also switches of the vaccine for various research. I think the PCV was mentioned, where we started with PCV 7, went to PCV 13, and now we do have even PCV 15, although this has not been deployed in the African region. So the switch is also an issue, even though mostly when you talk about the switch, you think it's very simple, but it's actually like an introduction. We've had countries that have been switching the products, rotavirus vaccine and also PCV, and it's quite an intense process as well. So even when you are planning to switch, you also have to go through the preparations, just like an introduction of a new vaccine. And then once the vaccine is introduced, of course, WHO recommends that within the 6 and 12 months, you need to do what is called PI, post-introduction evaluation, to learn in terms of the programmatic implications when that vaccine was deployed. Well, there are some good lessons to learn, and there are some mistakes that were made when the vaccine was being deployed, so that you can be able to make adjustments. And that's why you have to do it within at least 6 months, maximum maybe 1 year, so that you can make adjustments to make sure that the vaccine is deployed in a way that it provides a desired impact in terms of reduction of the disease burden. You can also do the impact evaluation. These are, and also effectiveness studies. I think it was mentioned in some of the vaccines that were discussed earlier. The purpose of these is mainly to understand whether the vaccine that has been deployed, and this is usually what we call the real-world impact or effectiveness. Because when you're doing the clinical trials, it's very controlled setting. So there are certain things, sometimes even the target population is not exactly what you encounter when you deploy that vaccine in the EPI. So we've been talking about EPI. When was the EPI launched? Sorry? 1974, more than 50 years ago, or close to 50 years ago. Many of us in this room, not me, not others.
All right.
Many were not born at the time.
Sorry, Prof. Amina, you got it right, but because you did not give me the luxury of calling you no gift, but because I want Gavi support, there is a gift. So let's jam our hands together for Amina for getting it right.
Thank you very much. Yeah, safety monitoring is also quite important. Of course, this is done during the clinical trials, but even after the vaccine is deployed in the EPI, you also need to do the safety monitoring as part of the post-marketing surveillance. Okay, so in terms of the vaccines or the vaccine-preventable diseases, I will not spend much time on this because we have gone through this. But right at the beginning, 1974, there are only 7 vaccines targeting 7 vaccine-preventable diseases. And currently, we have how many? Close to 18, maybe moving to 20. And there are many more coming up. So this is quite, it's getting exciting, as Ben said during the introductory remarks during the first day. The immunization area of work is becoming very exciting, but also very complex. Because when you look at it, the immunization system in some countries is very weak. So really being able to take up these vaccines and really make sure that the vaccines are being used properly to have the desired impact is not very easy. So it's becoming really, really complex. And you can see some of the vaccine preventable diseases on this slide that are targeted by these vaccines. So we've done very well in terms of development of the vaccines and deployment of these vaccines. And I think right now it's really focusing on making sure that we drive the desired public health impact. In 1974 and over the years, it's been EPI, which is Expanded Program of Immunization. But these are sort of evolved. We now refer to EPI as Essential Program of Immunization. To really highlight the importance of this program in terms of the public health impact. So as you can see on this slide, we have those traditional vaccines. We also have the new vaccines. But let me pause here and ask, because I'm very passionate about the rotavirus vaccines. I've been working on rotavirus vaccines for a long time. Which was the first country to introduce rotavirus vaccine? Anyone? In Africa, the first country to introduce rotavirus vaccine. Hey, Ben, we are not doing very well here. Ghana? Are you from Ghana? We've had this debate before. Was it Ghana? Yes. Which country? Sudan. Okay. Okay. I think, who said South Africa? Excellent. I think you'll be the only one who will be allowed to leave the country. South Africa. So Ghana was the first Gavi eligible country to introduce. But in terms of the African region, it was South Africa. And many of you may have seen this photograph. This was the Minister of Health at the time. And it was quite interesting the way the rotavirus was. Actually, South Africa introduced PCV and rota almost at the same time, which was quite a remarkable achievement because it's really kicked in the introductions of rotavirus vaccine in the African region. But also I wanted to mention that in terms of the new vaccines introduction, what we've observed and what we encourage is that high-level engagement in terms of the introduction. You can see the minister here was one who administered the first dose of rotavirus vaccine in South Africa and many other countries, which is really a good thing. And you can also see that following the introduction of the rotavirus vaccine in South Africa, this is the coverage over the last 4 years. So South Africa is not doing too badly in terms of the coverage. The latest figure is 78%, but it's just been about 80%, which is really good. And many of the countries, except a few, where in terms of the coverage, we are struggling quite a bit. And then 10 years later, of course, Uganda also introduced, and you can see the coverage. So these are some of the countries where you have very strong health systems. You introduce the vaccine, and immediately the coverage goes up. But we also have disparities in terms of the coverage in a lot of countries. So these are some of the lessons that we've learned in terms of deploying the vaccines. And what I'm really trying to get to is how can we do this in a way that it is effective and it has the desired impact. So on this slide here, we're showing some of the key elements of the new vaccines introduction that make the introduction effective and successful. So we have the guiding principles, and we'll go through some of these in the next set of slides. The planning is extremely important, and also assessing the readiness of the national immunization program and also the health system in general. The decision-making process, the opportunities, and also the current and future perspective. So these are some of the slides really to emphasize some of the important aspects of the new vaccine introduction. One is to remember that you are deploying this vaccine within the context of the health system. And what we don't want is for that new vaccine to have a negative impact, because it could have either negative or positive impact in terms of introduction. Negative means once the vaccine is deployed, then you're actually weakening the immune system. You really don't, the health system rather, to weaken the health system. You want that introduction of the vaccine to be an opportunity to strengthen the health system. It's challenging, but that is the primary goal. So to make sure that the new vaccine strengthens the immunization program and also the health system. So for those of us who have been involved in production, how did you do it? A few of them. Colleague from Ethiopia. You said you have been involved in introduction of the new vaccine. Can you maybe share some experience on how that was done and whether you feel that introduction strengthened or weakened the health system?
So I take it as a positive because it has strengthened our API program. For example, recently we have switched Rotarix to, Rotarix to Rotacil, which is a 2-dose. It was a 2-dose vaccine, and we switched to a 3-dose vaccine because of the global production shortage. So we take that opportunity to also include other vaccines to strengthen, to provide trainings, to include trainings and provide the training. And overall, we take that chance to train the whole country's health workers that are working on EPI to just aware about the COVID integration, the Rotacil and other vaccinations, and we are doing pretty good right now. So I think it's a positive thing for us. And we are also planning to introduce other vaccines in the near future, including the hepatitis birth dose. And I hope the experience that we have on the previous introduction will help us a lot in the future.
Thank you very much. I think this is really important because when you think about some of the vaccines that are in the pipeline, which the countries will consider, if you introduce a vaccine, a new vaccine, and it's having a negative impact on the health system can actually collapse. So we need to do very well planning. And the next set of slides is perhaps to just look at some of the key parameters that are important. One is planning is extremely important. Planning is extremely important. And these are the 5 key elements that ensures that the vaccine is successfully deployed, and to ensure that actually that deployment of the vaccine strengthens the health system overall. One is the decision-making process, which is extremely important, the planning, the prioritization, how this is being done, and also ensuring that there is that high-level government engagement and leadership is extremely important. Coordination is important, integration, ensuring that whatever you do, you are not establishing a vertical system, but it's part of the existing system. It's extremely important. And making sure that you are also strengthening the existing system. Personnel is extremely important. Ensuring that you have well-trained and motivated workforce. I think earlier we talked about the workforce and ensuring that they are well-motivated and well-trained. The cold chain is extremely important because most of these vaccines, as you know, you have to maintain the cold chain logistics and waste management. All these are key important aspects to consider. Waste management, ensuring that the syringes that are being used are also managed properly without contaminating the environment, monitoring the safety. And surveillance is extremely important, as we said, for monitoring the impact and the effectiveness of these vaccines, and ensuring that as you establish surveillance for these new vaccines, it's part of the integrated disease surveillance. Resource allocation is extremely important, and also ensuring that you are maximizing the opportunities as you deploy the vaccine. Budget is extremely important also to ensure that you have planned and adequate resources and then funding is allocated for the introduction. Vaccine supply, we mentioned that to ensure that for Gavi-eligible countries, at least the Gavi pays for the vaccine, but there is also the co-financing and also the budget for the operational costs, ensuring that all that is in place. So how do we ensure the readiness for the introduction of the vaccine? So I have a few slides. I will not go through every detail, but just to highlight that it is extremely important to conduct an assessment in terms of assessing the readiness of the program to actually take that vaccine. We just had, I think last week, for the first time, Chad introduced the vaccines simultaneously. Previously, we had countries introducing 2 vaccines at the same time. Ghana did Rota, South Africa did Rota, PCV, Ghana did Rota and PCV. But Chad last week introduced 3 vaccines at the same time. Last week, introduced malaria, rotavirus, and PCV. And those of you, I don't know that you have anyone from Chad. It was quite, yeah, from Chad, yes. Maybe you can share the experience at some point. But I think that's really amazing how Chad was able to do it. And hopefully, the introduction of these vaccines at the same time will strengthen the system. I'm not sure whether you did the readiness assessment to make sure that no? Yeah.
Actually, I also learn from it online while I'm here. So I don't have like practical, but for what I understand is a conjoint the government with some partners that motivate to realize that you should launch, but I'm not specific on anything else. Yeah. But for malaria, I know it that is going to happen for a while. Thank you.
Thank you. So Chand introduced 3 vaccines at the same time. So that is quite a lesson to learn, and maybe we inform future introductions. If Chand can do it successfully and doesn't have a negative impact on the health system, I think with the other new vaccines that are coming up, maybe this could be something that could be considered. But just to say that is not all the countries can do that. I think for some of the countries where the health system is very weak, introducing more than 1 vaccine at the same time can be quite quite challenging, and it may have a negative impact. So in terms of assessing the readiness, there is a checklist of some of the critical things that need to be placed before you even introduce the vaccine. One of which is, of course, having a very well written plan with a budget, ensuring that the NITAGs are also fully functional to support their decision making, and making sure that the government is fully involved. The other one, of course, is to make sure that you have assessed the system and that the introduction of that vaccine will have the desired public health impact. And these are some of the critical elements that you need to look at and make sure they're in place, one of which is the national immunization strategy, what we use called multi-year plan those days, and making sure that the plan is updated. You have a system for monitoring the coverage. You saw the coverage for Uganda and also South Africa in terms of retrovirus vaccine, but making sure that you have the necessary tools, you have the necessary expertise to be able to collect the data that you can be able to monitor the coverage. Because it's not good enough just to introduce the vaccine. You need to make sure that you have the system in place to monitor that the vaccine is having the public health impact in terms of reducing the disease burden going forward. The other one is, of course, we talked about the training, which is extremely important. Plan the training ahead, making sure that you're also integrating, you have enough and adequate resources distributed across the district. If you are doing a pilot or phase introduction, making sure that in every district or province you have enough workforce that can support the introduction of that vaccine. There are, of course, the resources like this course, for example, building the capacity for vaccinology, MLM, which are medium level training programs that are there to build the capacity. Implementation research is also important, maybe hacking institutions that can be able to support, so that as you deploy that vaccine, you're also gathering a lot of information that can help strengthen the system and also that can inform future introduction of the vaccine. There are cases where the turnover of staff is quite high, and that is something also to anticipate, that some of the critical staff may leave, the staff that you have trained, to make sure that you have a continuous program for retraining and also replacing the staff. Cold chain is always a challenge, because for any new vaccine, and some of these newer vaccines take quite substantial space in the cold chain and capacity. So making sure that you do the readiness assessment of your cold chain so that you have adequate space, and if you need to replace some of the equipment, you can replace the cold chain equipment, and making sure that also the transportation across the country, because you have to move the vaccines from the central level to the district and the provinces. So it's quite a complex exercise. And what I'm trying to emphasize here is that if the planning is not done well, you have not done the assessment, readiness assessment using this checklist, then you'll be stuck. And in most cases, you may have a vaccine that maybe the car breaks down, and the vaccine, and these are very expensive vaccines. So what you don't want is a situation where by the distribution process of the vaccine is affected, and the vaccines cannot be transported to the places where they need to be, and even when you get there, the cold chain is collapsed. And we've seen this in most of the countries, because again, the electricity, we have problems with the electricity and all sorts of things. So you need to make sure that you have done the assessment before the deployment of the vaccine. Monitoring of advanced events, we are the presentation on monitoring of safety, making sure that you have systems to monitor the safety and also managing the waste from the vaccines, making sure that the syringes. Well, it was in Kenya, I think, where there was a big challenge because of the restructuring of the government, where you have the central government and then the county government. And what Kenya did was that to sort of decentralize the services. So the central government was buying the vaccine, but then the syringes, it was the county government. So we ended up with vaccines, but no syringes, no essential supplies to be able to deploy that vaccine. And it was total mess, and that even affected the coverage. There were disease outbreaks during that process of devolving the type of government. And these are some of the issues that we really need to take care of. We talked about the disease surveillance and also the monitoring. I think we've already captured this, making sure that you have enough tools and the tools have been deployed for capturing all the essential information about this vaccine. And you can go through some of these slides, just highlighting some of the key elements for successful and effective deployment of the vaccine. Financial sustainability, 15 minutes, thank you. Financial sustainability is extremely important to make sure that the government has allocated the budget and where you can mobilize the in-country partners also to start to support the process. These are some of the groups or agencies that can support in terms of the informed decision making. The NRAs, the National Regulatory Authority, the NITACs are extremely important. The intercoordinating agencies are extremely important. Health Sector Coordinating Committee and the in-country partners are extremely important so that we can mobilize the expertise and also the resources to ensure that you have a successful implementation. I will not go through much in terms of the NITAC, but the NITACs are extremely important. And really continuous building the capacity of the NITAC is extremely important, the work that VACFA here is doing and niche to make sure that the NITACs are well trained and they have the knowledge to be able to support these processes, because making decision is not easy. They have to look at the temperature position paper, interpret that data to be able to support the deployment. Planning, of course, we mentioned how important that is. The budget, these are some of the elements that really need to make sure that we are in the plan. Planning for training, planning for personnel, planning to make sure that the cold chain and all the equipments are in place, and really making sure that in terms of the budget, some of these key elements are there so that you don't run out of resources as you deploy that vaccine and going forward. There are also opportunities that can be used as you deploy the vaccine to strengthen the system, and these are some of the key areas that the deployment of vaccine can strengthen the immunization system. Regulation is another area. As you establish the regulation process for new vaccine, you can make sure that this is integrated and also building on the existing systems. The planning and budgeting, service delivery, advocacy, coordination with other services, so that you are not doing this as a vertical activity, but more integrated. Human resource training, when you train, you're not just training on one vaccine. Of course, you focus on that, but you can also expand the training so that you're building a broad knowledge in terms of the expertise. Cold chain, again, as you do this, it's an opportunity. When you do the readiness assessment, you're also looking at maybe this time you're introducing the vaccine, but in future you might also be thinking of introducing another vaccine. So you can be able to broadly expand your cold chain capacity. Disease surveillance, again, as I mentioned, trying to make sure that there is integration of the disease surveillance. So in terms of the take-home messages, as we've had during the last 3 days, we're having more and more vaccines, and some of these are really very complex vaccines to deploy. And these are some of the other vaccines that are in the pipeline, and making sure that as we plan, we're also looking at the vaccines are in the pipeline and vaccines that we are likely to deploy in the future, so that we have a more comprehensive plan that accommodate not just the vaccine that we introduced at that time, but also future vaccines. and I really anticipating. I think we've talked about the RSV the vaccine and the target population for that vaccine. We have a lot of experience deploying the vaccine in children, but now the target populations are also changing. It's life course approach that we're using for deployment of the vaccines, and this is really not easy. We need to make sure that we are anticipating this. We are expecting more improved TB vaccines, combination vaccines, microarray patches. It was mentioned. I think these are some of the innovations. Also, the intranasal vaccines, which are also probably be coming up very soon. And as you think about the EPR, the essential program immunization, I think it's good to bear in mind some of these innovations and be able to plan for it going forward. Maternal vaccines, this is another area. We're just beginning now to deploy some of these vaccines. You know, maternal vaccination is a new area, and you just heard that the RSV was approved by or pre-qualified by WHO, so it's being deployed. It's coming up. The GBS is also on the way very soon. TB vaccines we just mentioned. So just to mention that, you know, oftentimes you hear people saying there are too many vaccines. What are we going to do? But we are not looking at the public health impact of these vaccines. This was a publication that I really find very interesting, which documented in terms of the public health impact. 800,000 lives are saved in the African region every year because of vaccines, and really the return on investment. I think for this paper is shown that for every dollar that you invest, the return on investment is about $44, which is really huge in terms of the benefit, public health benefit of these vaccines. So as I finish, just to say for each vaccine, there are resources. There are a lot of documents and information on the WHO website. This is an example of hepatitis B birth dose vaccine, some of the resources that are there. But for any of the vaccine, if you go to the WHO website, RSV, or any other vaccine, look at the position paper, look at some of these very important resources for every vaccine that can actually inform you as you plan to introduce a new vaccine. So these are very important resources that are available. So thank you very much for your attention.
Thank you very much, Jason. Are there questions online? All right. But before we take that, I want to ask a question, another honesty test. Because I noticed that some people went out, but for those of us who stayed, if you dozed during the presentation and you are honest enough to tell us, you'll get a gift now. Who dozed? Who slept? Who nodded during? All right, a gift for you. So shoot, Seppi.
Thank you, Elizabeth. We have 3 questions. The first one is from, I'll read the first one from Al-Haj. They're saying, thank you, Prof, for the insightful presentation. I would like to ask at what percentage in terms of vaccine coverage can we say for sure that we acquire herd immunity within the population? Because sometimes a waning coverage like the rotavirus example in South Africa. Yeah, so
I'm not quite full, but it's talking about the population immunity, right?
yeah.
I think this varies with the vaccine. For rotavirus vaccine, for example, in terms of the hand immunity, not very well established, but we feel that with the increase in terms of the coverage, right now the regional coverage for rotavirus vaccine is about 68%. So we are not at a level where we feel that there is sufficient hand immunity or population immunity for rotavirus vaccine. And the need for most of the new vaccines, even PCV, the coverage is still very low.
And then the other question is from Tambe. He's asking, what are the criteria to establish a disease, to establish that a disease is vaccine preventable? And he's saying, consider malaria vaccine and the Mpox vaccine. So,
yeah, that's a tough one. I think in general, any any disease where a vaccine is available then becomes a vaccine preventable to some extent. So the list that I showed, the list of the 18 vaccine preventable diseases, some of those vaccines are in EPI, others are not, they are used for mass vaccination, but you would say that is a vaccine preventable because there is a vaccine that can control and prevent that particular infectious disease.
I think Greg wants to expand on that. All right. Thanks, Epi.
No, I think besides what Jason was saying, I think the issue about when does a vaccine become a vaccine preventable disease, or when does a disease become a vaccine preventable disease? I mean, the point is it's quite simple. I mean, you know, all infectious diseases where there is a vaccine is potentially vaccine preventable. I think the issue really is, in terms of the country, is it a major public health problem in your country? And do you have the resources to implement it? I mean, we had a discussion this morning about hepatitis B, and somebody had asked the question about hepatitis A. All right. And hepatitis A is not part of any national immunization program on the continent, but it is now becoming a problem, certainly in South Africa. And WHO has recommended that when it becomes a problem and there's a change in the epidemiology of the disease, then you should consider implementing a strategy to prevent that specific disease. I don't know if that answers the question.
Yeah, but maybe also to add that for some of the syndromes, like for example, pneumonia, vaccine is just one of the interventions. There are other interventions, like for example, you know, antibiotic treatment and others. So it doesn't mean that if there is a vaccine, then we forget about other interventions that can control that particular disease or syndrome.
There was one other interesting benefit of vaccines that, and you just reminded me now, Jason, about pneumonia, with pneumococcal pneumonia, is that, you know, while vaccines are specific for diseases, there's also what is called the all-cause mortality effect of vaccines. So we know, for example, with respect to pneumococcal vaccine, it has an effect on all-cause mortality from pneumonia generally. It has a sort of, if you want to call it, a knock-on effect. The same with BCG, et cetera, et cetera. So that's an important point to consider. And when people start doing clinical trials with vaccines, especially in pediatric populations, one of the outcomes that they look at is not only cause-specific mortality or morbidity, but also all-cause mortality and all-cause morbidity.
Thank you, Jason. Amina wants to add to that.
Yeah, no, thanks so much, Jason. I really enjoyed the presentation. I think new vaccine introduction is indeed very complex in the implementation front. And it also creates a lot of opportunities. You know, it can divert a lot of the resources, a lot of the healthcare workers, but there are indeed opportunities when it's well done, it's well planned. It takes a lot of time, but it can create a lot of synergies for example, when we have malaria, hexa, measles, rubella, we have synergies across the second year of life platform, which strengthen, you know, catching up children who are missed, whether zero dose, under immunized, helping include them, identify them, reach them, and introduce them in the routine immunization system. So indeed, it's a unique opportunity to strengthen not only immunization system, but also other healthcare services. And I was wondering what has been done at a continental level, at a global level, in terms of knowledge sharing, whether with regional partners, WHO, AFRO, or even in academia across different countries to kind of showcase the success stories of some countries that, first of all, managed to sustain good coverage of the existing vaccine programs, but also succeed in launching new vaccines. And yeah, I had something else in mind, but yeah, I think there are a lot of opportunities. I know like for the rubella now we have, I think 15 countries that are yet to introduce rubella. And there's this idea of also looking at which countries have successfully introduced, what can they offer in terms of sharing those experiences to the countries that are still hesitant to introduce rubella, and what are the implications on terms of health impact, the cost effectiveness, should the country want to make that decision. Thanks.
I think that's an extremely important point in terms of knowledge sharing and sharing the experiences from one country to the other. And there are a number of opportunities to do that. The EPI managers meetings that are held every year, where the countries share best practices from, for example, we talked about Chad that has just introduced the vaccines at the same time for the first time. So in the next EPI managers meeting, I'm sure Chad will be invited to share that experience so that the other countries can see how this was done and also be able to evaluate whether this is really something that other countries could consider if it works well. So EPI managers meetings, we also have specific fora, for example, the African Rotavirus Symposium, which is held every 2 years. Countries and teams share their experiences about the deployment of the vaccine and also the monitoring of the coverage, looking at the zero dose and dropouts, and they share the experiences of different countries. So that, and also documentation is also extremely important. I've seen a number of papers from a number of countries in terms of deployment. Ghana, I think, published their experience in terms of deploying 2 vaccines at the same time. Tanzania, I think, also did the same. South Africa also have seen a number of publications. So documentation and also sharing the experiences is extremely important, and ensuring that we can also facilitate if a country is introducing a vaccine and another country introduce, we can even facilitate expertise from one country to another country to go and support these processes and really working as a team. So that's a very important comment. Thank you.
Any other?
Yeah, there's one more question online. Oh,
All right.
couple of questions. I'll take one. Nohan is asking, do you have any information about the introduction of the Tyvek vaccine in West African countries?
Tyvek, was it Liberia? I think Liberia did introduce, yeah.
So it's in Liberia?
Yes.
Okay.
It was like, yeah, in Zimbabwe, yeah. Yeah. Malawi, I think.
Yeah, there's Malawi,
Yeah.
Liberia, Zimbabwe. And I think Mali is thinking about bringing it in as well.
Thank you.
Thank you so much for the presentation. My question is regarding the gaps in both introduction evaluation or impact study. I have example from my country, like to provide evidence to introduce Rota vaccine, like we have a surveillance from 2007. Then we introduced the vaccine in 2011. Then I remember I participated in the Rota impact study, which had been done in 2016. And you mentioned that the evaluation studies should be from 6 months to 1 year, at least 2. So what is it to improve this impact post-evaluation studies?
Yeah, thank you very much. I mentioned the post-introduction evaluation that is supposed to be done within 6 months and 1 year. Mainly this one looks at the programmatic aspects, but also the sort of special studies to look at the impact of the vaccine and also the effectiveness. The country needs to have the system to be able to do that, and I think is extremely important. And what is recommended is that for the impact effectiveness, it's extremely important to have the surveillance existing before the introduction. Because for the impact, you're looking at the trends over time. So you're looking at the pre and post-vaccine introduction, so you can see the trends in terms of the disease burden. So having the surveillance before the introduction is extremely important and sustaining that. And obviously having the data tool, the tools to capture the vaccination history for the effectiveness. So that's extremely important. And I think for Rota, for PCV, and many other vaccines, there's a lot of data in the region in terms of the impact of rotavirus vaccine and PCV and others, and many of the countries here, Zambia, I think Malawi, South Africa, of course, with very good data showing the impact. And that can only be done if you have strong surveillance before and also continue the surveillance. Thank you.
Okay, thank you, Professor. It was very nice presentation. So my question is, so when you are purposefully introducing a new vaccine, you have time to plan for the training, for the logistic, for the cold chain, and for everything else. But when you are forced to switch from one vaccine to another, because the producing company have just stopped producing the vaccine. For example, in our case, recently we were obliged to change Rotarix to Rotacil. So the way of administration, the cold chain requirement is completely different from the previous vaccine. So it was an unexpected switch. So even though we were able to quietly manage the situation, it was very challenging. So in such situations, from your experience, what recommendation do you have?
Yeah, this is a tough one. I think the perception out there is that when you are switching the vaccine, it will go smoothly and, no challenges. But what you are sharing in Ethiopia is exactly what we've seen in Tanzania, for example. Tanzania also switched from Rotarix to Rotavac. Kenya also switched, Ghana switched, and we have a number of countries that have switched the vaccine. And Gambia also switched. I think the difference is that the earlier switches were actually better managed, because the Gambia, for example, the Rwanda switched from Rotatec to Rotacil, and they had enough time to switch because that was a country decision. What you've seen in the recent past is that the country has been forced to switch because of the global supply challenges. Ethiopia, for example, because the GSK, which has been making Rotarix, informed Gavi, WHO, and UNICEF that they will not be able to meet the demand of Rotarix. So a lot of countries were forced to switch within a very short time. So there were a lot of challenges. Kenya also had the same problem, Tanzania had the same problem, Ghana, I think, Burkina Faso, and a number of countries in the African region. In fact, what was really striking is that for these countries, they were given like 1 or 2 months to switch the vaccine from one product to the other. And remember, you're switching from a 2-dose to a 3-dose, and that's not easy. The vaccination cards, most of the countries in our region that have switched the rotavirus vaccine, they're still using their own vaccination cards. So the documentation, even we're talking about monitoring the impact, it becomes very difficult. We were in, I think it was Ghana recently, where we went there looking at the vaccination card. They are still using their own ones. And for that, you can't even be able to conduct the effectiveness studies properly because you need the vaccination history. So it's been quite challenging. The only way it can be done successfully is if you as a country, you're given adequate time to prepare, do the training. Because sometimes you assume even when you switch the vaccine, the health workers will be able to move from 2-dose to 3-dose very easily. But that's not usually the case. So where it's been successful is where you have had enough time to plan for that switch, just like planning for a new introduction.
Sorry, guys. The beauty of it is that Jason is resident. So please, don't say I told you, but feel free to kidnap him, in one of the tea breaks. So. Oh, okay, you wanted to add to it. Okay, if I give to the lady now, I'll be biased if I don't give to the gentleman.
In addition to what the prof said, the instant switching is also contributing to vaccine hesitancy, because the parents you're giving my child this vaccine, and which one is this? Because there is reduced time for sensitization of the communities, and this is affecting the the introduction of other vaccines on how the parents or how the communities or how the chiefs can receive the subsequent vaccines which are coming. Thank you.
Thank you so much for talking about that. That's my, for that, I'm going to give you a gift. All right, please make it short and sweet.
So thank you so much for your presentation. My concern has, can you throw a little light on introduction of new vaccine through EUL and the usual processes? Are there differences or what are the challenges that you think we should look out for?
For the normal pre-qualification and the
Like emergency use authorization,
Right.
where you're not going through the full phases of clinical trial, but there are safety data enough to introduce it into the system based on emergency, and the usual process where it had gone through all of the processes and things. Are there any difference in the introduction, or what are those challenge things we should look out for? Thank you.
Well, I don't know whether we have colleagues in the regulatory team, but the process is basically the same. You need to prepare the dossier with the relevant data from the clinical trial and provide that to SAGE. So the process is the same. The only difference is that for AUL, there are mainly vaccines that are used as part of response to an emergency or public health emergency or pandemic. But the process is the same in terms of the rigor that the SAGE looks at the data is exactly the same. It's just that because you are dealing with a pandemic, you're doing an emergency, and you have now a vaccine that could be an additional intervention to respond to that public health emergency, then you need to accelerate the process. But it doesn't mean that in terms of the review process of the dossier, there are any shortcuts. There are no shortcuts.
All right, with that, we've come. You see, I told you he will teach those who know and those who don't. Let's jam our hands together for Prof. Jason. All right. Thank you so much for that. You can take this way so that, all right then. So it's my pleasure, once again, I'm always having this pleasure, of introducing very interesting speakers. Our next speaker is a very, shall I say, confident, instilling speaker. I must tell the story publicly, because when I first joined VACFA and I was a little bit, quiffish, she told me, no, you can do it. It's no rocket science. You can do it. You can do it. And I'm like, okay, if Julie thinks I can do it. So I sat down and I looked at it, and I actually discovered that that particular assignment was no rocket science. So yes. And another thing I would like to say before I read the official bio, I told you, if they don't give me their bio, I'll make up stories, but the stories is not made up. The evidence will show itself. Julie was a tiny little lady the last time I saw her, but she has since grown up. You'll see what I mean in a sec. And then finally, after the talk, I'll tell you that. So Dr. Julie Coplin is a clinician researcher. Julie received a MBBS from the University of Sydney in 2007. After returning home to South Africa, she specialized in pediatric infectious diseases at the Red Cross War Memorial Children's Hospital at the University of Cape Town. She holds a master's in pediatrics and child health, global health from the University College London. Julie joined VACFA as a clinician, researcher, confidence instilling colleague in 2022. So I will want you to jam your hands together for my good friend, Dr. Julie Coplin. Now jam your hands together again for my good friend, Dr. Julie Coplin.
So, hi everyone, it's really nice to be here with you, and thanks Elizabeth for that introduction. As Elizabeth said, I worked with VACFA for about 2 years, so it's very nice to be back here with my old family, and thanks Adina and the organizing committee for inviting me. This is always a fantastic course, so I hope you guys are enjoying it. And just before I got up, Adina told me that I'm standing in between you and the waterfront. So I hope I'm going to be able to keep your attention here with me for a little bit longer. So before I start, just so that I get a sense of where we're at in the room, I just wanted to ask this question. So can you just think for yourselves, in your country, do you think that HPV vaccine should be prioritized as low, medium, or high? So not how much your country currently prioritizes the vaccine, but for you, how you feel yourself. So is there, and just to say there are no wrong answers here, obviously. I know just by being in the room, I know there are no anti-vaxxers here. I know we all pro-vaccine, but it's about prioritization. So I'm just wondering, is there anyone in the room for whom HPV vaccine is low priority, or you think should be low priority in your country? Just raise your hands. Okay, that's good. Anyone who thinks it should be a high priority? So almost everyone, fabulous. Anyone hedging on medium priority? Okay, so maybe just one or two of you who've got your hands up as medium could just say which country you're from and why you think medium priority. Is there anyone who?
She declined.
Anyone
So who
happy to
else
share?
raised their hands for medium? Yeah, the mediumites.
Thank you. I'm Hussein Abdraman. I'm from Niger. That's why I think that HPV is prioritized. Medium is now we have many issue, many challenge in vaccination. We are going to do the big catch up because we have many people or child who doesn't have their vaccines. And we are going to implement, we are going to done the malaria vaccine. Donc, I think that is very much. We are going to do so and so and so, but there is many challenge. That's why I think that we can prioritize that medium.
Thank you.
That's a very valid point. Shall we jam our hands together for him? There are so many competing interests for his country that HPV takes a medium priority.
Yeah, great. Okay, so what I'm going to do, for those of you who know me, you know I like to try and keep things simple. And what I'd like to do in this talk is just to answer 3 questions. But just to make sure that we all sort of starting on the basic level, similar level of understanding, we're just going to do a quick basics refresher on HPV, the virus, and then the vaccine. And then I'll try and answer these 3 questions. So the first one is, do we need the HPV vaccine in Africa? And I think we've all said yes, so that's great. The second question is, what is the current status of HPV vaccine rollout in Africa? Spoiler alert, not amazing. And the third question is, what are the barriers and how do we overcome them? So I'll spend quite a bit of time on that section just to try and give us a bit more of an update there. So let's jump into the basics. So the first thing is just to refresh our minds about HPV. So HPV, or human papillomaviruses, are extremely common viruses. They're DNA viruses. They infect males and females, and there are more than 200 types of HPV viruses. About 40 of these 200 can be transmitted sexually, and we categorize them as high risk. So the 16 and 18 are the ones I hope you've heard about, and low risk, which cause genital warts and laryngeal papillomatosis, which the pediatricians will be familiar with. So as I said, these viruses are extremely common, and it's expected that most people within 6 to 12 months of becoming sexually active will have been exposed to HPV. But most infections are asymptomatic and they resolve spontaneously. It's only when we have persistent infection with these high-risk types that we get precancerous and cancerous changes. So that's what we worry about. And the risk factors for persistent infection are early age of sexual debut, smoking, multiple sexual partners, and immune issues, so immune compromised. So HPV, as we know, causes cervical cancer, but it also causes cancer of the vagina, the vulva, the anus, the penis, as well as oropharyngeal cancers. So what about the vaccine? So I don't know why, but I love this picture. This is Harold Zurhausen, and he was a German virologist who back in 1976, so before I was born, he had already hypothesized the role that HPV played in causing cervical cancer. He then went on to isolate HPV DNA from genital warts and then from cervical cancer. But it took another 30 years before we got the first HPV vaccine. So 2006, Gardasil was licensed, 2007, Cervarix was licensed. We now have 6 licensed vaccines, 5 of which are WHO pre-qualified. They all have these virus-like particles, and they manufactured using recombinant DNA technology. So this means they're not live viruses. There's no viral DNA in the vaccines. They all have excellent safety profiles, limited adverse events. They're all highly immunogenic with excellent efficacy and effectiveness data. So in that table there, you can just see the 5 WHO pre-qualified vaccines, so 3 of which are bivalent, so cover just the high-risk 16 and 18. The Gardasil quadrivalent, which includes 6 and 11, so covering for genital warts. And then the Gardasil 9, which includes additional high-risk types. And then sort of sitting on its own there, Cervavac, which is produced by Serum Institute of India, not WHO pre-qualified, but also a quadrivalent vaccine. So let's move on to, do we need the HPV vaccine in Africa? I can see I don't need to convince anyone here that we need it, but hopefully just in case there's any doubts in your mind, let's just put those to rest. So this map, I'm sure you've seen lots of these maps, but this map is basically looking at which kinds of cancers cause the most mortality in women. And in pink is breast cancer, and orange is cervical cancer. So what you can see is that the vast majority of Southern and East Africa, cervical cancer is the #1 cause of cancer-related mortality in women. So let's dig a little bit more into the numbers. So these kind of heat maps, as you know, the darker the blue, the higher the incidence. So this is looking at age standardized incidence of cervical cancer. The dark blue that's covering all of Southern Africa is an incidence of 25 per 100,000. And this is data from 2022. And I've just pulled out a few countries below there so you can just see some of the numbers. And what you can see is that even though it's above 25, there's quite a broad range. South Africa has an incidence of about 33 per 100,000, Tanzania and Zambia up to 64 and 71. And then if you compare that to some high-income countries like Australia and America, you can see they've got much, much lower incidence. So now let's look at mortality. And it's not surprising that the countries with more disease have higher mortality. So that seems a bit obvious. But what I want you to do is look at the numbers below. So if we compare, for example, Tanzania to the United States of America, you can see that the incidence is about 10 times higher in Tanzania than America, but the mortality is almost 20 times higher. So what this is telling us is that not only are women in Africa more likely to get cervical cancer, but once they're diagnosed with the cancer, they're more likely to die from it. And that speaks to access to healthcare, late diagnosis, access to treatment. So really, we're talking about massive global inequities here. Every 2 minutes, a woman dies of cervical cancer. In 2020, in the African region, 100,000 women developed cervical cancer and about 70,000 died. In 2022, there were about 350 deaths from cervical cancer, and 90% of those occurred in low and middle income countries. So this is a marked global disparity. So this is what's projected to happen in the next 30 years, so between 2022 and 2050. So what you can see on the left is the projected incidence is projected to increase from 126,000 to almost 300,000. And then on the right, you can see the top line on that graph is incidence and the red line is mortality. So the mortality is tracking exactly as the incidence is, which is going to increase from an estimated 80,000 to almost 200,000 women per year. So we talk a lot about numbers, mortality, morbidity, but let's just think a little bit beyond the simple mortality figures. Who are these people that are dying? These are young women. They're women in their 40s, 50s, sometimes 30s. They're often breadwinners. They're central to their families and their communities, and they're often mothers and primary caregivers. And they leave behind young children. And what we know about maternal orphans is that they have higher prevalence of all-cause mortality, of mental health disorders, infectious diseases, including HIV, of teenage pregnancies, and school dropouts, which often leads into a cycle of poverty. So this comes from a paper which I found really interesting, which looks at, it's basically trying to quantify which cancers cause the most maternal orphans. And the colors are a bit similar, but hopefully you can see that that sort of second pinky purple color, that's cervical cancer. So with the exception of Northern Africa, the rest of our continent, cervical cancer causes about 1/4 to 1/3 of all cancer-related maternal orphans. So what are we going to do about it? In 2018, the Director General of WHO issued this call for action. And you can read what he said, but essentially what he was saying is, we already have all the knowledge and the tools and the cost-effective strategies to eliminate cervical cancer. We already have all of that. How do we get there? In 2020, WHO launched this global strategy to eliminate cervical cancer, which envisions a world where cervical cancer is eliminated as a public health problem. They've set the threshold for elimination as 4 per 100,000. And if you think of those numbers that we were talking about a few minutes ago, most of Africa is sitting well above 25, some up to 80. Okay, so we are way off this target. How do we get there? Through vaccination, through screening, and through treatment. So the targets that have been set for 2030 are that 90% of girls should be fully vaccinated with HPV vaccine by the age of 15. Screening and treatment are also obviously critically important, but they're outside of today's talk, so I'm going to just move along and ignore those. So let's get to question 3. What's the current status of HPV vaccine rollout? And this year I decided to put in a slightly more positive and optimistic map, because this map includes countries that have included or already rolled out the HPV vaccine. So those are the countries in green. In yellow, those are the countries that are still planning to introduce it. So it looks a bit better than the actual reality. But if you look at that continent, there's still lots of gray. So lots of countries in Africa where we're not even planning to introduce the vaccine. We've got 28 out of the 47 WHO member states that have introduced the vaccine. And let's look at coverage. So if you look at that graph at the bottom, the top dark blue line, the navy line, that's the coverage of 1 dose of vaccine amongst girls. And we're sitting at about 40%. So we're not even halfway to our target of 90%. So then the question is why? Why are we not doing so well with this vaccine? What are the barriers and how do we overcome them? So this is from a systematic review, from a paper that came out earlier this year, a few months ago, and it looks at the challenges and the barriers to HPV vaccination in low-resource countries. And I really like just the simple way they've divided this up into these 4 blocks. So they've divided the barriers up into policy issues, cost or financial issues, social determinants, as well as individual barriers. And I'll spend the rest of my talk talking about is these barriers. But instead of just listing all the issues that we have, because I think we can be quite good at listing all our problems, what I want to focus on is the advancements that we've made in the last few years that will help us to overcome these issues. So the first thing is I want to just draw attention to the impact of Gavi, the Vaccine Alliance. And there's lots of players in this space, but I think that they've really made a very important contribution. And this started back in 2011 when they agreed to this, they negotiated this price agreement of $4.50 per dose of vaccine. For anyone working in the EPI space, $4.50 still sounds a lot. It's still an expensive vaccine. But when we think of the fact that high-income countries at that time were paying over $100, you can see this as a paradigm shift. If these vaccines were costing $100, we would not be having this conversation. It would just not be possible. In 2013, they started funding demonstration projects. And again, I think this talks to the uniqueness of HPV vaccine because we've got a completely different target group that we're trying to reach. These are not young infants and toddlers who go to the clinic regularly. These are adolescents that in general don't come to healthcare facilities. So when we started, we didn't actually know how to reach these adolescents, and countries had to gain experience and knowledge of the best and most cost-effective ways of reaching this population. In 2018, Gavi started funding the full national introduction programs. And then 2 years ago, they recommitted to this revitalization campaign, which has got this amazing goal of vaccinating 86 million adolescent girls by 2025, again, with a huge financial investment. So the next major advancement from the last few years, which I feel like is all I spoke about for 2 years. So I'm going to skip over, I'm not going to go into too much detail, but if you're interested, we can talk about it in Q&A time. But this is an absolute game changer. So moving from a 2-dose to a single-dose dosing strategy for HPV vaccine. And what I wanted to show here is just that there were a number of studies over the last couple of years that have really shown us that a single dose of HPV vaccine is as effective at preventing cervical cancer as multiple dose strategies. And 2 of these studies were done here in Africa. So the first one is the Kenshi trial, which was done in African women in Kenya. And the second study is the Doris trial, which looked at immune responses to vaccine in younger girls, 9 to 14-year-old in Tanzania. And on the basis of all this data, WHO was able to change its position paper and update it with this alternative single-dose schedule. So they allowed basically a single-dose strategy to be implemented. So this is how we're doing in Africa with implementing single dose or moving to single dose. So that browny rust color you can see are the countries that have already moved to 1-dose schedules, and the blue, dark and light blue are still using 2-dose schedules. But if you just think about this, the number of vaccines that a country needs to fully immunize their target population has halved. That's an absolute game changer when we're talking about the cost of procuring these vaccines. So the next thing is what do we do about delivery strategy? I've already said we didn't used to think about adolescents and their healthcare. We didn't know how to reach them. We had to work it out. So we had to develop a whole delivery platform to vaccinate and reach these adolescents. And there were lots of questions. So the first question is, who do we vaccinate? Do we vaccinate girls only or girls and boys? And there's obvious advantages to vaccinating girls and boys. Not only do we also protect boys from HPV-related cancers that they're prone to, but we'll get much closer to herd protection. But the downside is cost. And our main aim at the moment is eliminating cervical cancer. And so from cost-effectiveness studies, vaccinating girls only is the current strategy. When do we vaccinate them? Is it based on their age or based on their grade at school? And the early study showed quite clearly that choosing a grade was a much more effective way at delivering the vaccines, particularly in school facilities, and it resulted in much higher coverage rates than targeting a specific age. And then where do we vaccinate? Do we vaccinate in schools or in health facilities? And again, there's pros and cons. So using a health facility is good because the health facilities are set up with the staff, with the infrastructure. But as we know, adolescents don't generally go to the health facilities. So the advantage of a school is that hopefully we've got lots of our target population in one place. We can do it in one time. The downside is it's much more complicated. We now have to work out how do we get the vaccinators and the vaccines to the schools on the right time, not interrupting the school system. So what are we doing in Africa? So here you can see that most of the countries are blue, which is a school-based approach, which has been shown to be the most cost-effective way in our setting. The rusty brown is a mixed approach, and there's 1 or 2 countries that use a facility-based strategy. So I put this slide in because I think it's important. Whilst a school delivery platform is definitely the most cost-effective, what you can see on this graph is the coverage rates in South Africa. So we launched HPV vaccine in 2014, and we went great guns. We had rates of almost 87, 85% coverage, and we were slowly drifting down to about 70%. And then you can see what happened during COVID. It is embarrassing. We got down to 3% coverage. And the reason I think it's important to put that up there is that we have to acknowledge in a continent like ours, where we are prone to instability, natural disasters, war, these systems need to be less fragile. So we can't have a vaccine program that just crumbles to nothing if there is, one of these events. These systems need to be more adaptable and more robust. Okay, so let's go on to the next block, which is policy and political will. So what about global advocacy? And I've really, in the few years that I've been interested in HPV, have really seen an increase in global advocacy around this issue. And I think this is just highlighted by the fact that earlier this year in March, the first ever Global Cervical Cancer Elimination Forum was held in Latin America and Colombia. And at that forum, there were leaders from all around the world, from ministries of health, development partners, UN agencies, and civil society. And the aim was to catalyze national and global momentum to eliminate cervical cancer. And at that meeting, there were huge policy, programmatic, and financial commitments that were made from governments all over the world, as well as lots of money. So that's great on a global level. What about on a regional level? And I think here again, the RITAG, or the Regional Immunization Technical Advisory Group, has taken a very strong position in Africa. And just last year, they issued a whole series of recommendations, including the adoption of single-dose schedule, extending vaccination to older groups and boys where feasible, prioritizing immunocompromised individuals, strengthening advocacy and communication efforts, promoting peer-to-peer learning for optimal delivery service and strategies. So again, I think this is critically important from a regional level, that we're promoting this vaccine on our continent. What about local advocacy? So I think it's obvious to everyone in this room, without strong political commitment by our national governments, we won't get adequate funding for these programs. It's essential to promote the coordination that's needed between the ministries of health and education, which is critical if we're rolling out this vaccine in schools. And I just wanted to say that the importance of local health champions can't be underestimated. So there are many countries in Africa where the first ladies have taken on HPV vaccine advocacy as part of their role. And I think this is really important. So the last 2 blocks on that chart were social determinants and individual barriers, which I've lumped together because there is overlap. And I think we've all lived through this not that long ago. So we've all lived through the COVID vaccine era. We've all lived through the vaccine hesitancy, the myths, the confusion, the chaos. I think for anyone in the vaccine space, it was a bit traumatic. And I think the fact that rolling out the HPV vaccine happens around this time has certainly not helped our coverage of the vaccine. I know you have a talk about vaccine confidence on Friday, so I won't go too into detail around vaccine hesitancy. But I think what we do know is that vaccine hesitancy in developing countries presents a serious barrier to HPV vaccine rollout, that parental beliefs about the appropriateness of pre-adolescent immunization limits the uptake of HPV vaccine amongst school-going girls, and that the engagement of community and religious leaders can reduce misinformation and vaccine hesitancy. So the knowledge and attitudes of healthcare workers, teachers, and religious leaders towards HPV vaccination are critical to the success of the program. If you look at these two little infographics, which come from the release of HPV vaccine in Sudan, just take the headmistress, for example, and she says, "I was very proud they chose my school in raising awareness about the HPV vaccine. It's so important for everyone." I think you can see if you have a head teacher who's got that attitude, you're far more likely to get good coverage in your school than if you've got a head teacher who's not educated and not informed and empowered around this issue. So I think that's also a lesson that we've learned, is that when we're planning to roll out HPV vaccine in our countries, it's absolutely critical to engage with all these key role players at the beginning. And then the last point that I want to finish on actually is the systematic review that came out earlier this year, which showed that good knowledge and a positive attitude towards the HPV vaccine in adolescent girls is associated with an almost 3 times odds of increased odds of vaccine uptake. And I think that in the vaccine world, we're so used to dealing with little children and babies and thinking about their mothers that we sometimes don't think about the actual recipients of these vaccines, which are young and adolescent girls. And I think that promoting education and health literacy in this group of young women is absolutely critical. And it's a very important step that's going to get us towards realizing our dream of eliminating cervical cancer. So just to finish off, I'm going to just summarize what we've done in this talk. And this is all you really need to remember. So the first thing was the basic refresher. We know the virus is everywhere. Persistent infection with high risk, 16 and 18 particularly, can cause cancer. All of the licensed vaccines are safe, highly immunogenic, and effective at preventing cervical cancer. Question, do we need the HPV vaccine in Africa? I hope everyone agrees. Yes, yes, and yes. So I've shown you that there's a huge burden of morbidity and mortality in women in Africa caused by cervical cancer. The current status of HPV vaccine rollout in Africa is improving, but just not fast enough. And what are the barriers and how do we overcome them? I think I've shown you that there are multiple barriers, which are particularly exacerbated by the fact that this is a unique target population for a vaccine, but that we now have a really good evidence base for cost-effective delivery platforms and vaccine schedules. And that with strong political will and global, regional, and local advocacy, we can overcome these barriers. Thank you.
Thank you so much, Julie. Any questions? All right. We'll start with the in-person first before we go to the online. Please make it short and succinct. We don't want to miss the blue and red bus to the waterfront.
Thank you so much for your presentation. I'm also a very big fan of HPV vaccines. I have 2 questions for you. Is there any research that has been done on whether the vaccines that are available are also protective against the other cancers that come from HPV? And now that we've switched to a single dose, how do we feel about the immunocompromised groups, such as people living with HIV, for example? Yeah.
Thank you. Yes, so the vaccine efficacy studies, and Adina, let me know if you want to add on to this, but the vaccine efficacy studies, particularly the early ones, were not looking at, many of them didn't look at cervical cancer as their outcome. They looked at persistent infections. So what we know is that the vaccines are effective against persistent infections, which is what causes cancer. So I must be honest, I haven't looked at the data in boys, but I think that it's pretty clear that the vaccines are effective against persistent infection with the high-risk HPV types, which would by default reduce the cancer, the other types of HPV-related cancers. With your second question around the immunocompromised, so that, I mean, is a big issue, right, particularly in our setting. And there is a study that's been going on in South Africa looking at vaccine efficacy of a single dose in HIV, a woman living with HIV, but the results haven't been published yet. So if you look at the WHO recommendations, they're still recommending a minimum of 2 or 3 doses in women living with HIV for the HPV vaccine. And that obviously is complicated because if you're vaccinating in a school system, how do you separate out those kids who are living with HIV? So it is complicated. And I'm hoping in the next year or 2, we will get the results to show us whether 1 dose is sufficient or not. But at the moment, we don't have that evidence.
All right. We have a lot of online questions, so we'll just take 3. Then you can answer them via the chat. Then I'll come back to you, Prof. And Amina.
So the first question is from Christelle. She's saying in Cameroon, the HPV vaccine is administered to both boys and girls at 9 years. And then she's saying that since they switched from giving, I guess, the HPV vaccine as 2 doses to 13-year-old girls, they've switched to the single dose at 9 years for both genders. So she's asking whether the HPV vaccine can be administered earlier than 9 years, and if not, why?
Yeah, so thank you. I think Cameroon is one of, I think, 2 countries, if I'm not mistaken, on the continent that has gender neutral, so girls and boys. The rest of the country the rest of the continent just vaccinates girls. And with regards to vaccinating earlier than 9 years of age, I think there are studies at the moment looking at that. But the thing that we need to know is the duration of vaccine efficacy, because what you want is an effective vaccine at the time that women are sexually active. And so at the moment, it looks like from immunological studies, it looks like the vaccine titers stay very high for a very long time. But we only have up until, the duration that the studies have been going. So we only have sort of, I'm not sure, it must be now sort of 15, 18-year data. And so we don't know what happens beyond that. I can try and show you, I don't know, can I just get my slides back up? Just show you. So I think it's important just to mention, while the slides come back up, we don't have an immune correlate of protection for the HPV vaccine as of yet, because the vaccines have been so highly effective. We don't have a sort of cutoff level at which we can say antibodies above this are protective. But what you can see,
Maybe, Julie, maybe because of time, you would like to engage that particular individual offline, since the person is online. Then,
okay, sure.
Yeah. So let's just take
the second
2 or 3.
Yeah. The
Yes.
second question, Elizabeth.
Thank you very much for your presentation. So I want to ask concerning sexual activity, because I know that's one of the criteria for the HPV administration. So when we go to school and we have girls that have had sexual exposure, though it's quite hard to determine that, do we screen them out? That's one. And then they don't get, and then is it also possible to have these vaccines for girls that are above 14 years if they're not sexually active? And what's the real role of sexual activity?
Yeah, great question. Thanks. So, I mean, what we're trying to do is to prevent persistent HPV infection. And ideally, what we want to do is vaccinate before there's exposure. But even in these trials that I've shown you, they looked at older women. So they were looking at women sort of 15 to 20 years, some of them. And the efficacy has been proven in that cohort of women. So in the Kenshi trial, for example, the women who were included had between 1 and 6 lifetime sexual partners, and it still showed over 95% efficacy of one-dose vaccine. So while the ideal is to get pre-sexual activity, even vaccinating post-sexual activity has been proven to be efficacious. There's also new data coming out, which is using the HPV vaccine in treatment of people who have HPV related diseases. So I think that's still an emerging field, but definitely you don't need to screen out the girls who've already been sexually active. Yeah.
Are we putting surveillance for cervical cancer in place as we are rolling out the vaccine to evaluate the impact? I know there were attempts for cancer surveillance in South Africa that was including, but I'm just wondering in general, like, I mean, with pneumocock or other vaccines, we are having surveillance to check what's happening.
Yeah, thanks, Prof. I think it's a great question, and I'm actually not sure what we're doing in South Africa. And I wonder, Dina, if you maybe have an answer for that. I don't have a perfect answer for it, but the only way that we know what's happening in terms of, I'm wondering, Prof, if you mean whether we are monitoring if after the introduction of the vaccine, we're seeing a decline. So the only way we can determine what's happening in terms of HPV prevalence and cancer is based on first for HPV our screening, and then for cancer registry. But I think we both know that those two aren't like the best, not only for South Africa, but for many regions. Our screening is really, really poor. We would hope that most women would be going for screening when they're eligible, but that isn't always happening. So that really impacts what happens for us on the region. But when you look at countries like Australia, the UK, they can already tell that after introducing the HPV vaccine, there's a significant decline in persistent HPV infection, significant decline in incidence of cervical cancer, which is just really positive for us in terms of the impact of the vaccine. So. Yeah, Prof, just to add on to that, I mean, I think the thing that's also important is looking at the high-risk types that are actually causing disease, because we know that 16 and 18 cause the vast majority, but there's quite good evidence that in Africa, type 35 is also quite a significant contributor. So I think in our setting, it's definitely important that as we vaccinate, we make sure that we're at least monitoring which types are causing the most disease. Yeah.
Thank you for the presentation. I am the Western Cape coordinator for life course vaccines. And in South Africa, we implemented the HPV vaccine to public girls in grade 4 public schools only. So not private schools, not girls who's being homeschooled. Because we focused on grade 4, some girls had their birthdays later in the year. So they were not eligible to receive the vaccine because they only had their birthdays after the second round of the campaign. Therefore, the decision was made in 2019 that we will switch from grade 4 to grade 5 in round 1, 2020. Therefore, the data that you have was for round 1, 2020 catch-up only. Because we were in lockdown with the COVID vaccine, during round 2, the last term, the schools were closed. So there was no campaign. In effect, we actually, with a switch, we provided catch-up to the girls. And from there, we vaccinated girls in grade 5, thereby getting all the 9-year and older girls. We've had good cover because we are aligned to a conditional grant, depending on the province's financial strength and other factors. I did find that for the Western Cape, during the COVID and thereafter, there was a drop of about 10,000 girls in the grade annually, and it started picking up slowly from about 2022. So the number of girls that were homeschooled also had an impact. As of 2024, we are switching to a single dose, and we are currently providing catch-up to girls up to the age of 14, and we are also starting with private schools. I do feel that having the HPV focusing on public schools only and the age group, it sort of stood as a stepchild to one side. Parents that were usually trusting our vaccines all of a sudden became not convinced. But as the girls get older, they then agree to it. Thank you.
Thanks. I agree. I mean, excluding private schools, I think is a big mistake. The idea that people at private schools should pay for themselves and organize themselves, I think just didn't work. And the number of, just anecdotally, the number of my friends who ask me about HPV vaccine for their kids, and they just don't know, and the schools are not very good at it. So I think it was a big gap. And I agree. I mean, ideally, we'd like to be vaccinating girls and boys. And I do think that will have a reduction in stigma as well, because I think a lot of parents are worrying about this as sexually transmitted disease, and we encouraging our girls to be sexually active. And I think if we kind of market it more as a cancer prevention vaccine for girls and boys, you have a chance of reducing that stigma. So I think they're really good points. Thank you.
Thank you so much, Julie. Just two last questions, purely online, and choose the easy ones, please.
So we have a question from Duncan. He's saying HPV is given to school-age populations of primary schools, and most countries have low coverages due to poor strategies in reaching the target population. So he's also saying integration of the vaccine into the school programs would be important. And I think this question is directed towards Gavi. He's saying, what is Gavi's basic strategy of improving demand and reaching the hard-to-reach populations with HPV?
Yeah, so I mean, I think it's a really critical point, and I didn't discuss much, but this whole move towards a life course approach, I think adolescent health is something that, again, over the last decade has become much more important. And I think we finally realized that we have to invest in adolescent health. And this is the first vaccine that targets this population. And I think certainly when Gavi was funding these demonstration projects, the plan was to tie HPV vaccine into other health promoting strategies. So in some countries it was deworming, others it was around sexual and reproductive health education. Each country had their own sort of thing that they did. And I think that's really critical, because as I said, these are new delivery platforms, these are new strategies of reaching a new population. And we have to be innovative and we have to promote it. Yeah, I hope I answered that question.
All right. Thank you so much, Julie. Shall we jam our hands together for Dr. Julie Cook? Beautifully delivered, excellently done. It's good to see you again. So, as we might have noticed, Julie is moving from one side of the epi to the other. Now it's my time to sign out, and I have 3 gifts. If you agree that the questions, the quiz questions were excellent questions, can I see your hands up? Okay, if you disagree, can I see your hands up? All right. So to the person who, pulled out such beautiful questions, and my beautiful colleague, Edina, step up and get your gift. For asking nice questions during my session. Yeah, bribery will get me everywhere. And also for my other beautiful colleague who assisted with the online questions. Something. Seppi. Thank you. All right, and with that, step up, girl, over to you for the evaluation. And thereafter, we jump the bus.
So we are done for the day. So we're going to do the daily evaluations. And I think the bus is almost ready. So if you guys can do it as quickly as possible, you guys will get your vacation quickly. Okay, 16 people in. Okay, so can I move on? Yeah? Okay. So what is your favorite food? It's been 3 days of very nice lunches. So what's your favorite food? What is Doro? Doro what? I'm very, huh? Ethiopian cultural, okay, okay, nice. And what is Amala? Huh? I mean, I don't know. It's Nigerian? Okay. Okay, we move on to the next question. How useful was the session on advances in local and regional vaccine manufacturing capacity and experiences from the mRNA Hub presented by Pia, Dr. Pia? Okay, we'll move on to the second question. The session that was presented by Lauren on meningococcal vaccines. Okay, so we'll move on to the third question, which session, which is on Hep B, presented by Michael. Okay, 2 more people. Okay, to the next, which was the second session for the day on maternal immunization. This was presented by Claire, Dr. Claire Cutland. Okay, this was answered fairly quickly. Thank you. The next one on HIV vaccines, which generated a lot of questions and interest for obvious reasons. Okay, so we'll move on. We have about 6 questions left. And then the session on traveling or travelers by Albie. Okay, so we'll move on new vaccine introductions by Dr. Mwenda. And then the last session that we just had with Julie. Okay, we'll move on to the next. List anything that you've learned that is applicable to all these sessions that we've had today. What stood out for you? Okay. The travel session is very popular. HPV, maternal vaccinations, okay. Okay, about 42 have responded, so we'll move on to the last question. How do you feel about the training so far after day 3? How are you feeling? Always good things, that's nice. But we'll also need to see those that are honest. Exactly, that's what I'm saying. We need to see the tired. Where's the tired? So after 3 long days, I'm expecting to see exhausted. Huh? That's good. Engaging. That's awesome. Thank you so much, everyone. I think this is, oh, okay. Dina has so one last thing. Tomorrow, can you please try to be punctual? Be seated by 7:45 so we can start on time. Is that okay? Okay, so go have a good time at the waterfront. See you tomorrow. The bus is leaving at 5:45, so. Oh, 4:45. Edina, you get me killed.